Hi everyone, I'm Saniya. Welcome and I hope you're doing well. Greetings from the United Kingdom. Okay, that's my attempt for a British accent. So in this video, I'm going to take you through a step-by-step example of how to run the Polygenic score PGS catalog calculator on the DNAnexus platform for the UK Biobank genomics data, the genetic data. Now you might be wondering, well, what exactly does this mean? So let's break it down. The UK Biobank is a huge research study with genetic and health information from about 500,000 people. It is one of the most important data sets in medical research today. And so the big question that we're asking here is, can we use someone's genetic information to estimate his, her, or their risk of developing a certain disease or trait? To make things concrete, I'll be using Alzheimer's disease as an example. Alzheimer's is a progressive brain disorder that affects memory and thinking. It's one of the most studied conditions in genetics and precision medicine, and it makes a perfect case study for this tutorial. And what is the PGS catalog? So the PGS catalog is short for Polygenic Score Catalog. It's a free open resource that collects and shares polygenic scores, PGSs, that are published by researchers all over the world. And a polygenic score is simply a number that adds up the effects of many genetic variants, often thousands of them, to give you one measure of a person's genetic tendency or risk for a trait. For example, instead of looking at one single Alzheimer's-related gene, a polygenic score combines information across the genome to get a fuller picture of genetic risk. So why is this important right now? And why is this video about working with DNAnexus important in general? Well, because recently, access to the UK Biobank genotype data is moving towards the DNAnexus cloud platform. And it's happening really soon. And I know a lot of researchers and people out there are a bit overwhelmed and nervous about the transition because now a lot of you may have had it stored on your local computers, machines, that data, but now no longer will that be allowed. It's all moving on the cloud. And if you want to use the UK Biobank data for your own research, you'll need to know how to run any type of genetic analysis, using UK Biobank data in this DNAnexus environment. So I'm going to help you with that. And even though our goal is eventually using the PGS catalog calculator, I'm going to be helping you get more comfortable with using DNAnexus. So by learning how to use the PGS catalog calculator on DNAnexus, you'll be able to apply polygenic scores to large data sets like the UK Biobank, replicate published findings, and extend these approaches to new diseases, traits, or research questions. And what you'll learn in this video and in this walkthrough, I'll show you how to set up and run a polygenic score calculation for Alzheimer's disease. I'll explain the key steps from preparing the data to running the calculator and share some common pitfalls and troubleshooting tips that can save you time. And I'll provide examples and explanations that are general enough for other diseases or phenotypes that you want to study. But here is the best part. We're going to be hopefully learning a lot along the way. And even if you're completely new to the UK Biobank, DNAnexus, or polygenic risk scores, this video will help you understand how genotype files are structured and used, what is a polygenic risk score, and how it's calculated, how to navigate and run analyses on the DNAnexus cloud portal, and it's all sorts of things along the way, not just the risk prediction, but even working with the basics in DNAnexus. And also, more broadly, how genetic research connects to predicting disease risk, the genotype to phenotype link. So whether you're a student, a researcher just starting with UK Biobank, or simply someone who's curious about genetics, this video will give you both practical skills and foundational knowledge. And finally, if you find this video helpful, please remember to like, subscribe, and share this video with friends or colleagues who might benefit, and buddies as well. And I'd love to hear your questions and feedbacks in the comments as you try this out for yourself. And again, all right, with that, let's dive in. And again, this is things that I've been discovering from my work that I thought might be helpful for you. and I really just hope it can make your lives easier as well. And whether or not you're calculating polygenic risk scores, if that's your interest or not, this video will be very helpful for you in many different ways, and I'll be organizing it in chapters so you can go to the parts that you find are necessary for yourself. And next, I'm going to tell you what are some of the few of many things I hope that you may learn from this video. So again, I'm Saniya Kuller, and this is a little bear is joining me, and this is a little moose. They're joining me on this journey, and let's get started. And again, don't forget to reach out with any and all questions that you have. So in this video, as I was mentioning, there are a lot of things we're going to be covering, and as they say, to infinity and beyond. But what some of the things I'm going to be covering, this is just some of them, just to give you a little flavor, but it includes this and beyond. So like navigating the PGS catalog, gentle introduction to polygenic risk scores and genetic scores in general, how to subset BGen files for a list of variants that you have, how to convert BGen to BED files, a Plink1 format, how to merge BED files across chromosomes, how to upload files using DX Upload, how to watch your jobs using DX Watch, using BGenX and Plink2 tools, using the DX toolkit and the terminal, running codes using Bash and Python. We'll be doing quite a bit of that. Running PGSC calc tool on DNAnexus to calculate these polygenic scores. An overview of the different genotype file formats, just going around the research analysis platform, looking at some of the UK Biobank genotype data and the cohort browser, working with the Swiss Army knife toolkit and beyond, and just working with jobs on DNAnexus. So there's a lot more, but this is just a little flavor. So whatever you think you can get out of this, I really hope it will help you in some capacity. Okay, so just a little bit of a background on the UK Biobank data. So its cohort is around 502,000 participants as of the time of this video in 2025. The recruitment was in 2006 to 2010 at 22 assessment centres across England, Scotland, and Wales. And the age of recruitment was 40 to 69 years. And the geography and ancestry are primary residents of the UK, United Kingdom. And the majority is of European ancestry, around 94 to 95%, with representation from South Asian, African, East Asian, and other ancestries. And there's phenotypic data like questionnaires on lifestyle, diet, physical measures, imaging like magnetic resonance imaging like MRI, DXA retinal, like health outcomes. And there's longitudinal follow-up, that means over time. And it's linked to the National Health Service, the NHS hospital records, primary care, cancer registries, death records. And so there's a lot of genetics and omics data, like multi-omics, like different types of omics. There's genome-wide genotyping for around 96 million imputed variants. There's WES, whole exome sequencing for around 470,000 participants, and whole genome sequencing, WGS, for a subset. And there are multi-omics data that are in subsets like proteomics, like Olink, SomaLogic, there's metabolomics, there's microbiome. And the use case for this data is for large-scale genetic association studies, for discovering biomarkers for different conditions, for disease risk prediction. We're going to be doing it in our example for like late onset Alzheimer's disease, and for therapeutic target identification. So trying to find what could be potential avenues to try to ameliorate, to lessen the impact of certain conditions and diseases. So it's all really helpful for drug target discovery and for trying to find out potential cures for different diseases. Research is all like a stepping stone. And so there'll be these two checks that we want to do. The first is to make sure that we can log in to the DNAnexus portal, the research analysis platform, the RAP for the UK Biobank. And the second is to please ensure that you have enabled the DX toolkit on your local computer or your server, wherever you'll be storing your local files that you want to run on DNAnexus. Just make sure you have the DX toolkit enabled, please. So the first check is going to be logging in to the DNAnexus Research Analysis Platform, the RAP. So that's what I'm going to show you right now. And this is just some little inside jokes about the Biobank. So now we're going to see if we can log in to the DNAnexus portal for UK Biobank data. So on your phone, you want to make sure, so this is my little phone here with my little cats. So you're going to want to make sure that you can go to the Okta Verify app over here. And on the Okta Verify app, it should be telling you some password or some code. And this will be like the verification code that you will use to log in to the DNAnexus portal. So let me show you how that looks. So over here, I'm on platform.dnanexus.com.login over here. And what I'm going to do is I'm going to enter in my username, which is my email, as well as my password. And then for the two-factor authentication code, that will be what I'll put here, which will be this code 759345. So let me show you there. 759345. Awesome. And now I've logged in to the DNAnexus portal, and this is the project that I will be working with. But I just want to make sure that you're able to access the DNAnexus portal. This will be what we're using. So this is the portal that we will be using for our projects. And you can see that on the DNAnexus portal, we will have like all sorts of, different types of data. And one of the things that I want you to know is that we will be kind of working in the bulk section with the imputation data. In particular, we'll be looking at the UK Biobank imputation from genotype data. Which I'll be showing you as well, but just wanted to make sure that you can access this DNAnexus projects platform. So that's the first check. Okay. And then for our second test and check, it's to log in to the DX toolkit on the terminal, and I show it from a Mac's point of view. But yes, just see if you can log in there. And I'll go through this in another video, but this is the link for the installation guide for the DX toolkit. And I know that DNAnexus has also put up some tutorials to help you out as well, and do reach out to me with any and all questions. But I also have this video that I'm working on for you guys on how to install and get set up with the DX toolkit. But basically, I'm going to show you live how you can log in to the DX toolkit on your local machine, like your computer, or if you're using a server for some high performance computing, from your university or your work. But basically, when you get into the terminal, you're going to write DX login over here, and then you're going to enter your credentials, your password, and a verification code. And that verification code you're going to be getting from there's the Okta Verify app, and you're going to click it, and you're going to get a password, a verification code that you'll put in, and then you will log in. And I'm going to show you what this looks like live right now. But this is the second, because we need to make sure that we can also communicate very efficiently with the DNAnexus platform from our local machine. So the next thing we're going to do is we're going to check out and make sure that we have the DX toolkit enabled so that you can log in and you can communicate with the DNA Nexus portal, the research analysis platform for the UK Biobank data. So what I will do, and this is on my local work Mac computer, is I'm going to log in to the terminal application using the DX login. So let me show you how that looks. Okay. So you see over here, what I have is I've clicked here on the terminal application, this one over here. And this brings me to my computer over here. And one thing I'm going to do is I'm just going to write DX login. And then you'll see that it gives me this sign. So acquiring credentials from this over here. And this says, what is your username? And my username for me is going to be my email. So I'm just going to type in my email over here. It's very similar to the login for the DNAnexus portal. In fact, it is the same thing. You put your email. I put my password in. I'm typing, but it will not show it here. And then it's also going to similarly ask me for my verification code here. So if you recall that for my verification code, I'm going to go here onto the Okta Verify app, and I'm going to see that the code here is 814275. So let me put that in over here. 814275. And then you're going to see that it tells me my current project, which is the same that you've seen on the research analysis platform over here. You'd see that the project tends to be the same as this project that we just looked at, IBD project Saniya. So you'll see that's the same over here, and it's in your credentials are stored here and will expire here, and I can log out over here. But this is what it means. And now I can also do DX CD bulk. And what you will see is DXLS. It's going to start listing everything that is in the bulk folder here in the DNAnexus portal. So what you'll also see here is that if we look at the bulk, we actually will see a lot of different folders over here in bulk, like activity, MRIs, brain MRIs, magnetic resonance imaging, ultrasound, whole genome sequencing, and so on. So this is just showing that from our terminal, we are able to access DX Toolkit. So this indeed shows that the second check is met. Yay! Yes, we are able to log in and access the DX toolkit using our terminal, and that's what we will be using going forward. And also, since we are in the terminal, what I will want to do for the ease is to also show you that the codes that I'm using are actually from on my desktop that I've created over here. So I have downloaded under my desktop the codes, and I've created the codes, and they're available in Saniya demo here on my desktop computer, So I'm going to click this over here, and this will be where I have the files that I want to work with. So what I will do over here is on my computer, I'm going to go and I'm going to do CD, change the directory. Actually, if I do pwd, it's just going to show my users. And this folder here, get info, this folder is in users Saniya, SK37 desktop. So what I want to do is I want to go to CD desktop, and then ls means I list it. Just like how I did this DXLS, it lists everything in this folder in bulk in the DNAnexus. The DXLS was listing all these little folders in over here. So similarly, what I've listed is that we have these 3 folders. We have the desktop original, we have the DNAnexus example, and Saniya demo. And I want to CD into Saniya demo, and ls lists it. So I have all these different files inside that I'm going to be working with you guys in this video tutorial. But this again just shows the second test, and I'm also navigated to the folder where I want to use the codes, so that will be easier in the subsequent steps. So I do pwd, and it will print the working directory, which is users SK37 desktop Saniya demo, and this is wherever your files are or your codes are. So that's what you have to consider. So with that, we're ready to get started. Okay, so this guy is as close to my little tubby as possible at this moment. So as you can see, that in the spirit of GitHub, the cat, that's my little orange dude, a little boy, Tubby. I'm his mom, and that's me here. So I'm giving an example for how we go through and do this analysis. And this is for my example for you guys for like late onset Alzheimer's disease and so on. It's just an example for how to do it. So I'm showing you some specific examples and walking you through it. And I will be posting the code on GitHub, so it's publicly available for you to follow. So it's for your reference, for reproducibility, for you to use it as a template. So please see the video description information below the video for details. And also PGS catalog calculator, the documents, they will shortly be uploading my code as an example for all of you. So when you check the website soon, that should hopefully have the tutorial that I'm also making for that, for the team at PGS catalog calculator. So please be posted. But below, I'll be pasting the link for the GitHub repository for you to follow along. So please do check that out. But yeah, the goal is that I'm walking through an example, my example, but I'm hoping that you can apply it for your data. It's just, in reality, it's a cartoon example. So you can apply it to your own data set and for your own analysis, and adapt as needed, and reach out to me with any and all questions that you have. So that's the cartoon of us. This thought is really cute. And so yeah, please do follow along with the cartoon example that I'll be using, and reach out with any and all questions that you have. Adapt it as needed. It's just a cartoon example for you guys. And when you're running PGSC calculator, you can add more more parameters and other things as needed and more advanced data sets. I'm just trying to make a cartoon of the real world examples that you may encounter. So in this tutorial, we'll be going through the following files here, as you will see, and I will be calling it the Saniya demo folder. It's saved on my desktop. We'll be starting with the PGS calculator _test.sh. Then we have these other steps that we'll be going through, simple starter.sh to create a set of folders as an example. Then extracting PGS snips.ipynb. It's a Python Jupyter notebook using Python to basically go through the score file that we have and create a bunch of text files, one for each chromosome, basically telling you which are the reference SNPs to extract for that chromosome. Then we'll be basically using that set of SNPs to extract for each of the the text files for each chromosome, and we'll be using the BGen files, like the genotype files on the UK Biobank portal. We'll be kind of subsetting those BGen files for the set of SNPs that we need, and then we'll be converting each of those subset BGen files to bed files using this step 4. Then step 5 will be kind of like merging all those like bed files for each of the chromosome, all those files into one set of files across those chromosomes. So one set of BIM, FAM, and bed files. And then we'll be running the PGSC calculator in step 6. And so we also have a sample sheet we'll be using for PGSC calculator, and the score file that we will be working with for which we will be computing the scores, the polygenic risk scores, which will be Alzheimer's disease. But these will be the files that we'll be working with. And I've been making them publicly available so you can follow along, and these are the files that you have to use to follow the tutorial. So yeah, we will be doing a lot of different things in this video, and here's a step-by-step process, Magic School Bus style here. Welcome to Magic School Bus. Okay, so for the genotype to phenotype prediction, right? So I'm going to show how we run the PGSC calculator test. So that's a test one, just to make sure that everything is kind of working and that we can at least run the demo that they recommend. So we're going to run this. Then I will show how we can download a human score file from the Polygenic score PGS catalog. Then the first step we will be doing is creating sample folders in the DNAnexus platform, just to kind of get the hand of things, get a handle over it. So yeah, this is our game plan. Lesson plan, as they say. And so we'll just be creating some folders. Step 2 is to extract the PGS SNPs, from the score file. Then the third step is to subset the UK Biobank, the UKB BGen human genotype files for the PGS SNPs that we have for the given chromosome. So we're going to go through the score files, and we have in step 2 already extracted the set of SNPs that we need for each chromosome, and then for the given chromosomes, because some chromosomes may not be needed for the score, which we will see. So we're going to subset the BGen files for just those SNPs that are needed for the score. So we're going to have reduced BGen size files, and we will convert these BGen files into the corresponding BED, BIM, and FAM Plink1 binary genotype files for the given chromosomes. And then what we'll do is, so then we'll have one file for each of the chromosomes, one BIM, one BED, one FAM file for each chromosome. And so we will be condensing them all so that we just have one set of BED, BIM, and FAM files. So that's what we'll do in step 5. So merge these Plink files across all chromosomes, for the chromosomes that are there. And then in step 6, we'll run PGS catalog calculator on the DNAnexus platform for our data, for our genotype data, and for our score file. And then I'll just show some additional downstream analyses that we could do. But this is just an overview of what you can expect. And along the way, we'll be kind of covering different topics that could be helpful for you. So now we're going to go through a test example of running the PGSC calculator function, just the test demonstration that they give us in part of the documentation for the PGS catalog. And so we're going to run this on the DNAnexus platform and see how that works. So yes, on the website, what they do is they show us, when we're getting started, that we are able to run this test profile, using Nextflow, PGS catalog, PGSC catalog, using this tool over here. And so we're just going to show, well, how does that apply for the DNAnexus platform? Like, how can I just make sure that it's running smoothly and that, I'm able to get something to run and to work, and I have that infrastructure. So I'm just going to show you that example. And it's always recommended whenever you have or are trying out any new tool, hopefully they'll have some mini demo that you can work with, something simpler, just to make sure that it's working as it should, that there are no bugs. Hopefully. So this is just the first check, just to make sure that it's working properly for you so far. And then we're going to get more into the specifics. Yes, we will. So what does this look like for the DNAnexus on the cloud portal? So this is a bash script that is designed to run on the DNAnexus platform, which is a cloud-based genomics analysis environment where the UK Biobank data is. And the goal of this is to check that the tool PGS catalog calculator, it works in your environment. So what the first line is it's saying, okay, it's a bash script for us to run. So you can see it ends in a .sh. And this will create a multi-line command that will later be passed into the DNAnexus Swiss Army knife app. I was always really obsessed with the Swiss Army knives. I think they're really cool. And they're very multifunctional, like so many things they can do. And this tool can do a lot of different things. So you'll see here that these are the different parts in the command. And we will run dx run Swiss Army knife. It's like the tool here. And then we have the command. This is the CMD that is going to be run here. So this is what each part will do. The first part here, this first line here, what it will do is it's going to download and install Nextflow, which is a workflow manager that is widely used in genomics. That's what the first line will do. The second line, this make dir /p, this local.bin, is going to ensure that the directory, this tilde /.local/bin exists where user binaries can live. Then this mv nextflow tilde /.local bin is going to move the downloaded Nextflow executable into this directory using this mv command. And so this mkdir is making the directory here, and then we're moving Nextflow there. And then this export path equals this area here. It's going to update the path so the system can find the Nextflow executable. So what we use this tilde $ path. It's escaped so that the variable expands later when the command actually runs inside the container. And then this last line here, this Nextflow run pgs catalog /pgsc _ calc. This is the function. /profile test docker. It runs the PGSC calculator workflow from the PGS catalog GitHub repository. It uses the test profile, which is a small data set on Saniya, some data, and it uses a Docker profile so that it runs in a containerized environment. So that's what this command is doing here in short. And then what we will do is we're going to run the command on DNAnexus. So this will use the DNAnexus Swiss Army knife app. Again, I think it's really cool, which allows you to run arbitrary shell commands in a controlled environment. And it has a lot of bioinformatics tools at its disposal. And we'll be using this tool, going forward for the rest of the examples here. It pretty much does a lot of things that we need. So this imd equals cmd command, it passes this command that I just went through into the application. The priority is high. It requests a higher scheduling priority on the cluster, and that will also make it a little bit more of an expensive job because we're trying to bump up its priority and money talks. So if we want the high priority, we have it'll cost a little bit slightly higher to run this job. And the tag demo equals PGS calc here, PGSC calculator. this one here, it is going to tag the job with this demo PGSC calculator. So it's useful for tracking. We'll see that as we monitor this job on the DNAnexus platform. And then this is the instance type here. I'm using memory 3 SSD1 version 2 X4. So it just specifies the compute resources. So a virtual machine that has around 3 gigabytes memory and 4 cores. And I'll show you how we can check out the instance types. But this is what this command is doing. So in short, what this command is doing, this shell script is doing, is it's running inside the DNAnexus platform. It installs Nextflow, runs the PGSC calculator test workflow, this tool using Docker, and ensures that it's all working correctly. And the computation is submitted via the Swiss Army knife app with specific resources and the priority. So that's what this code is trying to do in short. And we're going to run it using bash pgsc_calculator_test.sh, which I'll show you. So yeah, this is what it's going to do. We just have to write the bash pgsc_calculator_test.sh. That's the command we need to provide to the DX Toolkit terminal window. So now we're going to check that the PGSC calculator test demo works for us because I've been on the tutorial website. They mentioned to test it out and make sure that it's working. And that's what we're going to do right now. We're going to make sure that it's working for us. So what they do tell us is, as I was showing you, they were mentioning that we should run this command here. So I'm going to show you how we will do this in the DNAnexus portal. So the first things first is that we're going to go to the folder that I created, which is in my desktop, as I was showing you here. We have this folder that I have, and I load this in Visual Studio Code, or basically just any place that you can use to access and open the code. So I've opened up Saniya demo over here, and I've gone to the PGSC, so the C stands for catalog, calculator test.sh. And so now what I'm going to do is I'm going to run this. So I'm going to run this over here, and to do that, as you see over here, we are in the DX login in our terminal. So we are already in the Saniya demo folder. So to see that, I can see that I have the PGS calculator test.sh. And so what I'm going to do is I'm just going to do bash. And we see that this is a bash file at the top. It has this beginning, and it's in .sh, it's a shell script. And so I'm going to do bash, and I'm going to type pg as calculator_test.sh, and I'm going to hit enter. Yes. So the job ID has launched. This is the ID of my job over here. So what I'm going to do is, now I have typed this here, and I'm going to go to the monitor. And then I had just clicked refresh. And then what I will see here is I will click on the Swiss Army knife here, and I'm going to see that this is the command that was registered in the DNAnexus portal here. And these are the output files that we have here. We can see that these are all the different output files, including an aggregated scores.txt.gz, which will be the set of scores that we will use, that we get from this demo. So this just shows that what we get is, for instance, this is a file. We can download it if we like. And then I see over here that we get some scores for the Saniya example over here. And this is the denominator. That just means the number of variants times 2. And then we can look at the sum, which is what we will take. But this just shows us that the test demo example works and that we do get something in return. So this is just how you will run the test demo for the PGS catalog calculator on the DNAnexus portal. And that's just also an example of how you run a script using the DX toolkit here. We just did this bash command here. So this verifies that we are able to run the test example for this tool. Yay! So what is Nextflow? Well, Nextflow, it's a workflow manager. It's a framework for writing and running scientific data analysis pipelines. So when workflows become complex with tons of steps, large inputs, high performance clusters and cloud environments, it's hard to keep things reproducible, portable, and scalable. Just think of it like this. Normally you might glue together scripts like Bash, Python, R, Plink, and so on, with just some ad hoc shell scripts. I'm showing my example where I'm going through these 6 different steps, and that usually works for small tasks. But when workflows become complex, that's really hard, and Nextflow solves that. So we write our own analysis steps as small modules like processes, and we describe how data flows between them here. And Nextflow takes care of the execution by submitting the jobs, handling parallelization, and managing software environments, and resuming tests or runs even if they've failed, and so on. So that's really great, and just will keep on going. And why is it so widely used in bioinformatics? Well, I'll talk about the portability, reproducibility, parallelism, resumable, and community. So it's portable because it runs the same pipeline locally on a high-performance cluster or on cloud services, even like DNAnexus, Amazon Web Services, without changing your code, Google Cloud Platform. So it supports different schedulers, Slim, LSF, SGE, Kubernetes, AWS Batch, reproducibility. So pipelines specify the exact software dependencies via Docker, Singularity, or Conda. And it's possible to reproduce the same results years later on a different system. But parallelism, it automatically parallelizes tasks when possible. So if you're doing the same thing for in our case for different scores, it will compute them in parallel. You just specify a bunch of score files and it will handle the same task for each of the different score files, each of the different polygenic score traits. If you run an alignment step for 100 samples, Nextflow will launch 100 jobs in parallel. And if a job crashes halfway, it's resumable. So you don't have to restart everything. Just Nextflow run, resume, and it picks up from the failed step. And there are thousands of bioinformatics pipelines that are written in Nextflow. It's a community out there. Woo! The NF core community curates high-quality, peer-reviewed pipelines for common tasks, RNA sequencing, whole genome sequencing, metagenomics, and so on. And PGS Calc is one such Nextflow pipeline, and it's written by the Polygenic Score Catalog team. So how does Nextflow work under the hood? So Nextflow is written in Groovy, kind of like Goofy, the Mickey Mouse character, very cute. But you don't need to know Groovy. It's a JVM language, based In Java, you just write small scripts in your language of choice and describe your inputs and your outputs. So Nextflow is a workflow manager, it's a pipeline engine, and it makes these complex bioinformatics pipelines portable, reproducible, and scalable. In PGS catalog calculator tool, it is the engine that drives the PGSC calc pipeline. So you can see over here that we have, for instance, we've got an example mini workflow, thanks to, ChatGPT Insights as well. It shows its little example of a Nextflow pipeline here. So Nextflow figures out, it runs index genome first, this process, and then it runs a line reads, this process here. So once per input files read, input reads file, and then it parallelizes across samples if there are multiple FASTQ files that are involved. So you can just see this over here. Runs this process, and then it runs this. So yeah, we are not going to be writing a Nextflow workflow ourselves. That's not what we're going to do, but we're calling an existing Nextflow pipeline PGSC calc. And this pipeline knows how to take our, Plink genotype data or the formats that we've specified. There are 3 different current formats that PGSC calc supports, and a newer version would support more formats. And it knows how to, so it takes the genotype data, how to take a polygenic score file, with our weights, how to harmonize the alleles and variants, how to compute polygenic scores, and how to write out per sample scores. So for each of the individuals in our genotype data, each of those samples, what are their scores for that trait? And all we are doing is providing the inputs. We're giving the score file, the genotypes, the sample sheet, and we're calling this command here. In the example, in the demo file, the test demo, we're calling this command. And then Nextflow will then pull the pipeline from GitHub, download the required containers, execute the steps, and save the outputs. And so I also want to show you a little bit more about the demo file that we were using for the PGS calculator, just to make sure that that is working. So as I was showing you, that we were verifying that it works, and just wanted to show you that what was going on behind the scenes inside the log file for it, like when we were downloading Nextflow, just to give you a little bit of intuition. So over here, I can click here and I can go to the log file here. And you can see that it's, downloading a bunch of things. You can scroll down. So now this is where it sees that it is, it's downloading the files using 4 threads. So it's using the curl. This curl is a downloading thing, and it's downloading the Nextflow dependencies. It may require a few seconds. Please wait. So it's downloading Nextflow here. That's what's happening here with this command that we were mentioning. So that's what it's happening with the As you see over here, what we are checking out is this test file, the curl, this bash one. That's what we're seeing over here. That when we're running this one, and then followed by the bash, it's downloading Nextflow. Then we are creating over here these directories and moving Nextflow over there. And then ultimately, we are running PGS calculator, the code here, the test example. So it's just using test. Profile is used to install the workflow and verify the software is working correctly on your system. And the test input data and results are only useful as examples of outputs and are not biologically meaningful. So these are just some of the test information. And it's running it here. It's running. But the Saniya data set, as we're seeing here, it's running, and then it finishes. It succeeded in a minute, 15 seconds, great. And then we can see that it's, running all this here. It's uploading these files here, and that's what we wanted to test out. So yeah, that's just also showing you like what was happening behind the scenes, as we are running the PGS catalog calculator, like what is happening behind the scenes, and for the command, just to provide some more intuition there. So in this video tutorial, we'll be talking about the Swiss Army knife, the SAK, in the UK Biobank Research Analysis Portal, the RAP. So what is it? The Swiss Army Knife, it's a general purpose utility app in the UK Biobank. So when you think about it, I always find these, Swiss Army knives really cool. This one is very intense. It's got a lot of functions. And the way I think about it is it's kind of like, I have this little device here, and it's like a docking port. And what you can do is you can see it has like all the different, ports, HDMI, for VGA cables and whatnot, USB-C, Ethernet cords, and this is a USB connector. It's got all of this as well. So it's multifunctional, and it's got this little plug here that goes right into your computer. So it's just multifunctional. So, I mean, it may not have everything you need, but it kind of works for the most part, which is important. And that's kind of like what a Swiss Army knife does, It's going to have a little bit of everything. It has like knives, a little scissor, even a nail clipper too, which is really cool. A little magnifying glass, a screwdriver, little wrench. I see the nail clipper there. It's like really cool. So it's just a general purpose utility app in the UK Biobank. For most things that you'll need, it's going to have it. So, and these are really cool. Like, the Swiss really designed these awesome Swiss Army knives. I'm kind of obsessed with them. So you can think of it as your workhorse sandbox for quick, flexible analyses without building a custom app. And you know what, my brother once, when he used to be in Boy Scouts and we used to be going around selling popcorn, I would be really excited that if we sold enough, we would be able to win a free little prize pocket knives. That was really exciting when I was a kid, kind of helping him sell his popcorn so that we could rack up the points based on the sales and then buy these little pocket knives, which is really cool. Anyways, just some fun childhood memories. So yeah, it's just a workhorse. It's a sandbox for quick, flexible analysis without building a custom app. And it's available if you go here in the UK Biobank Access Portal. If you go under the under the tools right here. Sorry, if you go under the tools, it'll be right over there. So why it exists? In the research analysis platform, in the RAP, many tasks require command line utilities like, extracting SNPs, these single nucleotide polymorphisms, filtering samples, converting formats and whatnot. And building a DNAnexus app for every small task is slow and it's just overkill. So the Swiss Army knife, it provides a pre-configured environment with the tools you'll most likely need. So what's inside is it comes preloaded with bioinformatics software. So if you click the tools and you scroll down, and you check out the Swiss Army knife, it'll kind of give you more details on what it has, like its Plink 2, QC tool, BCF tools, SAM tools, TabX, for, reading different indexed files, R, Python, Unix, Linux utilities, awk, sed, grep, and so on. And it has input and output integration with the research analysis platform, the RAP project storage, and mounted UK Biobank datasets. So you can run single commands or script files, bash, R, Python. And so like typical use cases would be like extracting SNPs from bgen using QC tool, as we'll see. Converting between genotype formats like bgen to VCF to Plink or so on. Going back and forth between them, filtering samples or variants, quality control, QC exclusions, running small data wrangling jobs, joining phenotype files, reshaping tables, quick exploratory stats or plots in R and Python, preparing subsets of data before sending them to heavier pipelines. So the benefits is that it's versatile. It's got so many things it can do. Like in Spy Kids, how that watch could do so many different things. Spy Kids 2, The Island of Lost Dreams. It's time saving, so it avoids building custom DNAnexus apps. It's cost efficient, lightweight job. You build only for what you use. It's beginner friendly, so it's great starting point to get started on the RAP. So if I had to explain it, too long or didn't read, as they say, it's flexible. If you don't know which app to use, just start with the Swiss Army knife. If I just want to extract SNPs from a UK Biobank BGen file or exclude some participants and output to uplink format, I just say launch the Swiss Army knife, upload my SNPs that I want or my sample list, and run a QC command inside the Swiss Army knife tool. So yeah, I was mentioning that we tend to use different instances here on the DNAnexus platform when we're submitting jobs. And so if you go here to the documentation.dnanexus.com, developer API running analyses instance types, to this link over here, you'll be able to find a list of different instances that you can refer to in the dx command when you're running the code to perform your analyses. And it will tell you like how many cores each machine has. You can kind of see what you're getting into. And like, of course, it goes without saying that the more powerful the machine is that you're using, the greater the cost will be for your analysis. Otherwise, we would just all take the most powerful machine, right? So you can see the instance types, the cores, the memory, the storage, and then there'll be some part where it can mention the costs, hopefully. But these are some things that you can note, and you can specify the name in the dx command when you're running it. So some of these might look very familiar, the format, like what is a machine, what is its capability. So these are just for you to keep in mind that this is where you can find that information in case you have to perform very extensive analysis. You can use a more powerful instance type, and you can specify that in the dx command when you're running your bash codes. So now I'll give a gentle introduction to polygenic scores, PGSs, or polygenic risk scores, which are special types of polygenic scores. So as you can see, they're kind of like having the risk of disease versus the reference group. It's about a risk of having a certain disease. That's what makes it a polygenic risk score. So to begin with, I'll be talking about SNPs, SNPs, single nucleotide polymorphism, SNP. Okay, so these SNPs, they're the most common type of genetic variation among people. So a single nucleotide, A, T, C, or G in the genome, in the DNA, it's altered in a specific chromosome and a specific position. So a DNA sequence might change from AAGCCTA to AAGCTTA. So for instance, it could just be different, like the C is replaced by a T here in that example. And you can see over here, for instance, that instead in one individual, it's an A in this position, and the other individual, it's a G here. That's a single nucleotide polymorphism. In this other one here, it's a C in this position, a cytosine, and in this other person, it's a T. And they're pretty common. So they occur approximately once every 300 nucleotides. So across the whole genome, there are roughly 10 million SNPs. And most SNPs have no effect on the health or development. Some SNPs are associated with diseases, with drug responses, or with traits. So for instance, if you were to take individuals who have a disease and individuals who don't have a disease, get their genomics information and find out where their genetic variants are relative to a reference genome, you could then find that some of these mutations, these variants, are associated with the disease more than others, and others might not be. So these are really important because some of these SNPs will be key for predicting someone's a risk for having a condition. So what is a polygenic score, a PGS? A polygenic score is a general score for any trait, like high intelligence. It could also just be disease. Disease can also be a trait, like having Alzheimer's disease, or having schizophrenia, having heart disease, having certain cancer, right? So it's a genetic score for any trait. It's just in general. But a polygenic risk score will be a polygenic score that is focused on disease risk prediction. So you'll see that this is like low risk, this is high risk. It's like a bell curve. And a person's polygenic risk score is a statistical calculation that is based on the presence or absence of multiple genomic variants without taking environmental or other factors into account. So it's a single number that basically encapsulates and summarizes an individual's genetic predisposition to a trait or disease. And it's computed as the weighted sum of risk alleles across many genetic variants like single nucleotide polymorphisms, SNPs. So we find risk SNPs for a given condition, and we can use those to see if an individual has those risk SNPs, and what are the dosages. Do they have them on both the chromosomes from both parents, from just one parent, or from neither? Kind of sum up those risk variants, and then find out what could be their likely score for having the condition. And if it's a PRS, which is what we're looking at in this example, even though PGS calc can work for all sorts of traits, we're just looking at our disease risk prediction for late onset Alzheimer's disease in this tutorial video. So it'll be a polygenic risk score. So that's just a specific type of polygenic score. And many traits like height, body mass index, diabetes, schizophrenia are polygenic. They're influenced by thousands of SNPs. That also makes it very challenging to predict the etiology, the start, and what's causing the disease, right? It's very difficult. And that's why there's a huge need for precision medicine. There are thousands of SNPs that can be contributing to these traits that we observe. Why are certain people taller than others, and so on. And a polygenic score captures the combined small effects of many variants. So it's useful for risk prediction, for stratification of individuals, putting them into different groups, and for personalized medicine, also known as precision medicine. So it's a great day to score above the curve. Well, maybe if it's a polygenic score. I'm not sure if it's such a good idea, if it's for, disease prediction. I'm not sure. Probably not. You want to be under the curve, SpongeBob. So, as I was saying, a polygenic score, it's a number that summarizes a person's likelihood of having a given trait. And if it's a polygenic risk score, it's a number that summarizes a person's genetic risk for a trait or a disease. So think of it like a credit score for your genetics. The higher or lower the score, the more or less genetic risk you may carry. So how is it made? Scientists take results from large genetic studies like genome-wide association studies, where you have individuals and then, you have genotype them, and you find the individuals who have a condition and individuals who don't have a condition, and then you try to associate and find which are the regions in the genome where the differences are. You kind of align them to a reference genome, and then you see, it looks like on this position at this point, a lot of people who have that base instead of another base, if they have a G instead of a T, it seems that those individuals have a high likelihood of having that condition. So that could be like a risk factor from a genome-wide association study. So, we take these results from large genetic studies, genome-wide association studies, GWAS, and each genetic variant gets a small weight based on how much it influences a trait, and they are added up across thousands of variants to give an overall score here. So, a polygenic score is not one gene equals one disease. Instead, it's many small effects added together. So it's advanced analysis of our DNA beyond one SNP at a time. So you can't just say, you have this, like for breast cancer, it's usually the BRCA1 gene, BRCA2 gene. Those have high risk if something goes wrong with them. But as you can see here, and this is what these SNPs usually look like. They have these RSIDs, reference SNP IDs, in front of them. It usually corresponds to a chromosome number and a position and a base change, and like instead of having a C in a spot, you may have a G or something, for instance. So it's like a credit score, right? You have these, this is your genotype, this is your score, and then, you kind of compute what this is. And you can kind of find this genetic risk score. And these small effects, they come from, these SNPs, these single nucleotide polymorphisms, the tiny differences in DNA between people. Like, we're remarkably very similar to these monkeys and so on. So a lot of the DNA is shared evolutionarily. So a lot of us share a lot of the DNA. It's just this few bases that make us very different. So for instance, most people could have an A at a certain spot, but some have a G, and that's a SNP. So kind of taking somebody's genotype, finding what SNPs they have, and then we have like models that kind of associate with certain SNPs with the disease, and then we can kind of predict somebody's risk score for having it given their genotype. And that is what precision medicine fundamentally is, taking somebody's genotype and coming up with a predictive number of how likely they are to have a certain condition. So as I was mentioning, SNPs, they are a single base pair change. This is a reference SNP ID in the DNA sequence. So over here, you can see that at this particular position in the DNA, at this chromosome number and position, a person has a C over here. Kind of just zooming in along, this is the DNA over here. The DNA, if we're unwinding it, these are histone proteins, we're unwinding it, and we're looking inside the nucleus of a cell. And we're seeing, the DNA across the chromosomes, and then we can see that this cell belongs to a certain tissue, and in the human, for instance, and this SNP could be why an individual could be affected for a disease. For instance, if we do a genome-wide association study and look at the Manhattan plot over here, what we can see is that this SNP at this chromosome position 6, at this spot here, it's above the genome-wide association threshold of significance, which is usually 5 times 10 to the negative 8, because given that we're testing millions of SNPs. So this SNP is strongly associated with having increased risk of the disease or being affected, in people who have this SNP. So they can be common with a minor allele frequency greater than 5% or rare, usually a biallelic, 2 possible alleles at one position. And the importance is that some SNPs affect gene function or regulation. They can be coding found right within a gene itself. Genes are along the DNA, codes for genes. The RNA polymerase 2 makes a transcript of it, messenger RNA leaves the nucleus, gets translated by the ribosome into proteins, which do a lot of functions, right? So other SNPs are neutral markers, but they can still track inheritance. So it's used as genetic markers to find associations with traits or diseases. So these genome-wide association studies, they test millions of SNPs for association with phenotypes like the diseases over here. And there can be like linkage disequilibrium mapping to identify regions of the genome related to the disease. Functional studies like SNPs may disrupt protein function or regulation, and population genetics, they can be used to study ancestry, migration, and genetic diversity. So SNPs are the fundamental units of genetic variation, and they form the basis for studying the genetics of complex traits and diseases. So in this slide, you can see, the genotype here to the phenotype links over here. And they are the building blocks of the genetic analysis in various data cohorts. All of us, I'm a participant in that actually, UK Biobank, Our Future Health, so on. This SNP is associated with DNA base C at this position, and it's strongly associated with the disease based on this Manhattan plot. And there can be various analyses to determine the impact of this disease risk SNP on an organism. For instance, there's expression quantitative trait loci, EQTLs, that associate SNPs with changes in gene expression. So SNPs can impact phenotypes in various conditions that we observe. Basically, it's what are the differences between individuals that are pretty common. So single nucleotide polymorphisms, it's a one nucleotide difference in a particular position of the deoxyribonucleic acid of the DNA in a specific position. They're the most common type of genetic variation in humans. And so this Manhattan plot here, because it looks like the Manhattan, New York City skyline, that's why it's called Manhattan plot. It's from this recent genome-wide association study, GWAS, for Alzheimer's disease. And so what we can see is, this is genome-wide association study. We have a genome-wide significance threshold, a p-value of less than 5 times 10 to the negative 8, because we are testing millions of SNPs, so it's a pretty strict value. And you can see along here going across the chromosomes. And these are the negative log 10 p-values here for the association analysis of this SNP's association with Alzheimer's disease risk in cases, people with Alzheimer's disease versus controls. And as of now, this peak here corresponds to ApoE4, so the E4 version of the apolylipoprotein, has E2, E3, and E4 alleles. And the E4 allele here, it's the only known causal determinant for late onset AD, the largest known risk factor for late onset Alzheimer's disease. A lot is unknown, and researchers are still trying to figure out what are risk factors, SNPs, and variants for Alzheimer's disease. But in general, polygenic scores and polygenic risk scores, PRSs, they estimate an individual's genetic predisposition to a trait. That's what polygenic scores do, or a disease. So polygenic risk scores are polygenic scores that are focused on diseases, because the trait can be like intelligence, height, it could be something positive like a polygenic score. But a polygenic risk score is a polygenic score that is applied towards a disease. And so basically what these scores are trying to measure is how likely is the individual to develop a certain trait or a disease, based on their genetics, based on, whether they have certain SNPs or not. And it's calculated by combining the effect of many genetic variants like SNPs across the genome, across all the chromosomes, in our cells. So how they calculated for the polygenic risk score, we have the effect size of a SNP and then the number of risk alleles. So for each SNP version that you have, what is its effect size on the disease or the trait? In this case, the disease, because it's a PRS, but same thing for polygenic score. So what is the effect size multiplied by the number of risk alleles, like the number of alleles that you have that are of the risk allele. So I will show you an example, but this is sort of based on data from the GWAS, Genome-Wide Association study, and it can include hundreds to millions of SNPs, and I'll show you a very toy example, but that's real, that's just 21 genetic variants. But you basically take, for each SNP, its effect size, for a given individual, if it has an effect size of that SNP times how many risk alleles the individual has for that SNP. And then you keep on, it's a product, and then you just keep on multiplying, you keep on summing up these products, multiplying the effect size of a SNP for another SNP times the number of risk alleles for that SNP. Then it's like a weighted sum, it's a sum product here. So yeah, a SNP, a single letter change in the DNA sequence, most common type of genetic variation. Most SNPs don't cause disease directly, but when you study millions of them, some are associated with traits like height, cholesterol levels, disease risk. SNPs are like spelling differences in a book. Most people don't change the story, but some can change the meaning or give clues about the plot, So I'm from the US and I moved to the UK recently, and I can see that they spell things a little bit differently. Like realized, that's something I realized, is that I spell it REALIZED and they spell it REALISED, realized different, right? So it's a SNP in my definition, I guess. So, this Manhattan plot is actually from Alzheimer's disease, a 2021 study here, with so many cases, people with the disease, and so many controls. And researchers use a variety of metrics to link disease risk SNPs to disease genes. And ApoE4 is the only known causal determinant for late onset Alzheimer's disease. A lot of Alzheimer's disease metrics are still unknown. So linking back to polygenic scores, a polygenic score is built from lots of SNPs. Each SNP contributes a little bit, positive or negative, to the final score. Together they create genetic risk estimate for complex traits. And so a lot of the genetics remains unknown about Alzheimer's disease, but polygenic scores and polygenic risk scores, they estimate an individual's genetic predisposition to a trait if it's a PGS, or a disease if it's a polygenic risk score. So how likely is the individual to develop a certain trait or disease, and it's calculated by combining the effects of many genetic variants across the genome. So we have an effect size of the SNP from a genome-wide association study, and we multiply that by the number of risk alleles that the individual has. And it's based on data from genome-wide association studies that can include hundreds to millions of SNPs. So an analogy from one of my favorite shows of all time, SpongeBob SquarePants, is that a polygenic score is like a recipe, for making Krabby Patties. Pinch of salt, what are the different things that go into it. So, and the point is that as part of this recipe, this product of Krabby Patty, to make that, you have certain ingredients that come in. I'm sure like eggs don't just turn right into Krabby Patties, but you need a certain set of ingredients to make them, and to follow that recipe, the ingredients are SNPs. SNPs are what kind of makes the final polygenic risk score. The ingredients are what goes into a Krabby Patty. So similarly, the analogy is that a polygenic score is the recipe and whatever we're trying to make, whether it's a Krabby Patty, like Patrick is devouring it so much, and the SpongeBob, he's looking at Krusty Krab making the Krabby Patties, and the single nucleotide polymorphisms, the SNPs, are the ingredients that go into that Krabby Patty. So what goes into making a polygenic score are the SNPs. They're the building blocks of that score. So the recipe is a polygenic score, and it's kind of made up of a bunch of these ingredients, which are the SNPs here. A SNP, specified here, for what that position and that base pair. And what you have, the XIJ is a genotype for the ith individual, and the jth SNP, and it can either be a 0, 1, or 2 for the effect allele dosage, and I'll explain that next. But basically, what this beta is it's the effect size of that SNP from like a genome-wide association study analysis and so on. So what you do is, for those risk SNPs, you basically kind of find out, if the individual has those. Like if it has that genotype for that risk SNP, and you basically are summing across the risk SNPs and how much each SNP is supposed to contribute to the disease. So the SNPs are the tiny building blocks of genetic differences, and these genetic scores, they're the summary score we get by adding up the effects of many SNPs. And if this doesn't make sense, I'll be walking you through an example. But each polygenic score is calculated as the linear sums of the effects of these M variants. You're summing up these M genetic risk SNPs, based on the effect weight in the scoring file, beta. With this XIJ is this genotype for the ith individual and jth SNP, and this genotype is usually 0, 1, or 2. 0 means that you don't have both of the effect alleles. 1 means that you have one from one parent and one from the other. You have one effect and then one non-effect allele. And then 2 means you have both of the effect alleles, and the effect alleles are the ones that are considered to be contributing to the condition. So I'm going to show you an example of how we calculate, in this case, a polygenic risk score for disease. It's just a polygenetic score, but here it's a disease. So let's just see this example. So RSID, these are SNPs, reference SNP IDs. So we have a score file that has J equals 3 SNPs that contribute to the disease. So there are 3 SNPs that are considered to be contributing to the disease. This one, and this is just a toy example, so it's on chromosome 1 at this position here, and the effect allele is an A, and usually we also would have another allele, like a non-effect allele, but just for simplicity I've put the effect allele here, it's an A. And for the disease, it's considered to have this weight, this is the weight that it contributes towards the sum, towards the polygenic risk score. And this is this other SNP on chromosome 2. So these other 2 are on chromosome 2 at these different positions, and they're both a T over here. So you can see this one has the largest contribution to the disease, and this one has the smaller contribution, the smallest to the disease. And then we have the genotypes of 2 individuals, so the count of the effect allele. So 0 in the genotype beta means that you have both non-effect alleles from your parents. That means that you don't have this allele of an A, for instance, or so on. And 1 means from both parents. If you have a 0 for the genotype, then it's from both parents you don't have the effect allele. So for instance, what we see over here is that for these 3 SNPs and these 2 individuals, we see the individual one's genotype is a 2, which means that for this SNP over here, they have both effect alleles of AA from each parent. So for this parent, for this guy, it would be an A and an A for this person here. A and A for this point here, for this person. It would be an AA over here. Sorry, that came out a little bit messy, but the thing is, it would be an AA here. And then for this individual, they have a 1. The same person has a 1 for this SNP here. So it means that they would have a T from one parent and the other non-effect allele from the other parent. And they have a 0 for this one, which means that for this SNP, they actually do not have this effect allele T for, in their genome. They don't have it at this chromosome 2 at this position. They don't have a T on either of their chromosomes, because we have one set from our maternal from our mothers, biological mothers, and one set from our biological fathers. So that means that they don't have the effect allele. So how many of the effect alleles do they have? And usually we would know what the non-effect allele is, but it's not mentioned in this example just for simplicity. Whereas for this individual, they have a 0 here for this SNP. So that means that for both parents, they do not have an A at chromosome 1 at this position here. And similar to the first individual, they have like 1 of the T alleles at this position from one parent, and from the other parent, they do not have that. They have the other one. And lastly, for this one, it means that for this individual at chromosome 2, at the position 234,068,476, they have a TT at that spot, 1 from the biological mother, 1 from the biological father. So this is the genotype data that we have. And this is the polygenic risk score calculation formula. So it's basically saying that these are the weights that each of the different SNPs contribute towards the disease or the condition. If it's a polygenic score, it's the same formula, but this is just like any trait. It can be a disease or not. So a polygenic risk score is just a specific type of polygenic score that is focused on a disease. That's what it is. It's just a specific type. And since here I'm just showing a disease, it's a PRS. The same thing is for a PGS in general. It's the same thing. It's just that we're measuring some trait in general. That's what a polygenic score is. So all we're trying to do is these are basically counting the number of effect alleles that we have. And we know that for every effect allele, this is you have from each parent, this is how much it contributes to the disease. So basically, what we are doing then is we're combining the 2 tables into one. We can do that. So for the SNP, we have the effect allele, the weight, and the individual's 1's genotype and individual 2's genotype. And all we're doing is this is just telling us for every effect allele that you have of this type, for this SNP here, if you have 2 of them, you multiply it by 2. If you've got 1 of them, you multiply this by 1. If you've got none, you just leave it at 0. So you're basically doing for this individual one, based on this formula, you're taking the genotype of the individual and you're multiplying it by the effect weight beta. So for individual 1, for this SNP, here we're just taking the 2 and we're multiplying it by the 0.166 and we're getting a 0.332. That's what we're getting here. That's how much they have based on this SNP and its importance and their genotype. For the second SNP, we take this 1 and then we multiply it by this 0.199 because they just have 1 copy of this from one parent. If they had a 2 here, it would be multiplying this by 2. So we just take the genotype here and we multiply it by the effect weight. So this times this. And we'll note that it's the same for individual 2 because they also have a 1 same effect weight. So it's the same thing here, 0.11 and a 0.199. And lastly, for this person, since they don't have this effect, they don't have this particular effect alleles, it's just going to be 0. So no contribution of this SNP to their risk score. So it's a 0 here. And all we do is we just sum them up. So this plus this plus this is equal to 0.531. Similarly, for the second individual, we take this this SNP here, multiply it by 0. They don't have either of the effect alleles from either parent. So it's a 0, no contribution from this SNP. Again, this is 0.199. And this one here is just a 2 multiplied by this value here. They have both effect alleles and for this SNP, and this SNP has this contribution to the disease. So it's 2 times that. And we add this up. And you can see that it's a 0.353. So this is individual 1's polygenic risk score for the disease. It's 0.531, which is higher than individual 2's polygenic risk score for the disease, which is 0.353. So individual 1 has a higher risk of the disease, a predicted risk, than individual 2 does based on this toy example. So that's how we compute a polygenic risk score. We take the genotypes respectively and multiply by for that given SNP, and we multiply it by the respective weight. And the genotype is just measuring how many copies of that effect allele do you have, that risk allele, or so. So yeah, basically, each SNP has an effect size from a genome-wide association study. The individual's genotype at each SNP is multiplied by that weight, the effect size, that contribution of that SNP, that's usually given by that beta, that weight of that SNP for the disease. And then, if it has a higher weight, it's a bigger contributor to the disease. And then you add it up across the SNPs, those weights, and then you get a polygenic score. And if it's for the disease, it's a polygenic risk score. Otherwise, if that score is just like height, intelligence, body mass index, and so on. And the case can be so various. It can be, are you a morning person or not? Like your likelihood of being a morning person, having insomnia, being risky or risk averse, right? So when we compute the polygenic risk scores, we can kind of put them over here, and this is like a standard z-score where, in the middle it's like a bell curve where it's like, oh, I cannot draw. Let me show you, for instance. It's just going to be, and this would be like a 0 and like a 1, a 2, standard deviations away from the mean, and a 3, so it's accented at 0 if it's a z-score, and so on. And then on the other side it would be like -1, and so on. It's just like -1, -2, -3. And you can see that, if an individual is in the 95th percentile, they're in the highest genetic risk group, based on their z-score. They are extremely high predicted risk for having a certain condition, like in the disease in particular. So yeah, this is just a toy example for how we can compute like a polygenic score or a polygenic risk score in the case that this is a disease, for instance. But we're just going to be expanding on this idea using the PGS catalog calculator for real-world disease or a real-world trait, and kind of how we can use that to compute this. It's sort of, this is the kind of like the idea behind the scenes on a very simple level, and then from there is just a little bit more complicated with the real-world data, with a lot more SNPs and different genotypes aligning strands and so on. But this is just the general idea as well of how you can compute a score for the disease, and we're going to be running this using the PGS catalog calculator tool. So to compute these polygenic scores, we'll be using the Polygenic Score Catalog PGSC calculator tool here. So this is the workflow. We'll be using this function here. This is the GitHub here. And it's based on this recent paper published in Nature Genetics called Enhancing the Polygenic Score Catalog with Tools for Score Calculation and Ancestry Normalization. So it says that these PGS scores have transformed human genetic research and have numerous potential clinical applications. So they present recent enhancements to the Polygenic Score Catalog and highlight this calculator. It's an open source, scalable, portable pipeline for reproducibly calculating PGSs. So it's useful across a lot of applications, and we'll be using that here in this example to compute the polygenic risk score of an individual developing late onset Alzheimer's disease. And currently, there's no tool in the UK Biobank for using the Polygenic Score Catalog calculator. So I had to find a workaround, which I'm showing in this video, for how we can run this tool using the DNAnexus platform. And this can also be a guide just in case you have other tools that you want to run that are not readily available in the DNAnexus UK Biobank platform. This is sort of like how you can run those tools exactly, like how you can download them, how you can run them in the DNAnexus platform. So even if you're not interested in running PGS catalog calculator, you can kind of use these concepts, hopefully, and the example codes to extrapolate and see how you could run your codes, potentially. So I hope it can help you. So you might be wondering, why not use Plink2//score, the tool? And we do use Plink2 in this video for other things, so it's no hate against Plink2 at all. Because we do use it for other things in this video, but it's very, very powerful of a tool. It's a very powerful tool. But you might be curious what PGSC calc does that Plink2 score doesn't, and so they've written this part in the PGSC catalog calculator tutorial part, and they say that we use Plink2 internally to calculate all scores, but offer some extra features. So this is what they've mentioned here that they do that is a bit more advanced than what Plink2 does. So they match the variants from scoring files, often from different genome builds, against the target genome using multiple strategies, taking into account the strand alignment, ambiguous or multi-allelic matches, duplicated variants, matches, and overlaps between datasets with a reference for ancestry. So if reference and alternate alleles are mixed around across datasets, or the strands are different, the DNA strands, it does all the matching. If there's flips and whatnot, and in other types of inversions and DNA strands, it handles that. One important output is an auditable log that covers the union of all variants across the scoring files, tracking how the variants match the target genomes and provide reasons why they are excluded from contributing to the final calculated scores based on user-specified settings and thresholds for variant matching. From the matched variants, the workflow outputs a new set of scoring files, which are used by Plink2 for scoring. The new scoring files combine multi-polygenic score in a single file to calculate scores in parallel. These scoring files are automatically split by effect type across duplicate variant IDs with different effect alleles, and then the split scores are aggregated. The pipeline also calculates genetic ancestry using a reference panel. The default is 1,000 genomes, handling the data handling, variant matching, derivation of the principal components analysis space, and projection of target samples into the PCA space using robust methods. So they implemented here. And scores can be adjusted using genetic ancestry data using multiple methods. So they have this here. So they have a summary of all the benefits here of why we should use it. And I will be computing polygenic scores using the Polygenic Score Catalog calculator going forward. But yeah, Plink2 is also great, but this is sort of building on it. So now I'll show you how we download these polygenic scores or these polygenic risk scores from PGS catalog. How do we do that? So what we're observing here is that in PGS calculator, what we have is scoring files that have this following format, where it says PGS catalog scoring file, and it will tell you, where to find additional information, the format version, the polygenic score information, the PGS identifier that it has, the name, the trait reported, mapped, ontology trait ID, the EFO, the genome build, the assembly like GRCH38, which refers to HG38, GRCH37 refers to HG19. They do have another video going over these concepts. And then we also see the variance numbers, the number of variants listed in that file, that PGS file, the weight type, and if it's beta, if it's odds ratio, if it's HR, the PGS publication identifier, citation, license. So these are some of the headers that we would expect for typical score files here, and they usually led with RSID reference SNP IDs. So that's fairly common. So we can go to the PGS catalog, and then we can click on the polygenic scores. And then we'll see that there are a lot of different scores here that we can see. For instance, we can look at this one here, PGS 000018, which is for coronary artery disease, and it has like 1,745,179 variants here. That's a lot of variants for this heart disease, for the coronary artery disease here. There's a lot of variants that they've identified. And we can click on that, and we can see that its name is meta-GRS, coronary artery disease. And it is original genome build, that is HG19 over here. And this is when it was released in the PGS catalog. It's by Mike Inoue. And then we can download the score here and get the variance and weights data file for this. And we can click this one here. The harmonize would just be trying to do the lift over from HG19 to HG38, but the current, it's okay if we are using like genotype data from HG19, we can use this one here. So we can download this guy over here. It clicks allow. And then we can see that it is a zipped file, and usually in Macs we can just quickly unzip it, and on Windows computers you can write gunzip, G-U-N-zip, G-U-N-Z-I-P, and then gunzip the file, or you can extract the contents from that scoring file. And you can see there are like a lot of SNPs here if we look at the scoring file, and it has like the header format that I was just showing you. It has the scoring file information, the version, the score, all of this information here in the publication ID, and it has the SNPs, the chromosome number, position, effect allele, other allele here. So this is effect allele column, other allele is this other column that is not associated with the with coronary artery disease, and then effect weight, where a positive effect weight means that it is leading to increased risk of coronary artery disease, and a negative effect weight, for those SNPs that have negative effect weights like these ones, these are really good because they are associated with a lower risk of developing coronary artery disease. So there are a lot of SNPs here. It's huge, and I'm sure in other videos I could kind of go through this, but I want to focus, instead of having 1.7 million SNPs, I'll just focus on 21 just to make it a little bit simpler. But you can see there's a large scope of variance for computing, these polygenic scores. So, this has a lot of variance, and, we can keep scrolling down, but this is just a little snapshot of what this looks like. But yeah, you'll also sort of see how, for this example, they have effect allele and other alleles. And then in the simpler example that I will show, they don't have the other allele mentioned, but that just really depends. But they do have the effect allele and effect weight. But some score files will have effect allele and other allele information. Some may just have effect allele only and effect weight. I just wanted you to see that. So in this example, I'll be working instead on a simple score example, which is so much simpler. And it is the polygenic score given with the ID 53 here. So this is like the link, and we'll be going through downloading it and all of that. But essentially, it's for late onset Alzheimer's disease. And it is from a study from 2019 for 21 genetic variants, these SNPs, using the HG19 GRCH37 human reference genome. So Alzheimer's disease, it's a progressive neurodegenerative disorder, and it's the most common cause of dementia in older adults. So it's a brain disease that causes memory loss, cognitive decline, and behavioral changes. So Alzheimer's disease has two main forms. Early onset, which is like when you're younger than 65 years, it's usually rare. And it's caused by strong mutations in specific genes like APP, PSEN1, PSEN2. And what we're focusing on here is late onset Alzheimer's disease, or LODE. It's the most common form of Alzheimer's disease, usually after age 65. And so Alzheimer's disease has these hallmarks, which is usually the buildup of amyloid beta plaques between the neurons in the brain, and these tau protein tangles that are inside the so these are plaques between neurons, and then there are these tau protein tangles that are inside neurons. So the plaques between neurons and these tangles inside neurons, and there's like brain shrinkage, especially like the hippocampus, which has our memories in the cortex. And so some of the symptoms are like memory loss, confusion, trouble with problem solving, mood and behavior changes, loss of independence, and there's no known cure. But some medications are out there. So it's a progressive neurodegenerative disorder leading to memory loss, cognitive decline, behavioral challenges. And it's sad because you start forgetting. People start forgetting and essentially regressing, becoming like babies again. So up next, we're going to show you how we downloaded our score file for late onset Alzheimer's disease from the PGS catalog, the polygenic score catalog. So let's kind of dive into it and see how did we download those scores, and just kind of show you a little bit more about it. So yeah, let's dive in, little guy. So now we're here on the pgscatalog.org, and we have these polygenic scores, and it was refreshed as of early August this month. So we're going to go here to polygenic scores, and we can browse through different traits here. And the one that I was looking at is related to late onset Alzheimer's disease. And here you can see that there's so many variants that are associated with these different traits and diseases. So what one we chose is the one that is the 53, which is on the next one here. So this is it here, G et al. Neurology, Alzheimer's disease, late onset. And so I used it because it has 21 variants. So it'll be like a little small case for us to use. So we can see that the original genome build is GRCH37. And then we can download the score here, which is where we go. And then we click this one here, and then we allowed it. And then basically what I do is I take the score file that we will get now. which looks just like this. Over here, I mean, the two is just because I've downloaded it quite a few times just to like make sure it all works before I made this demo. So that's the thing, and Macs automatically gunzip that compressed file because again, we can have lots and lots of variants, so it compresses it. So what we do now is what I do is I just sort of take that and I migrate it, this text file. I just move it over to this score file folder that I have in Saniya demo. So we just have this score file here. And you can see again, these are the 21 variants that we have. These are RSIDs, reference SNP IDs. Just to make it a little bit cleaner for you to understand what it looks like, let me show you. If we throw it in and we thrash it into Excel, I don't recommend doing this when you have like a lot of variants. This is just because it's 21, so it's a little bit easier. So you can see reference SNP ID, chromosome number, position, effect, allele, effect, weight, and OR is for odds ratio. So this is the score file. Yay! And I've moved it into that folder for easy reference. Because I'm going to show you in step 2 how we organize that score file into separate files. So Alzheimer's disease, it's a form of dementia, you know, as I was mentioning, memory loss, cognitive disability, start forgetting, memory start fading, people start forgetting aspects of themselves. It's the 6th leading cause of death in the United States. Neuroinflammation, like the role of the immune system, it may play a key role. I've written a couple of papers related to that. The heritability of Alzheimer's disease is rather high. It's around 80%, and there are various phenotypes out there that measure the extent and severity of Alzheimer's disease. So we can typically look at Alzheimer's disease progression from initial to moderate to severe stages. You know, there is resilience, psychological, that even if you have like Alzheimer's disease pathology and brain regions shrinking, you still kind of like have that cognitive resilience where you're not really showing signs of dementia. And that usually can show that if you keep up with your cognitive skills, you know, reading, learning, in socializing, playing board games, doing things that keep your mind healthy, eating well, maybe you could stave off some of the genetic effects of and risk factors for Alzheimer's disease that could happen. So that's what psychological resilience is, and people are studying that, like how can you kind of deter or delay the effects of Alzheimer's if it's meant to be. And then there's epigenetype, where the E4 alleles are the huge and the largest known risk factor for late onset Alzheimer's disease, though a lot is still unknown. And the CRED score for there's neuritic plaques, there's mini mental state exam scores to see how well you can do on like thinking and recall and memory. So in early Alzheimer's disease, what we typically see is mild memory impairment. Especially like for recent events, difficulty with planning, problem solving, word finding, and then as it progresses, you know, confusion, disorientation, impaired judgment, behavioral and psychiatric symptoms, depression, agitation, hallucinations, and then late is severe memory loss, you know, dependence on care, you know, need full-time treatment, loss of motor function, incontinence, not able to go to the bathroom. So for instance, you can see this grandma here, like let's say that she's age 77, she's been healthy most of her life. At first she starts forgetting small things, where she put her keys or what she ate for breakfast, and over time she begins to repeat questions, struggles to remember names of close relatives, gets confused about the day or the place. Later on she finds it hard to manage daily tasks like cooking or paying bills, and she eventually needs help with dressing, eating, remembering family members. So Alzheimer's disease starts with mild memory problems and slowly worsens, affecting our thinking, our behavior, our daily life. So we want to know, like, you know, the genetic variants, like these SNPs, they contribute to Alzheimer's disease phenotypes, and what could be somebody's risk of having late onset Alzheimer's disease. So yeah, the polygenic score, the PGS, from the PGS catalog, is the single number that reflects someone's genetic likelihood of having a trait in a way. And the polygenic risk score is a single number that reflects someone's genetic risk for a disease. So they're just numbers that reflect somebody's genetic risk for a disease or a trait, and it's built by adding the effects of many SNPs, like tiny DNA changes, each with a small weight. So it's like a risk calculator. The more variance you carry, the higher your score. So also in the PGS catalog, you can see here that it might tell you the original genome build is this human reference genome, HG19, 21 variants here. And it was released in the PGS catalog in 2019. So yeah, as I show, like we click this, download score. Then we click this to download the text.gz file, and we click allow, and then it comes here as this zipped file, and then it gets extracted into this text file here. And then when we have the text file, it looks just like this here, for instance. So you could see that this is the genome build, GRCH37 again. These are the headings for the PGS catalog. This is information about the name, the trait reported late onset, trait mapped, all this information, the citation, the paper. And you can see again Alzheimer's disease. So the most famous SNP region for late onset Alzheimer's disease is the APO gene. APOE4 allele is the strongest genetic risk factor for late onset Alzheimer's disease. Having one copy increases the risk, having two increases the risk even more. But APO isn't the whole story. Hundreds of other SNPs across the genome contribute small effects. And I was just showing a very simple example here, just 21 SNPs. But yeah, these SNPs are discovered in genome-wide association studies, GWAS, where scientists compare DNA from Alzheimer's disease patients and healthy controls. So late onset Alzheimer's disease, it's polygenic. It's caused by the combined effect of many SNPs, each with a small influence. And a polygenic risk score for Alzheimer's disease like this, for the late onset, it adds up these small genetic risks to estimate the overall susceptibility, given these SNPs and effect allele. So at this chromosome, using this build, this HG19 build, so, based on the build, the positions, differ. It's kind of like translating different languages. So in a way, so we have like this position here, having an A kind of increases your risk of the late onset Alzheimer's disease. And this is the odds ratio. So whenever the effect allele is positive, the odds ratio is greater than 1. When the effect allele is negative, the odds ratio is less than 1. So as you see here, when it's negative, then the odds ratio is less than 1. And when it's positive, it's greater than 1. So these SNPs here that have a negative effect weight, they tend to be protective against late Alzheimer's disease, because their contribution, when we multiply them and we add them up to find the polygenic risk score, their contribution will be to lower the risk of late onset Alzheimer's disease. Essentially, this polygenic risk score is calculating the risk, somebody's genetic risk for late onset Alzheimer's disease. So if a SNP has a negative effect weight, it means that having that SNP will decrease your genetic risk for Alzheimer's disease. That's why it's protective. It's a good SNP to have. And if you have a positive effect weight, that means that having this SNP here would increase it for every copy you have from like one if it's from one parent, it increases it by 0.166. If it's from both parents, it increases it by 0.166 times 2. And if you don't have it, you don't have this being contributed to your score if you don't have this effect allele at this position in the chromosome. So that's sort of what it's trying to do. So people in, they calculate these polygenic risk scores, and they sum up these small genetic risks to estimate someone's overall susceptibility to late onset Alzheimer's disease. And people who have the highest polygenic risk scores may have several times higher risk compared to the lowest group. So that's what it's trying to do. And you can compare from the effect sizes from these genome-wide association analysis. So that study, those variants, they've come from here, from this paper in Neurology, as we see here, Tosto et al. So polygenic risk scores in familial Alzheimer's disease. And so this is the study that kind of released those 21 variants. So the objective was to investigate the association between a genetic risk score and familial late onset Alzheimer's disease and its predictive value in families affected by the disease. So they talk about how they did this here. And again, this is the score file, but I've just kind of organized it. And these RSIDs, this is the SNP ID, reference SNP ID, chromosome position, effect, allele, the one that if you have it at this given chromosome at this position, if you have this, then it increases your risk of Alzheimer's disease. It contributes this much to it for each copy that you have from a parent. So yeah, this is sort of like how we kind of build out these polygenic risk score trait prediction models, from 23andMe, Ancestry DNA, different types of analysis, for individuals, like which mutations do they have, and then we kind of build out this polygenic risk score trait prediction model, set of SNPs and effect weights, and then we can use that to kind of predict in a model like somebody's risk of having that given condition. If it's a polygenic risk score, it's a disease. If it's a polygenic score, it's a trait. And so PGS catalog is focused on a variety of traits, including disease. But since I'm focused on Alzheimer's disease, I'm going to use PRS and PGS interchangeably. So in this first step, we're going to create the sample folders in the DNAnexus platform. So I'm just going to be creating a few folders and just showing you how you can do that. It's a very simple initial bash script that we're going to be running. So this little guy here is a Liverpool guy. So well, anyways, I'm just in the spirit of UK and British soccer, or as they call it, football here, English Premier League. This is just an example of Arsenal football team, English Premier League, soccer or football as they call it here. So if you do make directory /p sports, football, league, EPL, Arsenal, what it will do is it's going to build out from your desktop, it's going to build out this tree where you have the sports folder. Then this within that folder you'll have a subfolder called football, then the subfolder called league, then a subfolder called EPL, English Premier League, and then the subfolder called Arsenal. So that's just how you can create a set of directories of folders and subdirectories all inside each other just using this command. And that's going to be helpful because we're going to run this using the DX toolkit now. We're going to be creating some folders and subfolders. And you can see here that this at the top means it'll be a bash script. And this is the file that we'll be running. And I'll be showing you a live demonstration next. But this will be the file. Here, step 1, simple starter.sh. So this means like we run the bash code. This is the name of the main folder here. This one will be over here, main folder that we're creating. And mkdir is like a Linux, it's like a command to make new directory. Mkdir means make directory. And usually on our computers, we can do mkdir if we're in the terminal to just create new directories. But since we're using the DX login, we will be also putting DX in front. So it tells us to do this in the UK Biobank DNAnexus platform to create that directory there. And we're going to create this main folder, Saniya tutorials. And then under Saniya tutorials, we're going to be adding this PGS SNPs one here will be the first folder. So that's what this will be here. This guy here. This one will be this over here. And the second one, which will be the PGS scores, this one will be created over here if it's not already there. And last but not least, we're going to have this one here, and we'll be also adding more folders along the way, but I just want to show you a few examples of how we will do that. You can see we have the folder, it's making the directory here, and then this is how we specified the subfolders. So that's how it does it. And if it's already there, it just doesn't worry about it. If it's not there, it'll make the directory. So I'll be showing you two runs, one when it's creating it for the first time in the DNAnexus portal platform, and the second one when it's already there. And you can also do this manually, and I'll show that too. You can do it right in the portal, old school. but this is just how we can create folders in the platform, and just a very simple way to see how we can work with the terminal and the DNAnexus platform, because things will get a little bit more intense after this. So yeah, I'm going to show you the live demo next, but to run this we will just do bash step 1 simple starter.sh, so that will run the shell script. And what we will see here is it's going to be creating the main directory Saniya tutorials here, which is what you see over here. Then it's going to create the subfolder PGS snips here. Under Saniya tutorials will be PGS snips. Then it's also going to be having PGS scores, which is the second one, PGS scores over here in the Saniya tutorials. And then Saniya tutorials PGSC calc output by PGS ID will be created over here. So you can see that this is the main Saniya tutorials folder over here. And it's going to have, and this main one will have PGS snips, PGS scores, and PGSC calc output by PGS ID. So these will be empty folders and we are creating them. And I'm going to show you the live demo right now for this. So now we're going to see that in the DNAnexus portal, we have a bunch of folders, and I'm going to show how we can create a folder for our results using the dx toolkit command here. So let me show you what that looks like. So we're going to go right here to our code, and now I'm going to show you here that we're going to open up the step 1, the simple starter.sh file. So we're going to be running these commands to create folders in DNAnexus. I'm going to have the Saniya tutorials folder followed by these 3 subfolders: PGS snips, PGS scores, and PGSC calc output by PGS ID. So polygenic score ID here. So the command is the mkdir make the directory. So let me show you how it looks when we run it. So right now, what I'm going to do is I'm going to go to, and you can see the time here and the date of when this was is going to be run. So I'm going to go and write, or I could have watched the job now, but it's over. So I'm going to just do the previous job. Sometimes you can watch the jobs. And also quickly while I'm at it, I'll just show you that, this job ID is the same as that this job ID, this J2FF77, this one here is the same as what I have here. They're both exactly the same, which is great. Ooh, this is the same thing as this, just to help you see how they kind of tie together a bit. Okay, so with that gone, now let me show you the code part now. And I'm going to do ls again just to say it, and I'm going to do bash step 1 simple starter.sh. So what you're going to see is that it's creating this main directory, Saniya tutorials, happy face here, that's run. Then it's going to say subfolder PGS snips is here, and that the subfolder PGS scores is here under the Saniya tutorials one. And then the subfolder PGSC calc output by PGS ID is here too. So it's going to create these subfolders. So please note that we ensure that all directories and folders are set up and ready. And so what if I just run it again? What if we run the command again, just back, then what we will say is it's just going to say that error while creating this cannot create folder, it already exists. So it's not going to override any folder that already exists because we just created it. So it's just not going to do anything. But it just tells you in the end that all directories and folders are set up and they are ready for our usage. So let me just show you what that looks like on the platform itself. So now we will see in the DNAnexus portal, we will have the Saniya tutorials folder that's right in the main area under the project folder, followed by these 3 subfolders that are empty for now. And this is what we'll be populating as we go through this tutorial video. So let me show you what it looks like. So right now, what you'll see is that we have this PGS snips that's empty. We just created. In the Saniya tutorials folder, we have the PGS scores that's also empty. And we have this PGSC calc output by PGS ID that's also empty. So all of the subfolders have been created. Yay. So that's how you create folders and subfolders in the command. Another just simple way we could have also done it is, you can always go here and you can always just create, you can just always create a very simple folder like this. You don't have to run it using dx command. You can just always create like new folder, dummy, create. You can also do that too. But I just wanted to kind of show you how we can do this, using the dx command. So I just also want to show you that if we go into our terminal and we just do dxls, it'll list a bunch of folders, including the bulk one that we were looking at and Saniya tutorials and a bunch of other things. So what we can do right now is we can just check from here that we can get into the dx Saniya tutorials one. So that's what I'll show you right now. 1 second. So you see here, I did dx cd Saniya tutorials, and then I can do dx ls. And it will show me these 4 folders that we have here. It will show me these 4 folders, including that dummy folder, but we don't really need the dummy folder. So let me just delete it, but I'll just still show you that like it has these 4 folders. And then I'm going to just click delete. And this action is irreversible. Okay, so now let's just see what it does now, like now that I do the listing. It has those 3, so it's kind of synced up now. It got rid of that dummy folder. So that's just also showing you how you can communicate back and forth with the DNAnexus platform using the terminal. So in steps 2 to 5, I'm going to show you how we can crush and subset these giant BGen files that have millions of variants just down to a small subset format of it's just a smaller BGen file size, and then how we can also convert that to a Plink1 format, BED, BIM, and FAM files. So how we can go from these giant BGen files, tens of gigabytes, even 180 gigabytes, for instance, just a lot of space that they take up, how we can go from those giant files, extract the variants that we need information from in a subset BGen file, and then reformat that BGen file into a Plink1 format friendly file, which is like a BED, BIM, and FAM file. So that's what we're going to do. We're just going to organize the input files so that it's something we can work with, because what is heavy for one can be light for somebody else, and what's light for somebody else could be heavy for somebody else. It's all relative. So now I'm going to show you the genotype imputation data that we're going to use, little guy. We're going to check it out and see what it looks like. And that's just for this example, this is the genotype data that we're going to use. And it's also because it's in HG19 or GRCH37. And I have other videos about the human reference genomes in case you're stuck or confused. But let me just show you the ones that we're using right now. And let me show that to you right now. So if we go to the DNAnexus portal, then what we will see is under the bulk, IBD projects bulk, and then I'm going all the way down to imputation. And I'm just going to use this one over here, and I'll have other videos on these different other types here, but I'll use this UKB imputation from genotype. This one, I believe, is in HG38 or GRCH38. So this is an HG19. So this one is HG19, HG19, GRCH37. And then this one over here is HG38, GRCH38, So we're going to use, in this case, we're going to use this one here. So when we click it, we'll see they have those 4 types. We'll see that we have around 96 items, and my dates may be different from yours. The number of samples I have may be different. It all really depends on your sample information. It's also changed across projects and permissions. So it really depends on your project, the time that you accessed it, you refreshed it. There's so many other things that it depends on. So please don't worry if it doesn't look exactly like my data, or if the number of samples that you have differs. This is just what I currently have at this moment. And again, my sample IDs and your sample IDs may correspond to really different people, because UK Biobank switches it up so that we can't really transfer easily across projects. And this is all to protect patient and the private data that we have entrusted the UK Biobank with, to protect patient information. So they kind of make sure we can't really map easily across projects. Though we can have a mapping file, and that's a whole other thing. But anyways, let me just show you what this imputation looks like over here, this imputation file. So I'll just show you, we have like dot samples for the chromosomes. We have this mfi.txt files. We have this. Sometimes it may not load properly with the BGI files. So those are some of the files that we have here. And we're going to be working with the sample files in particular. The MFI will tell us like the structure of the BGen files. These are the indexes for the BGen files, the BGI on top of it. And then these are the genotype files. So that's what we will be working with here. And again, this is because our scores are for HG19. So we want to use this format so that it's in the same format as our score file, the polygenic score file for our trait of interest, which in this case is late onset Alzheimer's disease. And also why this conversion from the BGen files also makes sense is because in PGS catalog calculator, as of now, what it mentions is that the supported file formats are VCF, variant calling format, the plink1 fileset .bed, .bim, and the plink2 fileset .pgen, .pvar, and .psam. And it says that compressed input is supported and automatically detected. For example, bgzip compression of VCF files or zstd compression of plink2 variant information files .pvar. So we're going to focus on in this video on the plink1 file set, the .bed, the .bim, and the .fam. And that's what I'm going to focus on, is how we go from the bgen files to subset them and then convert them into these files. And it's also because that's the requirement, one of the possible requirements for the PGS catalog calculator tool as of now. So yeah, little guy, these are just some metrics on the BGen format. So it's a compressed binary format for typed and imputed genotype data. So this is version 1.2 of the BGen format. So, you know, the UK Biobank interim release imputed genotype data was released in this BGen format that you see here. So most genetic association studies routinely employ data on tens to hundreds of thousands of individuals genotyped or imputed at tens of millions of markers genome-wide. So traditional data formats based on text representations of these data, such as the gen format output by impute or the variant call format VCF, are sometimes not well suited to these data quantities. Indeed, for simple programs, the time spent parsing these formats can dominate program execution time. So this format, this page, it describes the binary gen format, BGen, which aims to address these problems. It's a robust format that has been designed to have a specific blend of features that we believe make it useful for this type of study. It's targeted for use with large, potentially imputed genetic data sets. And the key features include the ability to store both directly typed and imputed data, store both on-phase genotypes and phase haplotype data, and small file sizes through the use of efficient variable precision packed bit representations. And then they have a per variant compression, makes the format simple to index and easy to catalog. So they have the index files as well that are available. So the current genotype format on UK Biobank that we're working with, that genotype format is this BGen format here. You can see under imputation, we've got the UKB imputation from Genotype, and we've got these BGen files that we'll be working with. So here we're using imputed data. So our genotype data will be the dosage. It'll be a value between 0 and 2. Otherwise, the genotype would typically be 0, 1, or 2, just those fixed numbers. 0 would mean that it's homozygous non-effect allele, so the individual has both copies of the non-effect allele. You know, whether it's like 2 A's, or it's 2 G's, or it's 2 T's, or 2 C's, you know, they just will have 0 if they have from each parent the same allele, and that's the non-effect allele. It'll be 1 if from one parent they have the effect allele, and then from the other parent they have the non-effect allele. So you can think of it like that. And then a 2 is the homozygous effect allele. So from both parents, instead they have the same allele, and that is the effect allele. So whichever allele is the effect one that we're considering, that's what it means. That's what the 2 is here in the genotype data. But right now, because we're imputing it, and some of these will be probabilities of genotypes, so the values will be between 0 and 2. And of course, like the closer they are to one over the other, then you can kind of gather that it might be more of a homozygous non-effect allele, or it's heterozygous, or it can be a homozygous effect allele. So this will be more of our case. It's a value between 0 and 2 because it's imputed. So yeah, what we're going to see is that we are using the UK Biobank imputation from genotype, genotype information, and the data field is this 22828, and in general we can look up any of these fields online for UK Biobank. And so this is what it is over here, and the reference genome that is used is HG19, also known as the really sucky G, or GRCH37. That's what it will have there, GRCH37. And you can see these are the participants, but of course our numbers will differ. But this is just how you can find out certain information on different data fields in the UK Biobank resource. And you'll notice that we will be calling this field UKB, this number, for the genotype information. So you'll notice that we also have this .sample file. So it's sample information, 1 row per individual / sample. So we have 487,409 in my data set. Again, guys, your number might differ based on your project and, when it was refreshed and so on. But, I'm just kind of zooming in on an example file. So for instance, we're looking at the chromosome 1, what its file is. So like we're zooming in on this file over here, for instance. And we can see that it's a text, kind of like a text file, but it's a .sample file. It accompanies the BGen files, and it contains sample information. So the first 2 rows are the header. So this first row is the header, and this is just the metadata here. And the subsequent rows, they contain the sample ID. So these 487,409 rows here, these remaining ones, will have the sample IDs. And in our case, these will be the same. It'll have, the gender information as well. So this is important because the BGen file itself, it does not store sample IDs. It just has the ordered genotypes. And tools like QCTool or Plink, they use the .sample to map the genotypes to the correct participant. So this is really important to have this sample file. And when we're subsetting the BGen files right now, we're not, in my case, I'm not going to be filtering out any samples at this moment. You can do all of that afterwards. Once you have the data and you've kind of computed things, you can always filter down the samples. My goal is just to filter down to the set of SNPs of interest, those set of variants, single nucleotide polymorphism. So I'm going to be holding on to all these samples right now for the analysis. But this is the sample files. And we also have these .mfi, .txt files. So this marker info file for the imputed data. So markers are like these genetic variants. And it's a tab delimited text file with per variant metadata. So what you can see here is that we have the variant ID information. We have the chromosome number, all sorts of information that we have. What we have here. So this is for chromosome 1. We have the chromosome 1. We have the position number. We have the variant, the reference alleles, the alternate alleles. This can be a variant ID signature, which is very common, but we'll be kind of focusing on the reference SNP ID because we have that in the score file as well. We have the position, the reference alleles, alternate alleles, minor allele frequency, the minor allele, and then the imputation information as well. It just gives imputation quality metrics like the info score, minor allele frequency, and so on. And the purpose is to provide a catalog of all the imputed variants in the corresponding BGen file. And it's often used to filter variants, like maybe you want to filter variants with info greater than 0.8 and minor allele frequency greater than 0.01. It can also be used to map SNP identifiers if my list is in chromosome position _ ref alt format. So yeah, this is the format that we're using here. And yeah, this is the MFI files. And we have one for each chromosome. So yeah, you'll also see that we have these BGen files here. So it's the main genotype imputation file that we are using here. It's in the BGen format. It's compressed, it's binary. And since we're using the imputed, it's under imputation, it will have the per variant imputed dosages. So it can be expected allele counts, like 0 to 2, but it can be fractional. It can be probabilities for each genotype. And each .bgen file typically corresponds to a chromosome here. It really depends on different cohorts and different data. Sometimes they can combine them all together for all chromosomes. But here in this UKB imputation from genotype in this field here, this UKB22828, as now it kind of splits up the BGen files for each chromosome. And these will be dosages here, which is just probabilities because this is imputed genotype data. So you can see here that we are visualizing the first few rows of the BGen files here. They're in HG19, GRCH37. So PGSC calculator, it'll also make sure that we match the format of the score file for the trait. And the format of the BGen file, it may differ based on the genotype information that we're using. So that's also very important. But you can see they have alternate IDs here. They have RS IDs. Chromosomes, position, number of alleles. So sometimes be careful because if it's less than, this is just a snapshot for like this, of course chromosomes 1 begins with 1, but this is just some example of, what the format looks like for them. First allele, alternate alleles. So here we can see the first few rows of the BGen file for chromosome 22, one of the smallest chromosomes, for the UKB imputation from genotype file. And you can see that it has alternate IDs, RSID reference SNP ID, chromosome position, number of alleles, first allele, alternate alleles. So we can typically filter, either using the alternate IDs, which is just the chromosome number, then the HG19, or GRCH37, it's the same thing, that position, the reference allele, and the alternate allele. We can use that approach, or we can use the RSIDs, and since our score file has the RSIDs, we can just filter using the RSIDs, reference SNP IDs. So we will be using the RSID column for filtering, since our score file has the RSIDs. But this is just a little snapshot of the data that the BGen file has, and it has this data, the genotype imputed dosage information across the samples for these variants. This is just the first 10 rows. And similarly, this is just the first 10 rows of, the chromosome 9 here, and we can see that it has a 0 in front. So any number that's less than 10, it'll, in this format, it has a 0 in front for the chromosome number. Just, a little heads up there. And so you'll notice that there are these bgen.bgi files. So basically, it's an index file for the .bgen file. So this is just an indexing file for the original bgen files. And what it does is it enables fast random access to variants, to these genetic variants in the bgen file without needing to scan the entire file, because those files are huge, like tens and tens and tens of gigabytes, right? So it helps with fast random access, and it's required for efficient extraction of specific SNPs or regions using the QC tool or BGenX or any other tools. So it's really helpful to have this. And, I'll show a command that we'll be running, using the BGenX command to index it, a BGen file, and then it is created really quickly. But it's a very important indexing file, and it should be found alongside the BGen files, which it is here. So what you can see is that for each of the chromosomes, we have these 4 files, right? So suppose you want to extract a subset of SNPs and also potentially samples, but in this case, in our example, in this video, we'll just be subsetting SNPs. But in general, why we will use this is the dot sample will tell the tool which individuals are in the file so that we can include and exclude them accordingly. But in our case, we won't focus on that. But just so you know that's what the dot sample is there for. The mfi.txt will help you identify the right variant IDs and positions to extract. The .bgen will store the actual genotype dosages, because here we're using the imputed data in this example, so there'll be like genotype dosages between 0 and 2, some values. And the bgen.bgi will make the extraction nice and quick by indexing the .bgen. So if you want to talk about chromosomes, this is the information that we have, these 4 files for each of the chromosomes there. So next we'll be organizing the score files for the given trait. We're going to be doing it so that we have the score file organized. We're going to upload it to the DNAnexus platform, the score file that we have for the late onset Alzheimer's disease. And then we're also going to extract the set of SNPs that we need for each respective chromosome. And we're going to have that in a bunch of text files, which I'll be showing you. But here is just organizing the score files, because that's what we're going to use as well to feed in for subsetting the BGen files in the step 3. So yeah, we want to now upload the score file from our local computer, where it looks just like this is on our little computer. This plain text format, less than 1 kilobyte or so. We want to upload it from our local computer. We'd find it in the Finder all the way to the cloud platform, to the DNAnexus portal. So we want to go from the local computer up to the cloud. So we're going to use the DX toolkit to do this, to go from our local machine to the cloud platform. So I'm going to show you how we do that. So now I'm going to show you how we can essentially start to upload our files from our local machine all the way up into the cloud, just like Mario jumping up into the cloud. Yay! So the first thing we're going to do is we're going to upload our score files that we have. We're going to upload those from our local computer all the way to the cloud. So what we will observe right now is that on DNAnexus, if you see here, what we see all the way here is that we have these empty folders. So we have these PGS scores, polygenic scores, and this folder is empty. And what I want to do is I want to upload this text file here that we have into that folder. So we're going to be using the DX toolkit, the terminal, to do that. So we are in the terminal right now, and we're going to see again that this file right here, that the info for this file, it is under users SK37 desktop demo, the score file here. So what I'm going to do is I'm going to write this command here in the terminal. And what it's doing is it's a DX upload users SK37 desktop Saniya demo score file. This score file right here is going to be going to the destinations IBD project Saniya tutorials PGS score. So we're moving this file to this destination here. That's what it's going to do. So before I hit enter, I'm going to show you that this currently is empty. Then I'm going to hit enter. And we see that this file has been uploaded here. It has this information. And we're just going to click refresh. And so what we see here is that we've got this file here that we wanted. It's uploaded. Exactly the file that we wanted. So it's already there. This local file has made it onto the DNAnexus platform. Hooray! So that's how we jump like Mario into the cloud with our data. So yeah, as I was showing you, want to make sure that you're logged in to the DX toolkit on your terminal of your machine. And then you want to run this command here that I was showing, and it'll really vary based on your file locations. But in general, it's DX upload, the file path to where you are. And this is where that score file is located on my computer. So you need to put the file paths there. This slash is just like a new enter. It's just like an enter a new line. Just so that we can put things on different lines, but it's just telling the editor, because I've put the the commands on different lines. It's just telling it like, hey, this is the continuation of the same command, because every time when you put it on the terminal, if you put a new line, it's just going to say, oh, it's a different command. So this just says that this all is a continuation of the same, like it's all one command, so that when we run this, it looks just like this, So I'm on my computer in the Saniya demo folder. So I'm just doing this here, and this just tells it like it's one command, and this is what it's saying, that it has uploaded the file, this score file, with this ID here in the DNAnexus platform. So I'm telling it to please upload this file here to this location. This is the destination. So that's what I'm saying here. So I'm uploading the score file, this score file here for this example. So that's how you upload a file. Or you could manually also upload it, but I think it can be sometimes a bit fussy, the portal. And so this is just a little easier to do it. So next I'm going to show you how we take from the score file, and I'm going to use Python to do this, but how we break apart the score file into separate files of SNPs for each chromosome. So we see here it has the RSID, so I want to extract the SNPs from the BGen file. And I'd seen that the BGen files for the UKB imputation from genotype, those have RSIDs. I saw that. So I want to go through, and for each chromosome, I want to create a score file. So you see here for chromosome 11, I want to create like the CHR11 here, where it has these 4 SNPs that are contributing to chromosome 11 here. That's what I want to do there. And you can see that these are all the different chromosomes we have. That these are the 4 for chromosome 11. And they're in one file. And then these are the ones for chromosome 14. These are the 2, and they're saved in this text file. So basically just going through and saving the SNPs for a given chromosome as a separate text file, because then I'll be telling the BGen files, I'll be telling them to just please subset themselves for just those SNPs. So like for the chromosome 1, it's just going to be subset to this one tiny little SNP, and so on. And for chromosome 20, just this one SNP, and for chromosome 14, these 2 SNPs, and so on. And I know this is like a toy example, but it's a real world example, but it's just 21 variants. But it just kind of shows you like how it's done. So now we're going to see how we can take this score file and make it into tiny files of just SNP IDs for each of the chromosomes. And you may just think, okay, they're just 21 SNPs, right? Like it's, 21 variants in this tiny example. But again, remember, it's just a toy example. In reality, you could have fewer SNPs or more SNPs or that are important for a given trait or disease. So you can adapt this kind of idea. I use Python. You could do filtering using Excel or R or other programming languages, manually. It's up to you. But I just want to show you, and I've provided the code example for how I will do it in my situation. So let me just show you how I will break apart that score file that we have for the late onset Alzheimer's disease into individual files of the SNPs that we want to extract for each of the chromosomes. And that is because I'm going to want to subset the BGen files for the UKB imputation from genotype, that BGen file. I'm going to want to subset it for just the relevant SNPs, because the original files are extremely, extremely huge. What I was showing you before is that, for these like BGen files, if you just even look over here, they are enormous in size. You can see that some are like 80 gigabytes and 160.29 gigabytes, and you'll be charged for the storage and it if you create your own additional files as well, and it is not efficient in my opinion. And again, these are just the things that I did, and you can please feel free to adapt it according to your needs. So, but you can see that again, these are really huge files, and it probably would be better to subset these BGen files into something smaller, like 143 gigabytes, 156 gigabytes, and they have millions of SNPs, right? So to that end, what I show here in Visual Studio Code is example of how we will extract SNPs and variants from PGS catalog score files. So I import these libraries here, operating system, this is just like a progress bar, pandas, numpy, and so on. And so my input file is over here because what you'll see is that you'll see is in the score file, the get info, we are right here in the users, Saniya color, 37 desktop, Saniya demo score file. Like that's where we are right now. So users, this desktop, Saniya demo score file, this score file over here. So I'm just actually going to clear all outputs here. I'm just going to run this here. And I'm going to run this guy over here. And what you will also observe is that currently in the score file, there's nothing else beyond this text file here. So we're going to be parsing through and trying to extract and separate by SNPs. So it's going to be reading each line of the score data and see that if we find the RSID, like what that is, that's the line to start from. So it's basically kind of looping through the score file and waiting until we come upon the RSID in the title here. And then that's going to tell us that below it is the data that we want to use. So it's kind of like looping through this line, line by line, by line, by line, by line, by line, until we get to this RSID. Then it tells it that, hey, this below are the information on the variants that we want, for the scores. So it's going to be saving the SNP table to a tab separated format. So you'll see here that already now we have this TSV file. And this TSV file, and I open it up here, it will just have the RSID, the chromosome number, position, and alleles here. And you can see that these are 21. See, this is row 22, this is row 1. So this is 21 variants and the number, position, effect, allele. So we're basically just taken that text file and then just gotten the information, gotten rid of the header, and then just put the rest here. And this is just for our reference, to make it cleaner. And then what I'm doing over here is I'm essentially going through that data and I'm trying to extract the information for each chromosome. I have two things that I do. One is that I'm taking chromosome name, position, effect, allele information, or chromosome name and position information, and I'm like saving that as a range. And the second one is I'm also just taking the SNP information. So I'll just show you what the two things do for the code. You can run these and try these out as well, but it's kind of just going through the files, and it's also just saving the chromosome number, position. And then another one that it's saving is just the RSID. So it's just looping through each of those files for a given chromosome that is found. And essentially what it's doing is it's writing these out here. So we have the per chromosome SNP files. We have a set of the ranges that you can use if you're interested, where it will give a 0, 1, because what the bgen file has is that if it's less than 10 for the chromosome, it'll give it a 0 in front, and then the number, and then the number. And this is just if you're using a score file which doesn't really have properly formatted RSIDs, or for some reason that information is not ideal. But it should be okay for the most part. But just in case, I create this file, but it's not super important. Like, for chromosome 18, it looks like this. Let me just see, like chromosome 14, it has these two SNPs here, this, and this. And you can see that they come originally from chromosome 14, these two ones here. But what we're going to be using instead are the RSIDs. And these are the RSIDs that we get here. For chromosome 1, this one, like this SNP here. That's for chromosome 1. For chromosome 2, we get those 2. Chromosome 5, we get this 1 SNP. Chromosome 6, we get these 2 SNPs here. You can see that we have these 2 same SNPs here, which we've identified there. Chromosome 7, the 3 SNPs. Chromosome 8. Chromosome 11, 14, 18, 19, and 20. So we have all these like different SNPs here that we're going to use. And so we have this saved as a file of basically for each chromosome that's present there, what are the SNPs that we are going to use. And you can see that we just have these 10 chromosomes, essentially, these 10. And so not all chromosomes have a variant, and I'll show you in the code how we account for that. But this is just kind of showing how we take that score file and then how we are able to essentially break it apart into small files of SNPs to extract for each chromosome, which is important because then we're going to use that list to subset our original BGen files just for those SNPs of interest so that it makes the computation more efficient, the files a lot smaller, and we can also kind of see what SNPs we have found, if at all. It just makes the process a little bit easier than working with those giant files of millions and millions and millions of SNPs. If we're not even going to use them, we might as well just subset them, right? So little guy, I'm next going to show you how we can upload these SNP files. Similarly, these text files for each chromosome, what are the SNPs we want to extract that correspond to the score file. So each of these text files that we've created, how we can upload them to the DNAnexus platform. So how we do that? Using DX Toolkit again. So it's the same kind of idea as before. Going from a local computer to the cloud, DNAnexus on the cloud. And again, making sure you're logged into DX Toolkit on the terminal of your machine, your local computer or your server. And then this is the command that I did here. And again, this is just the same command, like DX upload. Going to the folder where it's located, and then the star.txt, that means that to upload all of the .txt. So instead of just manually listing out each text file to upload, the star means just upload all of them that match. It's like a wildcard thing, anything that matches txt format. And then this is the destination. I'm moving it to polygenic score SNPs folder, so PGS_SNPs. So that's the command that I do. And it will really depend on the way you've saved your files and what they are called in your file path location. But this is just how I did it, and you could adapt it, hopefully. So now, little guy, we're going to see how we can upload all those tiny little score files that we had, the SNP information for each of the chromosomes. To be subsetting the BGen file, we're going to see how we can upload those score files from our local computer, our server, machine, whatever it is, how we can upload that into the UK Biobank research analysis platform, the DNAnexus platform here, how we can upload that for many files. So if you recall right now, we just talked about how we essentially created these files here for each of the chromosomes, what SNPs that they have that map to them. And we have these per chromosome SNP files here. And it's really cool because we can get info here. We can see that this is located in Saniya demo, score file, per chrome SNP files, this one RSIDs. And it will tell me that this has this one SNP. It'll tell me that this has this previous kind of cool. It'll tell me that, this has this 4 SNP, so we don't even have to click that text thing open again. So I want to basically upload all 10 of these text files to this folder here, to the PGS SNPs, which is empty. But previously I showed how to do this for the PGS scores. So how can we do this for the PGS SNPs? So for those multiple score files, how can we push these multiple score files into the DNAnexus platform. So to do that, what I'm going to show you is the command that we will run. And so that command that we run is the following. And like similar to the last time, I'm just going to have this right here and then pull this little guy up. So we're going to be uploading from this folder, score file per Chrome snip files. This is the same folder that we were using. This folder, per Chrome snip files. This is the snip, the RSIDs folder, subfolder. We're going to be basically pushing all these text files. That's what this is saying. Then the RSID subfolder here. Star means take all of that. Take all those files. That's what it's saying. Instead of us specifying this file and that file and that file, like one by one by one by one, we can just do the star to push it all. And the destination will be the IBD project Saniya. And again, this will differ based on your project. Saniya tutorials, PGS SNPs. So that's what I'll be running. And again, right now it's empty. So when I run this, it'll look like each of the 10 file, 1, 2, 3, 4. You see them populating in the back, right? You know, so on. All these 10 files will be coming and getting uploaded. So it finished uploading all these 10 files here. Just have to click the refresh button here. We have the files for all 10 chromosomes. You know, like this is for chromosome 8, for instance. So that's how we push those per chromosomes, like SNPs to extract for the score files. That's how we push it from our local machine to the cloud using the DX upload command. Yay. So as you see here, for this score file in this example, we have only 11 of these different chromosomes. Human chromosomes have the polygenic risk score SNPs, single nucleotide polymorphisms present for the late onset Alzheimer's disease based on this study. You know, chromosomes 1, 2, 5, 6, 7, 8, 11, 14, 18, 19, and 20. So based on this. So in step 3, we're going to subset these BGen files. We're going to take them from these giant files and we're going to just subset them down to the variants that are important for our example, for our score file. And of course, for different score files, we could have different subsets. And this is just my example, my way of going about doing it. You may have your own approach. I know there are experts who've been doing this for years. So please, I would really love to hear any and all feedback that you have or suggestions for my own work and for my videos to help all of you. But in this next step, step 3, we're going to be subsetting these BGen files for just the variants of interest so that we can have condensed genotype files going forward. And you may wonder, well, why do we need to do that? Well, some of the files can be quite large over here. You can see that, like for this example here, for chromosome 1, it's like 181 gigabytes, which I know when people talk about terabytes, I mean, this may seem like it's not much, but it is quite a lot. And especially because we want to convert it into like different formats, like a Plink1 format for the PGSC catalog. Calculator, we're going to want to have like a more condensed file and it'll just be a lot faster for us. So I want to kind of shrink down these files to something more manageable. So that's why I'm going to subset it, because the original files, as you can see, are quite memory intensive. And that's great because that's all data. That's all like really helpful data. But for our score, for this example, we don't care about the other variants. We really just care about our variants, those 21 variants that were determined in that research study on late Alzheimer's disease. So ultimately, our goal is to make those giant files, BGen files, down into these subset files that will be easier to convert. These BGen files will be much easier. Like you see here for chromosome 1 now, it's just 15 megabytes. It's going to be so much easier to work with instead of 182 gigabytes. It's so much smaller. It'll be much easier to convert these into the bed files and then those Plink formats that we can use for the PGS calculator catalog calculator. So that's what we're trying to do. We're trying to shrink down the sizes to make them easier to work with. So yeah, the idea is that, we have for inputs, we have the set of the SNPs for the given chromosome that we want to extract. We have it like as a given text file, like for chromosome 14, we have these 2 SNPs that we want to extract. And then we have this UKB imputation from genotype, the original BGen files here. This is the original BGen file. As you see here, it is like 77 gigabytes. So it's pretty huge, pretty sizable. We have that file here, the BGen file, and then we have the index file to help us go through this file a lot quicker for chromosome 14 here. So we have the original BGen file, the indexing file for kind of going through it quickly, and then we have the set of SNPs that we want to extract. So these are like the inputs basically, and then the outputs will be these 3 formats. So the first will be the text file of extracted SNPs here, and it's optional because they're just 2. I wanted to show you that if you want to have a text file, you could have them to see which SNPs you have extracted, and it will tell us that from the BGen file, this is how it looked, and the BGen file had these RSIDs, which is what we were indexing. So it did recover these 2 RSIDs here, and this position and all that. So it's saying a BGenX success, total 2 variants, the 2 of 2 were captured, which is great. So that's the text file, the extracted SNPs. And yeah, we subset the BGen files using BGenX. So if we go to the BGen documentation here, what you can see here is that it says that BGenX is a tool to create an index of variants in a BGen file and to use that index for efficient retrieval of data for specific variants or regions. So the command line, or what option and what is here, and then what it does. So we're going to filter our BGen files based on the reference SNP IDs, these RSIDs. So this INCL RSIDs will say only output variants that have one of the given RSIDs. So we're going to be using this flag over here. That's what we will be doing. And you can see that they have other commands, like if we just want to include range, INCL range is only output variants in one of the given ranges, then we use this command. It says that, oh, I only want variants from this BGen file. So we use BGenX/G. Then, that's what this is saying, BGenX/G, my file.bgen. That's what you need to start off with, this part here. So as you see here, that we do need like this beginning part here, and then that's what we use here, and then we do kind of tag on to it like the other commands that we need. We will tag on to it like these other things here. This list the variants in the index, that's what list is doing. So the additional options here, so this one will say, oh, from this BGen file only include subset it to only those that are on chromosome 11, between this position and this position, for instance. That's it. But we are going to focus on subsetting it based on those RSIDs, those SNP IDs that we wanted based on our score file. So we're going to adapt this for RSIDs instead. So we have BGenX/G file.bgen include RSIDs, this command here. And then we're going to have a SNP file name.txt, then we're going to have it going to the output here. Because for instance, if we scroll down on this page, it says that that they have these types of commands here. So, the options above, like INCL RSIDs, so it says that this is what the inclusion criterion on a variant will be output if it satisfies at least one of the inclusion options and does not satisfy any of the exclusion passed in the command if you do the XCL RSID. So we're doing this one here. So it takes either value, so you can say only I want to include this RSID so and so, or you can give it a file name. So we, instead of doing the INCL RSIDs and giving a specific RSIDs, we're reading it in from a file name.txt. That's what it's saying, and that's what we're doing. So we're going to be subsetting it, a given BGen file, given the list of SNPs only in this file name.txt. That's what we're doing for a given BGen file. So then what you'll see here in the code is that when we run this bgenx/g, this file name incl rsids, this command over here, it's creating the subset bgen file. So for chromosome 1, it's creating this PGS. This is the score PGS ID here. This is the genome build that I mentioned. This is what it's trying to create here. The chromosome 1.bgen. This is the file it's creating. So this command over here is the one that is creating the subset bgen file for only the SNPs in the input file. And the command is bgenx/g. This command here, bgen file name, include RSIDs, SNP file name, and it's going to be writing it out to the output bgen. So for chromosome 1, when CHR equals 1, so I'm showing you what this looks like. Then this bgen file name is given as this. This is what I've specified here as my bgen file name. It's this here. That's what this is. So bgen file name based on chromosome 1, this is C1. It is the UKB22828. This is the field name, C1B0V3.bgen. Then include RSIDs part here. Then for the SNP file name, it comes from this PGS ID. Our PGS ID is this ID here that I've given. And again, you can adjust it based on other things. Like I don't need to have the SNP file name be a certain way, but this is how I named it. So I'm copying my naming conventions, and I'm hard coding, and I'm being dynamic, so I'm not hard coding it, so that you can adjust it if you'd like for your needs. But the SNP file name PGS ID is this in front, and that's what this bracket is doing in Bash. This bracket means that it's a variable name. So you'll see that whenever we have for SNP file name, we'll have this $, then the brackets, the $, then the bracket here. That just tells it that it's a variable to be filled in. So PGS ID, then chromosome 1, then this genome build. So this will be read as PGS, this ID here is PGS ID, then chromosome 1, then the genome build, which I've specified here, is GRCH37. So that will be over here, then rsids.txt. So that's what my SNP file name will be. And for each chromosome, what differs is what this is. This is chromosome 1, so that's why this is 1 here. And then the output bgen is what I specified over here. So the output bgen is this part here, where the PGS ID again is this. It's filled in for what this is here. Then the genome build. Then the UKB 228 subset part is all here. Then the chromosome 1 there, and then underscore rsids.bgen. So it's basically telling it that from this bgen file, include the RSIDs that are found in the SNP file name. In this SNP file, include only those. Subset it to only the SNPs found in this text file, and then save the bgen file as this file name. And this output bgen, where will it be? It's actually going to be stored in the directory that is mentioned here in the destination part. That's the output folder. When we do like the DX upload parts, we give the destination. That's like telling it where those files will be uploaded to. So here the destination, it's saying that whenever we have writing outputs, please put the outputs, whatever those are, in this location, this output folder. And the output folder here will be Saniya tutorials extracted genotypes bgen. So it's basically saying that, for this part here, take this, include the RSIDs that are found in this text file, and then store them and output them in this bgen file. And this bgen file, this command is saying, will be located in this output folder here. But this is the command for subsetting the bgen file. So this second line of this command, this bgenx/g output bgen/index, what it's doing is it's creating the corresponding subset index file, the bgen.bgi file. And then again in the bgen documentation, it says that to use BGenX with your BGen file, so if you have a myfile.bgen, you use the following process. So bgenx/index/g myfile.bgen creates an index file. The index file will be named myfile.bgen.bgi. So it's the same idea. So we're using bgenx /g, and again, these are flags, so we can shuffle them around as long as we specify them in the commands. So bgenx /g /output bgen. This is the myfile.bgen. So it was the one that was output here. We want to index this output bgen file here. We want to index it. So we're going to take it. This is what our myfile bgen is it's this bgen file here, and we're going to do -index. So for chromosome 1 CHR1, for instance, because it's looping through the for loop. So when CHR is 1, 2, 3, 4, 5, 6, all the way down to 22. So, when CHR is 1, so for chromosome 1, for instance, the output bgen is what we were just talking about. It's based on this PGS ID, genome build. This is the name for the output bgen over here. This is the name here. And so in this command here, it's bgenx/g, this bgen file that we've created. We're going to be doing the index. So you'll see that this is for chromosome 1, what it's going to be output as. So it'll be the bgen.bgi. It's going to be an index file that will be output for chromosome 1. And again, it's going to be stored in the output folder here. So it automatically is creating that index file, that bgen.bgi file. The BGI, the indexing for the bgen file. That's what it's doing for the file that we've just created. And the semicolon means like there's a set of sequence of commands to run. And the command line, this would be one line, it would execute, then this would execute next, and then this one. So the last line is optional, but and again, it's just because we have a small set of SNPs, so you can do this. You don't need to do this. This is just for like defensive programming and just to see like what SNPs were found. You don't need to use this. It's optional, but it'll give you this text file here. And all I'm doing is bgenx/g output bgen/list, the output found SNPs. So again, the output bgen this is the output bgen was specified in this way here. And then the output found SNPs is given this here, where again the dollar and the brackets, this means that the $ and brackets will be removed, whatever this text, this $ and the bracket sign will be removed and substituted with the name. So this part here will become PGS 00053, then this over here is GRCH37, and so on. This is the bgen name, then the list. And the output found SNPs, again, this PGS ID is this PGS ID here, genome build, then the rest of it here. And then because CHR is 1, it'll be chr1.txt. So just create a text file of the SNPs that were found in the bgen file. So just going through the bgen file that we've created in this set here, it's going through that bgen file, and it's just listing the variants to a text file. And then it's outputting that for our reference, and it's going to be saved again in this extracted genotypes bgen folder. And so you can do this if the number of SNPs to be extracted is small. It's optional. It's just for you to make sure that it's done. So now we're going to see how we can subset those giant bgen files using those SNPs that we've specified, and this will be the step 3. So we have those set of bgen files, those giant files for the UKB imputation from genotype, and again, this can differ based on your needs, but in this example, that's what we're using. So we have the UKB imputation from genotype information, those bgen files, bgen.bgi index files, and the .sample files. We have the information, and we have a list of SNPs that we want to extract from that we've created in those like small little text files for those 10 chromosomes. So we're just going to basically run the code to go through and see if the chromosome is present, like if it's one of those 10 chromosomes, just please select those set of SNPs from that bgen file and create a mini bgen file So what we will be doing is we're going to essentially go from the UKB imputation from genotype, this raw data we have like the index information, which will help. We have the BGen files, the original BGen files. You can see they're quite large, as I was mentioning earlier. We're going to be subsetting these BGen files using the reference SNP ID information that we have here, like for each of these 10 chromosomes. We're just going to be creating mini files for these tiny variants. And you may just be wondering, well, these are just 21. Is it really worth the hassle? But this is just a toy example. I mean, it's a realistic example for late onset Alzheimer's disease. But in your case, you may have lots more variants or fewer. But this is just an example to show you how you can do that. And you can adjust your pipeline as needed. I'm just showing you what I've done and what works for me right now, knock on wood. But we're going to be essentially like going through chromosome 8 and saying, okay, I want to just select both these SNPs here. And then we will be going through the chromosome 8 BGen file, which is 122 gigabytes, and we're going to be just extracting those two SNPs. So to do that, what I'm going to do is I'm going to be running that and monitoring this job. I have the code, as I was mentioning, step 3 subset bgen files.sh, and I'll be running this, which will be going through the UKB imputation from genotype folder that I just showed you. And it's going to be going through the Saniya tutorials PGS SNPs. That's what this folder here is, Saniya tutorial PGS SNPs, and each of the chromosomes here. So it's going to be going through that as well. And the output folder, which does not yet exist, the output folder would be created, and it's going to be called extracted genotypes bgen. That's what it's going to be called, extracted genotypes bgen. And that's what we're going to use, essentially. Extracted genotypes bgen folder is where those extracted files will be output as a new folder. And I'm showing you another way that when we're running this, again, just like when we were running the step 1, how we did the make directory, here we can also make this directory here for this output folder that we've created here. So this is just an additional folder that we needed, and we're going through all the 22 chromosomes. And if that chromosome score file, that SNP file for that chromosome, if it does not exist, we're just going to skip it. But if it does exist, we're going to be subsetting its original BGen file in this folder. We're going to be subsetting it for those SNPs of interest. That's what we will be doing in that code over here. So we're going to just include those SNPs that are necessary. And if it's not found, we're going to skip it. So how do we run this? So what I'm going to do is I'm just going to, we've uploaded those files. I'm going to do ls, again, just to show you. I'm going to do bash, step 3, subset bgen underscore files.sh. So what you'll see here is it's saying submitting job for chromosome 1, for chromosome 2. See, please note this file is not found for chromosomes 3 or 4. See, those files were missing, right? Those chromosomes were not found in our score file. You know, chromosome 5 is there, chromosome 6, 7, 8, 9, and 10 are missing, so it's skipping those. See, it's skipping those because they don't have any like SNPs in the score file that we're using. 12 and 13 are not found, 15, 16, 17 are not found, and 21 and 22 are not found. So it skipped those. So in the monitor, what we will see here is we just have these jobs here. And these are the chromosomes here. 1, 2, 5, 6, 7, 8, 14, 19, 20. Sorry, I mean for these 11 chromosomes. there are 11 chromosomes, so half of them are found here. For these 11 chromosomes, we're running the file for each of the chromosomes. So yeah, for these 11 chromosomes, 1, 2, 5, 6, 7, 8, 11, 14, 18, 19, and 20, it's still running. And you can see that this is what the command looks like here. It's processing chromosome 1. It's using this Biobank command here. It's including the RSIDs. Then it's extracting them into this bgen. This is the first command, which is just extracting those SNPs that were mentioned. It's in that SNP file. It's extracting them into this tiny bgen file. And then the next one is it's creating an index file, a bgen.bgi file. And then this one is kind of just listing, it's an optional one, but it's just listing what we found in the bgen files out to this text file. And I'm doing this because we only have 21 variants, so it's just a quick way to check. But when you have a lot of variants, I don't know if I would recommend listing them all out. Like if you've got like millions of variants, it might be a lot, but it's up to you. But you can see that it's running the command here, and it'll take some time to run. That we can monitor them using this command here. So as you see here, what this is showing you is that we're running the job for chromosome 1, and the monitor 11 means that there are 11 jobs that are running, one for each of the 11 chromosomes that we found, because the score file has 11 chromosomes. That's waiting. And you can see that these are the input files that we're given. We're given it the SNPs that we want to extract based on that score file. We've given it the bgen file that we want to extract the genotypes from, the original, that UKB imputation from genotype, and we've given it the indexing file for the bgen. So these are 3 inputs because we've specified that, we've mounted that, we've specified the location of those file paths. So this is what the command is essentially doing. So for chromosome 1, it's running this command line, the CMD. The bgenx/g, this command, this first one is including the, as I was mentioning, just subsetting this bgen file to only the SNPs found in this the SNP related score file, and then it's saving it as this bgen. And then this next one is indexing that bgen file that we had. It's indexing it. And the last command, which is optional, is just trying to list the variants found from the bgen file into this text file for our reference, and just telling us what was found. The bgen file will have the genotype data, the dosages of the individuals, but the text file is just going to tell you like which variants do we have that data for. That's what it's telling you. So yeah, little guy, now we find that there are these 3 outputs. And as I was saying here, that, this output here that is optional, this extracted SNPs, is just going to tell you what were the variants that were found in the bgen file. It's the BGen file has the genotype information. So for the samples, like what are their genotypes? Like, do they have the effect allele and the reference and alternate allele? Like, what is the information? That's what the BGen files is going to tell you. And the indexing file is going to index this BGen file. But this text file, which is optional, is just going to tell you in the BGen file, the genotype data, just what are the variants that we have that information on. That's what this is telling you. What are those variants? The BGen file will be giving you the genotype, that information for a set of variants, but it's not very easily accessible. So I just want to make sure that we have it for the variant of interest. So we do see that from this BGen file, it has been subset down to this one variant that we wanted for chromosome 1, because that score file just has that one variant. I know this is very tiny, but yeah, this is just showing you for one variant we have this tiny BGen file. And in the code, what you'll see over here is this command, and what this is doing is it's seeing that we have the SNP file DX here, and we need to sort of put this here by alerting the cloud platform, the UK Biobank DNAnexus, to check fully if this file exists. So if we have a score file for that given chromosome, so it's going through chromosomes 1 to 22, and essentially it's looking for the SNP file. So in Saniya tutorials, under the PGS SNPs folder, it's looking for the SNP file name, and the SNP file name is given with the PGS ID and the chromosome number and the genome build. So you'll see here it's like PGS ID, CHR8, the genome build GRCH37, RSIDs.txt. I know I'm a bit involved with the names, but it's just for keeping track of what's going on. So it's searching for that SNP file name here in this location. And again, they all have the same pattern of the ID, PGS ID, CHR number, GRCH37, RSIDs.txt. So it's just searching to see if that SNP file exists. So it's listing in this and seeing if that file is not found. Then it says, please note that SNP file for chromosome, whatever number that is not found, skipping for RSIDs. And then it's continuing. So in the loop, it's continuing, and it's not going to submit this job because it says that, oh, there's no SNP file, so I don't need to subset the BGen file. That chromosome did not have any SNPs for that score file, so we don't need to use that chromosome. So there are 11 chromosomes that, as we see here, that had any variants in that score file. So for instance, we will not see it for chromosome like 21 or 22, and it'll skip over those. That's what this is doing. It's defensive programming. So if the score file for the chromosome does not exist in the folder on DNAnexus, we skip this. And again, just to explain that when we're running the CMD, the command, this is using the original file names in the command part. It's using the original file names, BGen file name, output BGen. You see that they don't have any like slashes or anything that's telling it which folder it belongs to. When it's reading in BGen file name, it's this UKBCCHR, like B0v3.bgen. But you may wonder, okay, well, how does it know, right? But the way it knows is when we do the dx run command, we are specifying the location. So this will upload all of the files using the full file path so that we can reference them. So when we call this BGen file name, we are telling it here that I want to input the BGen file name based on this. So I want to go to the input folder, which is bulk imputation, UKB imputation from Genotype. And that's where the BGen file name is. And that's the file I want to upload into the workspace. So that's what it's telling you. So when you will see in the command that it has a certain like inputs that it has, this is where it comes from, mentioning it here. So that's how we can access the BGen file in this command, because it will be uploaded based on this part here. We've mentioned the file name, but we've also said where that file name is coming from. And again, the $and the brackets, they encapsulate the variable name. So that's what we're doing here. And then this destination output folder, which is over here, is just going to be where all like the outputs, this output and the bgen.bgi file output here is, and then this arrow is like output it, like where do you want to put the output? What is the file name? It's saying that I want the output file to be saved in this file name and the output to be saved in this file name. That's what that arrow is saying. Like, push it there. And the destination is telling you that whatever you're saving and writing out, like where it will be going. And you need to give like the full file paths here when you're using the dx run part, where these files are located and where. But in this area, you cannot put the full file paths. You have to just, for the command, you have to just give just the regular names. So you cannot put input folder /bgen file name in this part here. You cannot do that. You can just put bgen file name. It'll get confused otherwise. And but why we do this over here, we're not running any command. We're just sort of seeing why we give the full file paths. We're not running any of the like Swiss Army knife command parts here. We're just seeing if that file exists, which is why we need to have the full file path. So now we can see the progress of these runs for subsetting the bgen files. So it's taken some time, and time is money on the research analysis platform. So please be mindful. I think when you start off, you have a little gratuity of complimentary small amount that you can use, and that goes really quickly. So what we see here is, and this tells you like the cost and all that. So I've finished these processes for all of these 11 chromosomes. So for instance, this is the job that's finished. And the outputs would be this bgen file, this bgen.bgi file, and this txt file here. Oh. And we can see that this is just one of the SNPs that's been extracted, which is the one that we wanted. And it's extracted it from this, using bgenex. Similarly, we can also see here when we're running through it, just to see what's happening here. It's evaluated, it's processed one variant, and it was successful, and it was uploading these files. So this also shows you that what you have provided when we're uploading the files from the code here, when we've uploaded, using this input folder, the bgen file name, it uploads, for the Griffin chromosome 1, it uploads it as this bgen file and then this bgi file. And then it also has uploaded this SNP file here. We have the index file name, which is also corresponding to, bgen is the bgen file here. That's what this is here. The index file name is the bgi file. And, for chromosome 1, for instance, and this is the input folder, which is the bulk imputation, UKB imputation from Genotype. So we specified this, and this will correspond to this bgen over here. The second one here will correspond to this bgen BGI. And the third one, which is the third input here, you see these three inputs, these three here, this corresponds to these three respectively here. And so the third one, which is the SNP file DX, this is Saniya Tutorials PGS SNPs, and the SNP file name. And the SNP file name is PGS ID, which is the PGS 00053, then the chromosome, which is chromosome 1, the build, which is the GRCH37, and then RSID. So I added the name to be a little bit longer just to remind us like what the build is, just for reference, just for consistency. So it's uploaded this file as well here. So it's uploaded these three files here into the workspace. And then when it's running these commands locally, so these will have to be absolute paths where it needs to know where exactly in the project area are these files, where exactly in this IBD project Saniya are these files located that you want. Then it's uploading them. And then we're just using the local references. We're using bgen file name, not with the input folder. We're just using bgen file name by itself here in the command. We're just using the index file name. No, we don't use it at all here, but it's necessary for the command. And then what we're using is the snip file name, and the snip file name was what was part of this snip file dx here. It was part of that. But here we're just using the local snip file name. We're just using this over here. We're putting this in the command. So when you see, like, what it's doing is it's essentially just looking at the local command. It's just taking this UK, it's taking this here. This one right over here, which corresponds to this file here, it's just taking the local name of it. It's taking the local name of this file here, which corresponds to this once it's been uploaded. And that's because, like, in this over here, we're just using, once we've uploaded the files, we're just using the local name, snip file name. We're not using the absolute names. So this is what the command was for chromosome 1. And again, it skips over the chromosomes that were not found. It skips over those. That's what this part here was doing. It's skipping over those. So what we see is this is one example. We can also do this for this other example here, like for chromosome 20, for instance, we can see that this here corresponds to this one being uploaded. This is all for chromosome 20 specific files. This is what the command is run. And these are chromosome 20 specific output files that are given and returned. So similarly, that's what we also will observe for, like, this chromosome here, chromosome 14. We can also see what extracted SNPs those were here. You know, we got these 2 SNPs extracted from the BGen file that we needed. And that's what we were wanting for chromosome 14. We had those 2 SNPs that we needed. We had those 2 SNPs. You see for chromosome 14, we had these 2 SNPs, and those were what was extracted. And this was the command here again, and it's all using local. But to upload it, we needed to give those absolute paths in here so that it knows where those files are located. And then going forward, it's just referencing these local file names. That's what this is showing, local file names for this. And this is what is being output. And I'll show you a couple more examples. This is chromosome 7 specific information, C7, 7, 7, 7, 7. 5. And now what we're going to observe is the following. I mean, if we go here, we now see that there's a new folder. So we've created the new folder, extracted genotypes.bgen. So this new folder is here. And now what we will see in this folder is we will have these, chromosome files, the bgen, bgi files, and the txt. So for each of these, we will have for the required, the appropriate chromosomes that were found in the score file. So 3 files for each of the chromosomes. 3 times 11 chromosomes, 33, as expected. So then what we're going to do next is we're going to try to squish and squash and merge these files together into, well, we're going to do that eventually, but what we're going to do next is we're going to convert each of these into the Plink1 binary formats, the BED, BIM, and FAM files, and then we'll squish those into one file across all the chromosomes. So that's what's coming next. So over here, what we will see is that this is the inputs and outputs example for chromosome 1 when we're running it. This is how the command looks. We have these input files again, score files, the bgen file, bgen index file, and then these are the output files. This is the bgen, the output subsetting the bgen based on the set of SNPs that are in that score file that we need, the bgen.bgi file for indexing, and then just optional set of extracted chromosome SNPs. And this is what the command looks like for chromosome 1. This is an example for chromosome 20. Just for reference, you can see that this is 20, 20, where every part where the chromosome is mentioned, it's chromosome 20. And the corresponding files for chromosome 20 have been uploaded into that environment. So we can run these commands using these that are required. And even for the BGenX, it may not mention the BGI right away, but internally it does need to have that BGI file. Otherwise it'll complain that, oh, I can't find the indexing file, like where's the indexing file? So it really helps to kind of mention, the indexing file that is there, so that when it's running the command, it'll say, yeah, that's where the indexing file is. When we're going through the BGen, it's going to want to find its buddy. It's kind of like a cat looking for its owner, its buddy. And this is the outputs for chromosome 20. So we see here that the extracted files will be in the extracted genotypes BGen subfolder. So we had given the output folder in the code here, Saniya tutorials extracted genotypes BGen. So that's where they will be here, as you can see here, that this is where those files are. And it's specified in the output folder. And that's when we run the dx command and we give it the destination of output folder. That is telling it where the outputs will go. So step 4, we're going to be converting the BGen file to a bed file. So it's a plink1 binary format. So going from the BGen and the BGI files and using the .sample information to come up with the bed, BIM, and FAM files. Those are plink1 binary format files. So in the process, we'll be running bash step 4 create bed file all chromosomes.sh. So just for those 11 chromosomes that we were using for our score information in this case, but this is what will be what we're running in step 4. So yeah, we're going to be using the plink2 tool to convert the BGen files to the plink1 format binary files here. So what you see here, the binary formats, we have the .fam file, the .bed, contains the binary version of the SNP info. It's not in a format readable for humans. It's kind of like alien language a bit. And the BIM file here, like what are the variants that were extracted. So what you see over here is in this plink2 command that we'll be using, plink2 bgen, file a bgen, ref first sample, file.sample, make bed out prefix. Ref first is going to tell plink2 that in the BGen files, the first allele is listed as the reference allele, which is usually the UK Biobank convention. So that's important to note. And so this ensures that the allele alignment is correct. So when it's guessing, like, is it the reference, is it the alternate, right? And the sample file.sample, it provides the sample IDs, like the gender, the missingness information that is required for the BGen. And the make bed converts the BGen to Plink's binary format. So the bed file, the BIM file, and the FAM trio. So it's a trio, 3 musketeers. And the out prefix is going to give the out prefix for the files. Like, what are these files altogether going to be? Because they'll have the same prefix. So it'll be prefix.out prefix.bim, out prefix.bed, out prefix.fam. So what is that? So this is the code that we're going to run to create the bed file and BIM and BAM file from the subset BGen files. This is the code. So it converts the extracted BGen files for chromosomes 1 to 22, based on if they're found or not, to the plink bed format using plink2 with the correct ref first flag. So this is after we've created the filtered BGen files, based on the PGS catalog SNPs. In this case. So in UK Biobank, again, the reference is always listed first, so we use ref first. For instance, if we have this format here, where you see the T and the C, the first allele, the reference allele from GRCH37, this is the T, that's the reference, and then the alternate allele will be C. So the first allele will be the T, which is reference, and then this was alternate. And then this will be matches the UK Biobank convention, because the reference is always listed first in that. So make sure you check if you're doing this for anything else in general, how you do that. And then as we run this code, so similar to the third step, what we're trying to do is, so we're running a loop, so from chromosomes 1 to 22. And again, there could be other more efficient ways to do this. This is just how I'm doing it. So feel free to, give any feedback as well. But here, this is the same defensive step where we're going through all 22 chromosomes. I could have listed out the chromosomes, like for CHR, one and all of the different ones, but I want to make this dynamic so that because different score files will have different chromosomes that they could have scores for. So this is just dynamically checking, and now we're checking that if the bgen file exists. So it's the bgen directory here that we have, the one we're working with, the subset bgen. It's found in the extracted genotypes bgen, what will be created in the third step. It's checking in this folder, that's the bgen directory, if the bgen file name. So the bgen file name is that file name prefix.bgen. The file name prefix is going to be this PGS ID here, the genome build UKB 228828 subset, this one, the one that we just created. You see over here, this is what these files look like here. And we're focused that if the bgen file exists, the .bgen. And of course, if the bgen exists, then the .bgi will also exist because it's an indexing file for the bgen, but it's checking, we have for the 11 chromosomes. This is 22 because I was just filtering for bgen, so we have a bgen and bgi, so 11 times 2, 22 here. But anyways, it's searching if the bgen file exists. If not, then it says that the filtered bgen file for that chromosome is not found. It's skipping for the for the bed file. It's not going to be running plink2 otherwise. And then this one is going to be running the plink2 command otherwise. And we're going to be outputting our results in this destination, this output folder, which will be Saniya tutorials extracted genotypes _bed. That's where it will go. And we are going to be uploading here the bgen directory, bgen file name. So we're going to be bringing this bgen file name here, and the sample directory, sample file name. So this is the sample directory, the UKB imputation from genotype, and the sample file name, which will be the .sample. And again, we need the absolute file names here. We need to know where those files are located. When we're mentioning the bgen file name up here, we need to know where this came from. This is coming from the extracted genotypes.bgen, which is in Saniya tutorials. That's what we're saying, that when we mention it, this bgen file name was found there. When I'm mentioning sample file name, I really want you to upload it from sample dir here. And when I'm mentioning the output folder, I need you to be very specific and tell me exactly where you want this output to be, this output prefix file to be saved. And this extract command is run over here. That's what the ICMD is doing. So it's using the plink2 tool, using the Swiss Army knife, as you see here. So what you see here is we are creating a plink1 binary file set, like plinkton. So if we look at the plink2 in the data information, so plink2 bgen input, this kind of thing, for instance, what it's doing is that the make bed creates a plink1 binary file set. So that's what we're going to be doing. The plink1 binary file set, the BIM, the FAM, and the bed. It makes its bed with that one, trio. So now in step 4, we're going to be working with this new subset bgen files that we have, and the BGI files, the indexing files for the bgens. And we're going to convert those for each of those 11 chromosomes. We're going to convert those to the plink1 format files, the bed, BIM, and FAM files that we need. So to do that, we're going to see that here we're working with this folder. Now, no more the UKB imputation from genotype files because we've subset these. These are tinier. You can see how just, bytes, not like giant gigabyte, just small little bite-sized files that we can work with. So we're going to go here and we're going to go to the step 4. And what we're doing is we're going to be going through each of the chromosomes. And if the BGen file is not found for that chromosome, like because we only have it for 11 chromosomes. So this BGen file DX is this BGen directory, which is the extracted genotypes BGen. That's what this is. The Saniya tutorials, we start here. Extracted genotypes BGen. And what the name of the BGen file name will be, the file name prefix, which is this PGS ID, which would be, the file prefix would be this ID, which will be the PGS 00053. Then the genome build, which is GRCH37. Then the UKB22828 subset. And then CHR, and then let's just say chromosome 1. That's so wherever the $sign and the brackets are, like that just is a placeholder that you put the variable name in. So when chromosome 1, CHR is 1, right? So CHR 1, and then the RSIDs. That's what the prefix is. So it's going to be searching. So this is if CHR equals 1. In this case, then this PGS ID comes from here. You just pluck this in here. That's what this guy here is. And then this GRCH37 comes from this genome build here is the variable. And there should not be any spaces between the name and the variable in Bash. Otherwise, it'll give you a warning or an error. It won't work. It cannot have any spaces. It's just like in Python, like how you can't, like, it's very tabs sensitive and whatnot, like for indents and stuff. So it's going to see if this file exists because this is the name of, for a given chromosome. It's this is the format of it. The PGS, GICH, UK Biobank extracted SNPs, this .txt. That's what it is. So if that file, if it's null, that means that file does not exist. It's listing it and trying to see, does that file exist in this directory, in this folder? And if it doesn't, that means that that chromosome does not have any score files. So we should just skip it and not do the conversion. And just a quick check because, I could have hard coded for chromosomes in 1, 2, 4, 5, I could have hard coded the chromosomes, but I want to make this that it can apply for all sorts of cases for your data, my different traits and so on. So that's why I'm doing it this way. And then we're going to be running the plink2 command. If that bgen file, that subset file exists in that folder, in this folder here, then we're going to be running the plink2 command on it. And we're going to be making that bgen file into a bed file, and then also outputting with it files that begin the same essentially, but it's going to be a bed, BIM, and FAM files. So that's what this is going to do here. It's going to submit this job here. And again, we're uploading the bgen file. We're uploading the subset file as bgen file name that has come from the extracted genotype. So for that chromosome, we are uploading that file name, and we're also uploading the sample. So the sample information remains the same. Only thing we have done, all we did is we just have subset the bgen file for this set of SNPs. We have retained all the samples so far. We've retained those samples that we have genotype information, the participants, the humans. We've retained that. We have not filtered it. So the sample file would be the same as the original file. So what we're doing right now is we are just going to be using the sample files that are located here in the imputation. Let me show you here. We're going to be using these sample files in imputation. We'll be using these sample files for the corresponding chromosomes. That's what we'll be using. We'll be uploading this. And that's what we mentioned here. We'll be using the samples because it needs the sample information here. So the sample directory is the bulk imputation, UKB imputation from genotype. So you note that we don't have to mention this IBD project Saniya part in front. We don't need to do that. It just needs to start off from the current folder. So bulk is where it is. Saniya tutorials is this other folder. So we just need to do this. And again, this is the local file name here. And this sample file name is the local sample name here for that chromosome. So for chromosome 1, for instance, it would be, this is what it would look like, which is what this is here. You'd put C1. And what you notice that when we are uploading it, for sample file name, we're giving the sample directory. We're giving the complete directory here in the DNAnexus job, so it knows where those files are. So it can upload those into that environment when it's running this job. So now I'll show you how we run it. And right now, just to clear up all of this, I'm going to do ls. Oh, X, ls. And I'm going to do bash step 4, create bed file allchrome.sh. So it's doing this, converting bgen to bed for chromosome 1, for chromosome 2, chromosome 3. So it's skipping it for chromosome 3, for skipping it for chromosome 4 as expected. It's doing it for chromosome 5. So you can see where it's skipping it for like 9, 10. It's going through this as expected. And each of them is having its job whenever it's able to submit it. You can see the echo is what's being printed, right? Like it's printing it out, like submitted link conversion job for chromosome 19 here. There's a little smiley submitting the job. So that's what's being printed to the console. So now you can see these are the other jobs, the PGS extract parts. Now this is bgen to bed. And that's the tag that I put here. That's what this tag is saying, bgen to bed, and then the chromosome CHR. That's just telling me that for chromosome 1, and this is the command it's running, extract command ICMD. So for a given chromosome, it'll be like chromosome 1 will be this one here. So it's just waiting, and then it's going to be running them for these 11. So this was the previous one we just finished. Now this is this new one, converting the bgen to the bed files. And what we will notice is that I'm going to be putting the new outputs in extracted genotypes bed. Right now, that folder does not exist, but I'm going to be creating it. And I know I could have done this in the simple starter example. I could have put them here, those folders. But I just sort of wanted to show you, again, how we can also just still do this in this job, just to spice it up a bit. And you'll notice that now that it's starting, it's created this bed. It's empty right now, but it's starting this process. It's waiting, and I've given it like a high priority so that it runs a bit faster. I mean, it may cost a little bit more, but just so it runs more efficiently. So yes, let's see what happens now. So for instance, for chromosome 2, what we will see is that, we had these extracted SNPs here, these 2 files here that we had, and we had extracted them and saved them in this bgen.bgi, the index file, and the bgen file here. And it was filtered for the SNPs of interest, which were these 2, in the previous step 3, we had filtered them. The initial UK Biobank, the genotype data, we had filtered them for the SNPs of interest. And so this is what we're using for the inputs, the bgen file and the indexed file. And we also have the sample, the .dot sample. So this is the original sample file for chromosome 2. Because we did not do any subsetting for the chromosome, for the given chromosome, we did not do any subsetting by the samples. So we kept all the original samples that we had the data for. So that's why over here, we're just using the original UKB imputation from genotype .sample. So these are the inputs that are going in, and we're using the .sample and then the bgen information. We're using that, and we are supplying that to the plink command in order to create the bed file, the BIM file, and the FAM files. So we're going to be running the job for chromosome 2 here. And as you see here, it finds these two variants in the header, those two ones that we had found for chromosome 2. These number of samples imported from the dot sample file, so many females, males, ambiguous. And again, your numbers may differ based on your UK Biobank project and information, but just it should be in the ballpark of around 500,000 samples as of now. So then ultimately it says that writing this .fam done, .bim done, and .bed done. So then it's uploading them to the output directory that I've given. So it says home, DNAnexus, out. And this out is what folder we've given. But internally, it's read as that code. It's a destination that we'd given when we run the DNAnexus dx run command. It's outputting the log file, the fam, the bed, the bim files to that area. So you see here, the input ones that we were using are the bgen files and the .sample. Here it didn't use the indexing file because it's just converting from one to the next. When we were doing the subsetting using the bgenex, it needed the index files. But here, because we're just kind of converting an existing bgen file to the sample, and the sample to those formats, it doesn't need to use any indexing because it's not subsetting anything anymore. It's just taking the file and then converting it to a different format. So it doesn't need the indexing file. So it just takes the bgen file and then the .sample. So for chromosome 2, this is what it's reading in its command. It's figured out the bgen file and the .sample. And then its output, the log, the fam, the bim, and the bed files for that chromosome. That's the out. And this is the in. And this is the command that was run. And that's how much it costs. So what you see here for chromosome 2 is going to output the fam, the log, the bed, and the bim files for it here. In this output directory, the files are in the extracted genotypes bed folder. In Saniya tutorials, in the extracted genotypes bed, I'm just filtering for CHI2, so I find these 4 files there. Oh, now let's see the status update of this job that we've run. Let's see how that's been going and if it's finished. Okay, so let's just see this here. We see is now, yay, this job has finished for these 11 chromosomes that, in our score file, it has finished converting the bgen to a bed file. Yay! Woo! Okay, so let's just see what, for example, like what this looks like. Let's just go on in and see here, for example, for this one. What we've seen took around 2 minutes, is more efficient. It's just a very quick computation. So over here, what we had specified here, and you see this, okay, is that we'd given these 2 files to be uploaded, and these are the same 2 files here that we will find over here. And then we noticed that the first one is the bgen, and the second one is the sample. And we've given the information for how those can be uploaded, where those locations are, where the bgen directory is the extracted genotypes bgen, that filtered file, and this sample directory is the original samples. I mean, because it's the same, we didn't filter any of the samples when we were subsetting the bgen file. We kept the samples the same, so we can just use the original again. If we had subset the samples, we would probably be using like extracted genotypes bgen sample. We would have done something differently in the step 3. So now this is the command that has been run for chromosome 1. This is the plink command. So let me zoom out a bit. This is the plink2 bgen. We have the bgen file name here. We have the ref first. This just means that the reference is always listed first here. So like if we have, if this is what our bgen files look like here, that this T is the reference allele and the C is the alternate allele here. Like if this is what our bgen file is looking like, the variant information, RSID, chromosome number, position, and so on. And then this is the sample here, and this is the sample information, which is what the sample file name is. Then we have our make bed here, which is what this is over here. Then we have the out. And then the out is just using the same heading, the file name prefix, the same thing here, this file name prefix. I just wanted to be the same. So how it was reading as the bgen here, I just want the outputs to be, you know, all sharing this same information here. I want all the outputs to have this as part of them. So all the outputs to have this in the stem. Oh, eeks. This in the stem of the name. That's what this is here. And so on. And we have the outputs. We have the log file, the FAM, the BIM, and the bed files here. That's what it is. That's what you see here. And so for instance, and that's what the command is saying. So we'll have this log file. And this log file will basically show us. And this again, guys, this is just really dependent on your UK Biobank data. So this is just for mine. Please don't worry if your numbers are different. If you're following along and trying to do this example with me, it may differ based on your project information and so on. So a random number, this is when I was running it from the UK. This is the information here. We've got so many samples. This is what I have. So many females, so many males, so many, we don't know what the gender is. Many founders here, this number is same as the samples. And these all add up to the samples here. And then what it's doing is it's found this one variant for chromosome 1. Remember, like in the subset file, we just had one variant that we were wanting for this chromosome 1. This just had, in the get info, this just has this one variant, right? So that's what that's kind of alluding to, that just one variant was loaded from this. So it's created the FAM, BIM, and bed files here for chromosome 1. We have the FAM here. You see over here. We have the BIM. Just telling you about this one variant, that the same variant that we were wanting, the same guy here. And we also have the bed file. I also recommend being careful about what you can download given the permissions for UK Biobank. Please be mindful of that. So here you can also see this is for chromosome 20. Everything is chromosome 20 specific. And we also have the log file. This log file is going to tell us that, for this chromosome, again, we just found one variant and we've got the corresponding files here. I remember that like for chromosome 14, we had a few variants. So let me just show you that example here. So in the log file, this log is kind of like all the processes that we did. Again, just the same number of samples. It's just the same, the sample files. It has these two variants loaded from it. And for chromosome 14, we have these two variants. And I recommend just double checking that you find all the variants that you needed. We found those two variants from that. And in fact, just to show you that that's what was also picked up, we have the BIM file here. Just these two variants over here for chromosome 14. And so on. So that's what we would be seeing for all these different runs, chromosome 11 here for the log file. 4 variants over here were found. Again, the samples are the same. And that's the log file is saying what was happening. We have the BIM file, we've got the FAM. Now, these are the 4 variants that we'd picked up. And you can see that all of these are chromosome 11 specific. Chromosome 11, and these were the 2. Because that's what we're doing in the loop. We're going through each of the chromosomes and seeing if they do have this bgen file in that folder and the extracted genotypes bgen, and then we're putting them in the bed folder. So let me show you now what that bed folder looks like. Now we can see this is Saniya tutorials, and we've just created this bed folder in the process. And now we have the log file, the BIM, bed, fam. So we have these 4 files for a given chromosome here. We've got these 4. And for the 11 chromosomes, that leads to 44 files, 4 times 11. So we now have all of these. And so step 4 is done. And then what we will want to do now is we're going to want to gather all of these files together across these 11 chromosomes in our case. We want to gather them all together and stitch together all the BIM files into one, all the bed files into one, and all the FAM files into one. So that's where we want to merge the bed files. So one FAM file, one BIM file, and one bed file. And it's going to be easier, like for different applications, like when we're running the PGS calculator. It just makes it a little bit easier if we just have one file, I think, just it's easier to work with. So let's see how that works now in step 5 next. So the output files are a Plink binary set. For each chromosome, it produces 3 linked files and a log file, a .bed, a binary genotype matrix. So a compact binary file storing genotype called 0, 1, 2 copies of the alternate allele. Rows are individuals, columns are variants. .bim is variant information, and .fam is sample/individual information. And the log file is the process that was run on DNAnexus, just for our reference. Together, these files form the standard Plink dataset that's widely used for genome-wide association studies, so GWAS, quality control, QC, and downstream genetic analyses. As we will be doing like polygenic risk score predictions. Yay! Welcome to the fam! So we went from BGen to bed and fam and BIM files. So yay! Welcome to the fam! This is the new genotype information we're going to be working with. So an example BIM file here. So if we go to the extracted genotypes bed again for chromosome 2, this is the BIM file. So it's a tab delimited, so it's separated by /t, tab separated text file with chromosome 2, variant ID like the reference SNP ID, the genetic distance cm centimoigens, usually 0, the base pair position, allele 1, which is reference because we use ref first, that's what this is here, and allele 2, which is the alternate allele. This is an example of the .bim file. This is an example of a .fam file. So .fam, it has a sample and individual information. So 1 line per individual in our data. So 1 line corresponds to the given sample, like the given person. They don't have these headings on the top. I just added these in for reference. But this is the family ID here. And in my case, the family ID is the same as the individual ID. This is the paternal ID here, the PID. This is the maternal ID, MID. Here, these values are 1 if male, 2 if female, and 0 if missing, as you see in this column. And the -9 for phenotype means that it's missing and it's the default. And you might have seen in one of the log files that said like the phenotype data is missing. It's all good, don't worry about it. But this is an example of a .fam file, so about like the sample information, like family, paternal, maternal. That's what fam is, family. And so as you see here is an example of the log file for chromosome 2. It'll also be mentioning how long it took, and so on. No phenotype data present. All the stuff that's happened here in the log file, what was run. Options in effect, this bgen, ref first, make bed, output, and the sample. So it's running them as a set of commands using Plink version 2.0. So we have the Plink2 formatted genotype files here. These are all of the files. So we have 4 for each given chromosome. We have like 4. Yeah, like for chromosome 20, these are the 4. So you can see 4 for each of the 11 chromosomes that we have those score files for. Score information for needed. It's 4 times 11 chromosomes, so 44 items. It checks out. So this is where the files are all stored for the extracted genotype files. So for each of those 11 chromosomes, we've got the BIM, BED, and FAM files, as well as a log file for reference. And again, the reason we did this is the current input genotype file format supported for PGS C calculator are the Plink1 file set, the .bed, the .bim, and the .fam. That's how we did this conversion. So we moved from the BGen to this format. And now, give me 5, little guy. Woohoo! Awesome. The step 5 is to merge the genotype files across the chromosomes. So merge those into one set of files. So we're going to be merging those BED files, the BED, the BIM, and the FAM files into one across those chromosomes that we have. So just one set of files. It'll make things easier for us. So now what we're going to do in step 5 is we're going to merge all these files, all these BED files into one, all these BIM files into one, all these FAM files into one. So we just have one file across these chromosomes. So we're going to use the Plink2 command again. And in this case, what I'm going to show you is how we're going to do that. So we've got these different files here. We've got the BIM, BED, and FAM, 4 for each of the 11 chromosomes. So we're going to go to step 5, merge all BED files.sh here. So what we're going to do is we're going to run this command over here, and we're going to go into this input folder, which I'm showing you is the extracted genotypes BED with all of these files over here. Oh, wait, no, gosh, what happened? Okay, we're going to go in here, all of these, and then ultimately we're going to create a final folder that's going to be called merged genotypes. And you see that right now it does not exist, but this is going to be the output folder. And we are going to tell this here, this destination to the DNAnexus command is going to say that we want our output. Whatever is the out name, whatever is being saved here, the out name is going to be all chromosome SNPs merged, this PGS ID here, this name of the PGS 00053 and the genome build GRCH37. And again, this is just my naming convention to make sense for me, just to know what genome we're using and why, and like what's the ID. You can use whatever you'd like. This is just for me. We're all different. So again, just see that. Then what we're going to see over here is that the out name is going to be over here. And that will be over here, but it's going to be found in this folder. So we're going to have an out name that will be the BAD, BIM, FAM files here. And we're going to be having this SNP here. So let's see all these things here. Now let's run this. So let me just go all the way down here. So ls again, just for reference, so bash step 5, merge all BAD files.sh. So it submitted the merge job for all chromosomes. So just one job. So this is the ID here, so we can track it. Let's track the little guy. Just one job here. New Plink merge. So this is the same. This J2F. This thing is the same as this over here. So we can just see what this command is doing over here. And what we will notice is that it is essentially hunting, it's finding the list of chromosomes and file names inside. That's what this is doing. It's trying to find the name, and it's creating a folder that's a file that's called merge file paths name, which I'll explain. And then we're going to be merging all of those files that are in the file merge file paths name together and make the bed. So this is the merging command to merge all of them together. And yay, that happened pretty quickly, I think. Let's just see where that one is. We find the merged genotypes has been created already. I'm going to go back to the monitor and just going to do the past hour just to make it simple to find this. And we're going to see that it executed and it's done. If it failed, we'd see the red. And believe me, I have seen many, many, many fails on the process. So I'm trying to make it so that hopefully you can learn from some of my trials, errors, and tribulations. So hopefully you won't find that red fail as much as I did, as many times as I did. So what we will see here is that we get these output files here, show all inputs. So let's just see here, we have the bed files to merge. This step 5 file, that's what we had mentioned we want it to be created. It's creating this here. And that's what this is here. It's in the merge genotypes. And it's telling that this is what we need to merge. It's, for the merged list command in Blink, we need to give it a list of the prefixes of the files that we want to merge. And what you'll notice is that in the extracted genotypes bed, that these were all the prefixes for the different files. So whether it's a BIM, bed, or FAM, it all had the same prefix here. They all were given the same prefix. Oh, wait, let me just undo that here. So like, for instance, this just all has the same prefix. And like, these all have like the same prefix and so on for chromosome 14 and so on. But you can see, and also this one too, I forgot, these above here, they're all part of the same one. And this log as well, but it's just a log file. But you can see they all have the same prefix. And what we were mentioning here to this is we're kind of telling it that it's in the extracted genotypes bed, these files. And these are the prefixes. Remember, these are the prefixes. They're just the stems are all the same. The endings are different, right? BIM, bed, FAM. But the same thing is the same for like chromosome 18 RSIDs. What we'll see here is that chromosome 18 RSID is the same thing here, and it's found here. This is mounting this project. I know it's a bit confusing. Like sometimes you're wondering, well, why is it mount project and so on? And that's also part of what I'm trying to help with is like for my trial and error, like what I got to help with the DNAnexus commands to run, because I was like, why is it like why is it a mount project, or why is it this, why it's that? And it's still stuff that I'm learning and figuring out, and I appreciate all of your advice and help here. But that's one of the things that also is a bit confusing. Like sometimes we just give Saniya tutorials extracted genotypes bed as for it. Sometimes that's all we're giving. And then other times we're giving mount project Saniya tutorials extracted genotypes bed. But that's sort of like what we are putting into this text file, and then that is what the plink command is reading. That's what it's reading here. This is what that command is. And then it creates the corresponding files for all of these chromosomes. We have this log file here where it's kind of going through and it's merging them. And what it tells us is 21 variants loaded from BIM file. And what our score file had was 21 variants. So we have recovered all of the 21 variants that we were wanting. If you remember, in our score file, we have a number of variants, 21. These were the 21 variants. So we've recovered these 21 variants. And it's merging this here, and it's writing them to this bed file, BIM, and FAM file over here. So what we can observe here is that in the BIM file, we'll see all the variants, and we will see all of these 21 variants that we were wanting. And then we will see in the FAM file details about each of the samples. And again, mine will be different from yours, my numbers. And just this is the information here. And it just sometimes just gives a head, so it doesn't display all the information. So what you observe over here is that for the BIM file here, for instance, we take this and I'll trash him into this little guy here. And we have all the 21 variants. I know it looks a little confusing because you may not see that some rows are duplicated with some variants, but you see here, these are all the 21 variants for our score file, and we've recovered them here. So now what we have is we have this folder of merged genotypes. We have our FAM, log. We have all like the files that we've merged. This is just what we were using internally for this process. We have our bed, our BIM, and other information here. And so this is what we will be using going forward into the PGSC calculator. But now we finished step 5. High five, little guy. Woo! Awesome. So what this is going to do, this is the step 5 merge all bed files command, the bash script. So it uses DNAnexus to run the merge job remotely. So running it on the DNAnexus platform here. And you see here, the Swiss Army knife is the DX app for running arbitrary shell commands, all the ones of interest here, like the Blink ones. So this ICMD one, here this Blink merge command, it passes the Blink merge pipeline command here, this one that we've run. It's been passed in here. This tag is going to attach a tag for tracking, which we can see in the portal. This is going to be one job because we're just merging all the chromosomes. The instance type, again, is going to pick a compute node that has, in this case, 16 CPUs and solid state drive. Priority high ensures faster scheduling. It's slightly more expensive, but it ensures that your job gets pushed to the front of the line. And brief yes means it's a non-interactive submission. You're not going to be interacting, you're just going to be observing. So this script here, so that's the DX one, that's the merge job command on DNAnexus. But what this script is going to do is it's essentially, in the first part, it's going to be finding all the BIM .bim, .bed, .fam genotype files in our input folder that's mentioned here. Then it's going to build a merge list file for Plink. And then it's going to submit a DNAnexus job that runs Plink to merge all those files into one binary data set. So just one .bed, one .bim, and one .fam file. And it's going to save the results in the destination in the output folder, where the output folder is going to be Saniya tutorials merged genotypes. That's what it's going to do. So what you see here in the Plink merge command here, this part here, is it's a compound command. So this ls mount project input folder /**bim. I know it's a bit confusing. This is like mounting a project. It's going to list all .bim files, which is a companion to .bed, .fam. So they all have the same set of prefixes as you see. They all have the same set of prefixes. And what it's trying to do is it's trying to list the files. What files are in that folder? What files are in this extracted genotypes bed folder? What are the names? Then what it's going to do is in this part here, after this pipe here, this sed-e, this part here, is going to strip the .bim extension, leaving the base prefix .bed, .bim, and .fam all share this prefix. So it's just, I could have done this for .fam or .bed, because the .bim, the .bed, and the .fam files, they all have that same prefix. It's same output prefix .bim, output prefix .bed, output prefix .fam. They all have the same ending. So I just decided to use .bim. So inside the input folder, inside this folder, just list all the .bim files and get their names. Then from their names, because I'm doing this for all the .bim files, I'm just going to remove the .bim here. I'm stripping that the ending from the end of the file name, and I'm just trying to get everything with the .bim chopped off. If I did this for .fam, I would have put fam in here and to strip the .fam extension. If I did this for .bed, I would have put bed in here to strip the .bed extension. I just want the part before the ending, because all of those three types of files, they have the same beginning, but they're just .bed, .fam, .bim. So that's what this is. And then what I'm doing is I'm getting the list. So you see here that these are actually what those files are. So we had each of these files .bim for this one, the chromosome 11, .bim, .bed, .fam. We have those ones. So this is what we get when I strip the BIM extension. And then I'm redirecting this output into merge file paths name. So that's what I'm redirecting here. This merge file paths name, which Plink needs as a merge list. So I've gone through the folder. I've gotten a list of those files that are in this folder in extracted genotypes bed. I've gotten a list of them, and I have gotten them without those endings. So this will be the merge list. This will be telling Plink that these are the set of files that I want to merge together into one BIM, one bed, one FAM file. That's what this merge file paths name is here. This what this is here. And that's when I use the merge list. I'm calling the merge file paths name. I'm calling that. And this merge file paths name is here, bed files to merge. Then I have the PGS ID here, genome build step5.txt. That's what this file is when I finish running it. So then the command will do is in this part, it's going to run Plink with merge list, what I just created. It tells Plink to merge all data sets in that list. So the make bed is going to give the output in binary Plink format, .bed, .bim, .fam. So it's going to be taking the .bed files that were found in that list and combining them into one bed file. All the .bim files in that and putting them into one .bim file, and all the .fam ones and putting them into one. And the .output is going to be the merged output base name. This .out name will say that for those 3 files, the BIM, BAM, and FAM, the BIM, BED, and FAM files, for those final files that are combined across all the chromosomes, what is the out name? All chromosome SNP merged, then this PGS ID is here, and then the genome build. So it's going to be the names that is going to be the new prefix .bem, new prefix .bed, new prefix .fam. There's a compound command, and you can see here with the semicolon. So we run this bash step 5 merge bed files.sh. This is the job here. You see, this job is the same as this job over here. They're matching. So it says submitted Plink merge job for all chromosomes. The same job ID on the dx command when we do the bash step 5 merge all bed files. That's the same. So we just have this one job running for step 5 because we're just merging up from them for all the chromosomes. So this is the inputs here, and we're telling it what to do. And these are the outputs. So it'll be creating the bed files to merge. GRCH 37, the step 5, this is the file that it creates, and then it runs it within. And then we have the log file created. We have this file, the bed, the FAM, and the BIM files. That's what are being created. So just the one set, and it's combining them across those chromosomes, those original files. So for those 11 chromosomes, it's created this. So you can see again that these are the bed files to merge that we were using. This is sort of telling it, go find those BIM, those BIM files, the FAM, and the bed files for each of these, and then for the, kind of combine them systematically. So what you can see here is that this is the log file. This is some Star Wars. I'm a Star Wars fan. No need to tell me about your mother. It's all in your logs. So you can see that its options in effect are, make bed, merge list, out all chromosomes. This is the output file. So, it's found so many people, and it's gotten all the 21 variants. In that score file, it has 21 variants for late onset Alzheimer's disease. And this one here has found it. So it's found these 21 variants, and it's written them as the this output name .bed, .bim, and .fam. 21 variants. So that's what it was doing. So we have one set of output genotype files now. And you see that they all have the same prefixes here. This all the same. Everything before it is all the same. With the FAM, the BIM, and the bed. And we have the bed files to merge and the log, but we have the genotype information here. So yeah, hooray! So for instance, we can check that our merged BIM file here, this one, it has all the 21 variants here that are needed based on the score file. So you can see them across all the chromosomes, these 11 chromosomes, these 21 variants for the late onset Alzheimer's disease score file for calculating an individual's genetic susceptibility to late onset Alzheimer's disease. So we can see that we've captured all of them in the BIM file, and then we have the FAM file, which has all the sample information that usually stays the same. And then we also have the bed file, which will have the genotype information for these variants for those samples. So now step 6, running the PGSC calculator. We're on step 6. Awesome. So what we see here is that we are going to be doing bash step 6 pgsc calculator.sh, and this is now where we're going to be running the PGS catalog calculator. That's exciting. So the first input, one of the first key inputs that we need is to set up a sample sheet for PGS catalog calculator, and based on the documentation here. So the sample sheet describes the structure of your input genotyping data sets. It's needed because the structure of input data can be very different across use cases. You can have different file formats, directories, and split versus unsplit by chromosome. So for example, it's a CSV comma separated file here. And you'll have your sample set, you have your path prefix, like where do you find it, like where's that genotype file located, what chromosome, and what's the format, just to guide it. And the requirements of the sample sheet CSV file are as follows. So the sample set is the identifier for the batch or the sample group. It can be a string, and it's mandatory, you need it. But please do not put any underscores here, and I'll show you why. The path prefix is the path prefix of the genotype data without the file extension. For variant calling format, omit the .vcf.gz. For p file, omit the .pgen, .pvar, .psam. And for the b file, omit the .bed, .bim, .fam. So it's a file path prefix, the string, and it's mandatory. Chrom is a chromosome number of that file, that genotype file. Like, is it just for one, or is it like merged? If it's empty, it processes all chromosomes. Like, that file corresponds to all chromosomes. In our case, since I have created those merged files, one for all of the chromosomes, I'm going to keep it empty. It can be integer or empty, and it's optional because if you have that one file across many chromosomes, it's going to be empty. And that's also kind of why I was merging the files in the previous steps, because I don't want to have to keep listing, the files like 22 times or 11 times or however many times. And the format is the genotype format. It can be, as of now, like P file, B file, VCF, and it's mandatory. You need to tell PGSC Calc what type is the genotype data. So here are some examples of sample sheet CSV files, comma separated files. You can have a link to P file, and you need to have the first row having these headings: sample set, path underscore prefix, chrom format. Sample set, path prefix, chrom format. You need to have all of these. You can have a link to file, P file, it's pgen, pvar, psam. You can have the Saniya target genomes. This is like the path prefix for chromosome 22, and it's a P file. You have all three of these. Or you can have a variant call format. It's a .vcf.gz with a .tbi index. So again, the same headings, and so you can have Saniya, target genome, Saniya, synthetic subset, like the same file path. And then if they're like .vcf.gz, you can say, okay, this one is for chromosome 22, and it's a VCF file. If you have multi-chromosomes, like what we have, like, that genotype file, it's like this one means that it's just for chromosome 22, that one. But if you've got multiple chromosomes, then you would just have this chromosome being empty. This it's just empty there, which is kind of what we need. We need to borrow that idea of the empty chromosome because we've got multiple chromosomes in our file. And we're working in this case with a plink, B file, a bed, BIM, and FAM. So we're going to use the sample set, a Saniya, path prefix, same thing. The format is going to be B file. That's why we use B file, and we're going to make this empty because we're using the multi-chromosome. Otherwise, we would have to mention like all the different chromosomes, and it gets kind of tedious. So building a sample sheet. At first, just to show you how it went wrong for the sample set, I put like UK Biobank underscore this here, and that's wrong. It's wrong because you can't have these underscores here. If you try running it, you see in the Swiss Army knife, you will get failures. So I know I've been showing you how things work out. Believe me, I've done a lot of trial and error. And when I tried running it, it would say pipeline completed with errors, and I was like, huh? And then it says, do not call your sample set reference or use underscores in the name. So then I just did this. But like maybe instead of sample set, I could, like being UK Biobank, I could have just put this ID in here. You can change it, just don't have any underscores, and just don't have any underscores. So my sample set looks like this. So we prepared the sample sheet CSV file here. As you see here, I have the UK Biobank here. The path prefix is all chromosome SNPs merged, this PGS ID here, and then the GRCH37, and it's a B file because we're using the FAM, the BIM, and the BED, like all of these ones here. And all of them have the same prefix here. The all chromosome SNPs merged, this ID, and then this GRCH. So that's what I'll put in here. When I'm putting them in here, because I'm running it on the DNAnexus on the cloud, then what I want to make sure, since it is on the cloud, I'm not going to be giving the absolute file paths, because for the sample sheet, I'm just going to be giving the relative paths, because when I'm running PGS Calc, it's going to already know to go to this folder, because I will be putting it in the DX run. The DX run, I'm going to be telling it that this genotype information, that these files, where they're located. I'm going to take care of that later on. So right now, I just need the regular names here. This is just sort of one of the ways it works on the DNAnexus. And here again, you'll notice that Chrome is empty. Here, I leave it blank because this file has the merged genotypes information for all the chromosomes we need. That's why I leave it blank. Now we're getting to step 6, the real big thing now. Getting ready to run the PGS catalog calculator. So now what we're going to see here is that we have the following information. We have our merged genotypes information here that we've just created in step 5. So this is our Plink1 binary file format that we're using. And we want to create our sample sheet accordingly so that we will tell PGSC catalog calculator where to find our information. So we're going to tell it that this is where our files are for our genotype information. And because I have compiled them across all the chromosomes, we can just have this really simple list for our sample sheet. So if we go over here, we see that I've created a sample sheet, followed the examples, and if I open it up in Excel, this is what my sample sheet looks like over here. We have the sample set. We have to keep the column names. So we have the sample set, UK Biobank. Please do not have any underscores at all in this name, because I did, and I got a warning and an error before. It doesn't do well. I just put UK Biobank. You can put whatever works for you. A path prefix. So this will be telling me that this is where the prefix for the files. So for the genotype files, just close this little guy here. For the genotype files, this is what they are starting with. And the sample sheet is needing the local names, not the absolute, the IBD projects, Saniya tutorials, merge genotypes. No, what it's going to use is it's just going to need this prefix here, this prefix name. So it'll know, it'll use the FAM, and it's going to use the BED and the BIM files here. These are the Plink1 formats. And that's what we need to tell it. Please use, this is the path prefix. This is what this is here. This is what this little guy is, the same thing here. And because this is all merged for all the chromosomes, it's one file, we don't need to specify anything here. So we don't need anything here because we are using one file. So we don't need to specify anything for the for Chrome over here. So we just leave it empty, because this is merged. If we had like a chromosome 1, 2, 3, if we had if we'd gotten like this extracted genotypes bed, we would have had to specify like all the 11 different chromosomes in this example we would have had to put like chromosome 1, 2, and so on. And we would have had to adjust this name. But that's why like I wanted to merge them just to make it reproducible so that you, because your sample sheet can essentially look like this for your score and your build. If you've merged them and the other code has taken care of that, it's just easier, I think, just to have this one file. You could have had this, like we could have had this for this other file here. It could have been like, let me get this guy's name here. Like if we wanted to, we could have done this. And then it would have been like chromosome 18, and it would have been like B file. And, we could have done that. And we would have also had this for like, let me just put this back. It's too big. This would have been like, this would be the chromosome like 1, for instance, in 1. And then this would also be a B file, which is, bed file. But I'm usually using this one, the merged file. So, and I would have also had to like adjust the sample set and everything accordingly. So I'm just using that one file. And so that's why I'm doing this. And so this is the CSV. It's a comma separated file. You see it's CSV file here. And if I were to open it over here, the CSV, you can see that there's empty, it's all separated by commas here. So that's the sample sheet information. It's kind of telling where that will be. But when I will be uploading the information, when I'm running the code in PGSC calculator, when I'm running it on DNAnexus, I'm going to be telling it that my code, my genotype folders, is the Saniya tutorials merged genotypes. I'm going to be telling it that my code, my final genotype information is in merged genotypes. I'm telling that here, that this is where my genotype folder is. And I'm going to read in this genotype folder, essentially, when I'm uploading the files. When I'm uploading the bed file, the BIM file, and the FAM. And the prefix will be this file here, the all chromosomes SNP merged this. This is the prefix. I will be using the prefix locally in the command. I'm using the prefix, this prefix here that we have, this prefix, this one right here, this is the same as this all chromosome SNPs merged. This is the same prefix. This guy here is the genotype prefix. That's what this is here. I'm using this. Locally, I'm kind of based off of this information here, like subbing in for PGS catalog ID, putting that here, this PGS 00053 for genome build, putting that there. That's what I'm doing. And so that's what I'm doing. I'm using that local name here, but to tell it, and that's also what I'm doing in the sample sheet. It's the same prefix that I'm using here. And that sample sheet is what I'm also using when I'm telling it like where the sample sheet will be. But when I'm like telling the set of files, I have to upload them this way. I have to give the full genotype information so that the environment can load in that bed file, that BIM file, that FAM file. So that's where I'm telling it like where to go. It's like scavenger hunt. It's like running all over the place trying to find where these files, and then it's putting it in this environment for us to work with. So we're like telling it, okay, go in London, like go here, gather this for this treasure hunt, go there, gather that, gather this, and like put it all in this area. Like we're just guiding it, like where to find the files, then it loads it in, and then we can work with those loaded files locally in that environment, in that cloud. It's like we're taking something on an island to work with. We're going on I'm technically living on an island, but it's not really like island-like, I guess. But anyways, you can tell it which files to take. And then it works with those locally. So what you see over here is that's what's happening. And now that I have the sample sheet created, I want to also take it from my computer and move it up into the cloud, Mario style. So we're going to use the same dx upload command to do that. So let me just go down here. And this file is in, Saniya demo. It's the sample sheet, and that's this file here. Not this one. This one. B file. That it's a bed file, bed, BIM, FAM file. That's the format. It's a Plink1 format. Plink1. And we can have those 3 different possibilities as of now. So now what I'm going to do is I'm going to use the DX toolkit to upload this here. And I'm just going to be a bit lazy this time. And I'm just going to throw it in this main folder just for illustration's sake, but you can move it where you'd like. But it's going to the desktop Saniya demo sample sheet folder, PGS calc, this one here. This is what this file is called here. So yeah, here I'm going to be uploading this sample sheet CSV file from my local computer and server to the DNAnexus platform using DX Toolkit. Woo! So I'm going to be going from here, the sample sheet on my computer, the CSV file, all the way to the Saniya tutorials folder, where I'm just going to be lazy and put it in the main thing. You could put it into a subfolder called sample sheets, but I just put it in here. I got lazy. Anyways, you could just move it from here to here. And I'm showing you how we're doing that using the DX toolkit, DX upload command. So yes, let's see what this looks like with the sample sheet. Okay, and so yeah, I'm going to just be a bit lazy, and I'm just going to upload it here. I'm just going to put it in this generic folder, Saniya tutorials. You could create another folder if you'd like. So what I'm going to do is I'm going to put this command here, which will just essentially be going in, and it sees the sample sheet is here. The sample sheet is in Saniya demo sample sheet folder here. So get info, you can see the little preview, it's a CSV file, and I'm going to just be uploading it, this sample sheet, PGS calc, this ID here, GRCH37 sample sheet.csv, and I'm just going to be putting it in the Saniya tutorials folder, just lazily going to put it there in this DX upload, and then hit enter. And it's uploaded the file, and this is the property of the file. So here we go. I have it here. And this is what it looks like. It's a comma separated file. Again, this empty thing means that it's just for all the chromosomes. That's what it says. Like if you have all the chromosomes, no need to specify, leave it empty. And this is as of now. So we have it uploaded. Yay, that's how we upload the sample sheet. Woo, okay, awesome. Yes. So again, just please make sure you are logged into DX Toolkit on your terminal on your machine, and then I'm going to do DX upload, and this says where my sample sheet is located. So I'm moving my sample sheet from, users SK37 desktop Saniya demo sample sheet from this location to this destination. And it's a non-interactive job, it's brief. So I'm just uploading it. So that's how I upload the sample sheet CSV file from my local computer to DNAnexus platform. So we have these 3 inputs for PGS catalog calculator. And again, there are other input files, Ancestry, even though Ancestry is optional, but we can provide it to PGS calculator as needed. So you can provide that as needed. The Ancestry is optional, but the first is the sample sheet.csv, which is in Saniya tutorials, PGS calc, PGS 00053 underscore GRCH37 dot underscore sample sheet.csv. This is the sample sheet information that basically is saying, hey, where are the genotype files located? Or what are they called? So I'm mentioning here, this is what these genotype files are called. And later in the script, I'll show you how I specify where they're located, but I'm just kind of giving the title of those genotype files, the all chromosome SNPs merged the ID and then the GRCH37 part, I'm telling it, and this is where my genotype files have those prefixes. So if we had other files in this folder, the sample sheet is telling it to just focus on those files that have this prefix, this beginning part. That's what this is saying. So we have the sample sheet, and the sample sheet is kind of pointing to the names of the genotype files that we need to use. And then we have a score file, in this case for PGSC calculator. And sometimes you can specify them, or there's a flag that you can use for it to be like on PGS catalog calculator. You could just mention the IDs directly, but in my case, I'm just mentioning them, uploading the score files. It's all good. I just upload the score file. And this is the score file that we need. So really, the sample sheet and the score file. And of course, you need to have your genotype information. And there could be other inputs that you could use, like the ancestry information. But here, I'm not focusing on that. And yeah, this is my information for all chromosomes, which is why I leave this blank here for chrome. So this is the code file for running step 6, the PGSC calculator on DNAnexus platform. This is all the code here that we need. I'll be walking you through this. So, as a quick high-level summary, this bash script packages up a run of the PGS catalog calculator, PGSC calc, inside a DNAnexus job using the Swiss Army knife tool. The steps it performs. So it first defines user variables like the PGS catalog ID, the genome build, and various file locations. This link thing is just optional, but just for reference. So we define the user variables here, what those are. Where is the sample sheet path located? It's in Saniya tutorials, and this is what it's called. What is the genotype prefix? What is the folder that this is found in? So this is where I'm mentioning it, like that this, the all chromosomes SNPs merged files, those BED, BIM, and FAM files, they are located in this folder here. So I have a variable for that. And the output folder where I want the PGSC calc output to be is in Saniya tutorials in this folder here. Then the second, I'm checking whether an output for this PGS ID already exists on DNAnexus. If so, I skip it. So I'm creating an output folder if it does not already exist. So I'm creating that here. And then I'm running this check to see that inside this folder, I'm doing a grep. So I'm trying to locate it and see if anything related to this PGS catalog ID, if that exists. Then I can just skip it and say, hey, we've already computed something for this trait. No need to do it again. And this is just a defensive programming step. You may or may not need it. This, again, you can please adapt what I've done for your needs. But yeah, it's the same thing. If it exists, it'll skip it. And please make sure that you're robust with this check. Then the third is we're building out a shell command that will be executed inside the DNAnexus job. So it's building this nextflow command. The first part of this command is to install the nextflow bioinformatics pipeline. It's very essential for running PGSC calc at this point. So installing nextflow here. Then we're writing a params.yaml file for PGSC calc. And then we are running nextflow run PGS catalog PGSC calc profile docker. That's what we're running here in the last part of the command. So first installing nextflow, this part, creating a params.yaml file describing the score and genotype details, and then running the PGS calc workflow with the folder, with Docker. And then in this part, we are submitting a DNAnexus Swiss Army knife job that sends the needed input to the command cmd. And then we're printing a confirmation that the job was submitted. So in PGSC calc, they also mention here that we can set up parameters in a file using a YAML file. So parameters can be stored in YAML format. For example, input, PGS ID, liftover, target build here. And then when you're running Nextflow, you'll mention it as /params file here. So that's what I'm kind of going to be using when I'm running it here in DNAnexus, this idea of having it in a params file, some of the parameters. And if any of the parameters we mentioned here are not found in PGSC calc, it just skips it. It's not important. So now we have all the information that we need to run PGSC calculator. So the catalog calculator. Again, this is my example. I'm not going to go into the ancestry right now. I might do that in other videos. But I just want to show you this example right now where we have our score file for the 21 variants, in our case for late onset Alzheimer's disease, where we also have a sample sheet that is pointing to the genotypes that we have, which are just condensed for those 21 variants across the almost 500,000 participants in the UK Biobank. Their genotype data I have subsetted down to just the 21 variants across those 11 chromosomes. I've created those condensed BED, BIM, and FAM files. Those are Plink1 binary files, and I have created that. And I'm going to be using this information. So what they mention, this run ancestry, you don't need to use that right now. I don't use it in this case, but what I've been doing is I have been just using the score file here. We have got the score file of the 21 variants. I have the merged genotypes for those 21 variants. Again, we could have converted these original BGen files. We could have kept those into BED files. It would have just been a lot of memory. So we've subset those BGen files down to the 21 variants in a subset BGen, and then from that BGen we converted it to these Plink2 format files for the chromosomes, and then merged them together to have the FAM, BED, and BIM files. And this information we're pointing to in our sample sheet here. We're pointing to this information, this genotype information. So we have our genotype information pointing to it as the sample sheet. So we really are using the sample sheet, which points to this genotype information here, this FAM, BED, and BIM information, the genotype information, for each of the samples. What are the genotypes? How much of the effect allele do they have? And this is the weight we give to the effect allele. This is the column for the effect allele weights that we're using. That's saying how much are they contributing to the risk of late onset Alzheimer's disease. That's what this effect weight is doing here. And some are increasing the risk, they're positive, and others are negative. So they're really, really conducive to very, very protective of Alzheimer's disease because they have a negative effect weight, and you can see the odds ratio is less than 1. So that means that it tends to decrease your risk of Alzheimer's disease. So usually in our case here, if the effect weight for the effect allele here, if it's less than 0, then it's protective against Alzheimer's disease, in our case, AD. It's good for you. It's good for your cognitive levels. It helps stave off the effects of dementia. It's very good for you. And then the odds ratio, the OR, will tend to be less than 1 in that case if it's protective. But on the flip side, what happens if the effect weight is greater than 0, That means it contributes. It's a harmful SNP, contributes to increased risk of, and sorry, it should be late onset Alzheimer's disease, usually beginning after age 65. And then this corresponds to an odds ratio that's greater than 1. It means it's greater odds of you developing the condition. So we have these scores for these SNPs and how they contribute, and we have a genotype information, and we could also include ancestry and so on, but I'm keeping it simple here in this example. So this will be what we can use in our example to run the PGS catalog calculator. So those are the inputs that we're using. Again, just to clarify, it is the scoring file and a sample sheet that is pointing to the information on the genotypes, which is here. And then again, in the code, we will be uploading those folders and telling it exactly how to access those, like where they're found in the merged genotypes. We'll be telling that to it here. That's the job of this. And now the sample sheet, now you can see here is that I have mentioned that the sample sheet is just in Saniya tutorials, PGS Calc. I just left it in the generic part here. And that's what I mentioned in this code. That's the sample sheet path. And I've also uploaded that here, so it will know to access it. And I've also told it to upload the score folder. This is the score folder that I was mentioning here. This score folder is in the PGS scores part here. This score, I've told it that. And I have the score file, which is given as this id.txt again, where you have a dollar and you have these brackets. It's just a variable placeholder. So you put this, fills in over here. This guy right here fills into that area there. So that's what it is here. So we're telling it to upload the score file and the sample sheet, and then the sample sheet was referencing the genotype information, but these are the real folders where you can mention it. And in the sample sheet information, just keep it like local. Don't mention the whole like genotype folder. Just keep it local. And yeah, these were like some of the trials I had to figure out to see like what it needs to to run, and that's what it needs. So I'm going to be walking you through the different steps right now. So the inputs for the PGS catalog calculator. So what we see here in this part here is we have the PGS catalog ID, the polygenic score ID from the catalog, the genome build. Again, this is HT19, this is HT38. The input to the score files. This is the score folder. And this is the name on like DNAnexus, and then this is the score file name would just be this .txt. Or when we're running PGS calc, we could mention this flag directly of this instead, but I'm wanting to just put the score file in. For the sample sheet path, we have the Saniya tutorials, and this is where the sample sheet is located. For the genotype prefix, this is where the file, for all of the BED, BIM, and FAM files, they all have this as the beginning. And this is the genotype folder where that file is located. And this is optional, but I just want to remember it's a Flink file. And the output folder is where it'll be output to. The analysis, this is where we will be putting in the destination, the output folder. And then the genotype folder here, when we're reading in the files, the genotype folder will be telling us here that please read in the .bed, the .bim, and the .fam from the genotype prefix, which I'll explain. So yeah, just to mention again that these are the PGS catalog ID and the genome build. So these two variables identify the score you want to compute and the reference genome coordinate system we expect, and they're used to name output folders and pass to the pipeline in my case. Then the score file is the PGS, the polygenic score file, the weights and the variant identifiers, and the typical PGS score files are tab or CSV with columns like chromosome, position, RSID, effect, allele, and a weight column. The script expects this file inside the score folder and will pass it to the DNAnexus job. So it expects that the score file will be in this folder. And that's what I'll mention here. And this is the sample sheet path, will tell us where our sample sheet is located. That's what we need to. We'll later on be telling it the sample sheet path, because we will need it to upload that sample sheet for us to use. And then here, the genotype prefix is the file prefix for the Plink binary file. So the script expects .bed.bim.fam with that prefix and genotype folder, and the PGSC calc workflow is, is told like this genotype prefix is what it should look for. And I've just put the Plink just, for my own reference. You don't need it. And here, this is the output folder, the DNAnexus destination folder where results will be stored, and later the script creates it with dx make dir /p. And so this step here is like a defensive programming step. It's like defensive driving. So you skip it if it's already done. So the dx mkdir ensures that the DNAnexus folder exists. It just creates it otherwise. And then this part, the dx/ls output folder, lists entries in that folder. And the this grep part here, so this is listing all the entries, and then this grep part over here, it looks for a subfolder starting with the PGS ID. So it's like regular expression matching. That's what grep is doing. Get a grip on grep. If that entry exists, the script exits early to avoid rerunning the same job. So it's just basically checking, does this file exist in DNAnexus folder? So it's checking in this PGS, this output folder that we've created, this PGSC calc output by PGS ID, this folder here. It's checking, does this file exist in the DNAnexus folder? Does it exist for this one here? If yes, please skip. That means we already have run it. We don't need to run it again. If no, then we will run the rest of the code below. That's what it's doing. It's checking if that PGS ID exists and the subfolder starts or has that in some form, the ID. So this part here is building the in-job command that we're going to be running. This CMD, which is what's going to be run here by dx_run. This is the real meat of everything. So the first part, the CMD, is going to be downloading and installing Nextflow on the DNAnexus platform here. So PGSC Calc has a few important software dependencies, Nextflow, Docker Singularity or Anaconda, Linux or Mac OS, and so on. But we need to see that this is how we install Nextflow. But here, this is, based on the documentation there, but this is how we will install it for the DNAnexus part here. So this first line, this curl /fssil, this part over here, bash, this first line, downloads the Nextflow launcher script. This places a Nextflow executable in the current working directory. Note that Nextflow requires Java. You see here requires Java JRE 8+. If the DNAnexus runtime container lacks Java, Nextflow will error. On many systems, Nextflow will run, but check Java availability. That's what this line is doing. Then this line here is basically making a directory, and it's moving the Nextflow binary. What this is doing is it's moving the Nextflow binary into this location here, this tilde/.local/bin, and it adds that to the path so Nextflow can be executed. The /path here is escaped because CMD is a double-quoted string assembled by the outer shell. However, when the command runs inside DNAnexus, path will contain the inserted directory. So Nextflow, as I was mentioning earlier, it's a scientific workflow that system that is mainly used for bioinformatics data analysis, and it establishes standards for programmatically creating a set of dependent, step-by-step sequential computational steps, and it helps execute them on various local and cloud resources. So yeah, this part is just downloading and installing Nextflow on the DNAnexus platform. Then what you'll see here is we are creating the params.yaml file here. So this cat EOT part here, everything that's in between it, so will be part of the YAML file. Everything that is starting from here, all of these will go into the YAML file, as you see. And this script, cat, is a command that processes the document here. The the EOT command initiates the here document, and the final EOT indicates its end. We redirect all input between this to the params.yaml file. So it creates a params.yaml file that the PGSC calc workflow will use. So note that because the CMD string is assembled locally before submission, the score file here, the genotype format here, the genotype prefix, these will all be expanded at submission time, and their values are embedded in the here document. So that is usually fine, but be aware that the params YAML file contains a little text created by the submitter shell, not by the remote job. So basically what happens is that in the params YAML, when it's being created, it's going to create this file here, the score file. It's going to be the score file over here, which is this PGS catalog ID.txt. That will be populated here. The genotype format will be plink, which is coming from here. The variant ID column, RSID, which are hard-coded, effect weight column, effect weight. These three are just optional. We don't need them, and I just want to show you that if you do put extra things in the params.yaml file, if they're not needed, it's not going to do anything bad. They're just not going to be needed here based on what we're using. And again, you can include other parameters like ancestry and whatnot. I'm just kind of showing you a very simplified version here. And then the genotype path here will be coming from genotype prefix, which is this one here. And when it's running, you can see how it's automatically populated the PGS catalog ID and the genome build, how those already come in here. So this will be what is created here, this params.yaml file, and it's going to be saved in the output folder, the PGSC calc output by PGS ID. It's going to be saved there, the params YAML file. That's what you see here. So the params YAML file here contains a score file, just a file name, PGS 000053.txt. That file is also passed as an input to the DNAnexus job, as you see in the input below. The score file, the genotype format, plink to tell the workflow. The genotype files are plink binaries. The variant column, effect weight column. The genotype format, variant column, and effect weight columns, those are like not really needed here. But the genotype path is just the plink prefix name, no extension, and the pipeline will look for bed, bim, and fam with this prefix in the working directory. So yeah, the params YAML file describes the score file and a few extra parameters for our reference, and just the genotype data location, the genotype path for PGS catalog calculator to compute the scores. And yeah, we just really need the score file and the genotype path. So this is actually a simpler one where I've removed the other information. So we just need the score file and the genotype path, and this is the params YAML. If we simplify that, the others are just notes. I'm just showing you that you can have like more like parameters here, but you just need like the score file and the genotype path, and you can see the simpler params.yaml file here, and these will be passed in as parameters when we do run it. This params YAML will be passed in here. So yeah, you may use this simpler file to run PGSC calculator. Just need the score file, genotype path. And additional parameters can be specified as needed. In fact, guys, I'm going to show that there are a lot of different parameters that you can use for PGSC calculator. So you can see in the documentation here that they mention all sorts of advanced parameters. Let me show you. So you see here how on the PGSC calculator site, they mention workflow parameters and that how you can set parameters in two ways using a params file option, which is what we do. And then you can kind of run it, how to set parameters in a file for more detail. So they provide some documentation here that if you are running this with a YAML file, like what we're doing, all you have to do is mention /params file, my example params.yaml. So that's what you would do. And then this would be like what a example YAML file would be, how you could store it. So you can see that these are the different parameters, and then you can feed them in here and it will run them for these parameters. And if there are parameters that you use here that are not part of the PGSC calc emulator, it will do nothing about that. It will not bother about those parameters. So if I put some extra parameters there, it's not going to worry about those. And I put those for my notes as well. So it's not going to worry about those. And in fact, if you want to see the list of parameters that will be that can work in PGSC calculator, if we scroll down, it gives you input and output options and parameters here that you can go through here. And so on. It even has ancestry, but again, the ancestry, it is optional. You don't need it. You don't need to use the ancestry necessarily. If you did, it would help break down your scores based on ancestry. So you see here, there's some different parts that it's explained here. And in fact, like if this is optional, I don't show it in this video, but you can kind of figure out the polygenic score distributions and genetic ancestries across different groups, European, East Asian, African, and so on. But it's optional here. But then what you see here is if we go up over here, they have also given us a JSON file where we can, see the entire list. You can either run this in a command line or you can download the schema here. And the schema will kind of, you can make it pretty printed and it will tell you all the different parameters that you have, like what's the score file, what's the description to it. It'll tell you all of the different parameters that are supported. And if you specify something in the params file that's not like a default in the PGSC calculator, it won't do anything with it. It'll just ignore it. So that's just how you can specify these additional parameters using the PGSC calculator function. In fact, in the documentation for PGSC calculator, it will basically say that it matches between the score file and the genotypes. It'll be matching by chromosome positions and alleles. So variant matching is critical to reproduce scores deposited in the PGS catalog, and it's really hard to match a variant described in a scoring file against genotypes. So variant identifiers usually SNP. They're not usually stable or unique identifiers. In my case, I made sure that they mapped, but sometimes the genomic coordinates can change across different genome builds, and matching effect alleles can be technically challenging because of allele flips and strand mismatches. So I did verify beforehand that our SNPs in the BGen and the SNPs in the score file, that they all are found. But just be careful. And the PGS catalog scoring file, standard and genome build harmonized data uploads and simplifies this process. So what they do is they kind of look at ref and alt combinations. So in one file, if what is called a reference allele is called an alternate allele, it kind of does this flip flopping. It also kind of does all of this strand ambiguous matches, multi-allelic variants. It does all of this processing here. And then to basically align the set of variants from the score file and the BGen file. And in our case, we did our due diligence by making sure that all 21 of the variants that are in the score file are also those that are found in the subset BGen files. We've made sure about that. That's great. So that's really awesome. So we need to compare the best match candidates to the original scoring file. And what PGSC calc does is that if 75%, at least of 75% of the variants in the score file, if they are found in the genotype data, so if we have genotype data for at least 75% of the variants in the score file, which are contributing to the polygenic score. If we have genotype data for at least 75% of those variants, it'll go ahead and compute a score. That's what the min overlap is saying. It's currently at 75%. Otherwise, if we have, for instance, like just half the variants, just 50% of the variants in the score file have genotype information, we will not compute anything. So like if I have just 21 variants, if I only have data for like 10 of them, or 11 of them, so less than 75% of them, then it'll skip. It won't compute anything. It'll say, oh, we're missing data for too many variants. We're missing that genotype data. So this is sort of like how it's working behind the scenes. So matching logic. So this is again this score file for predicting the genetic risk of late onset Alzheimer's disease. Here, as you see here, these 21 variants, and the quality and completeness of our polygenic score will depend entirely on how well the position and alleles align between our score file and our genotype file here. So we have the score file, and then we have the BIM file for these 21 variants. You see they line up here. Again, the CMS centimorgans, the A1 and A2, they are stored as alleles in our genotypes here. So this is the effect allele, and this is the non-effect allele here. So what's really important is to first make sure that the score file, the chromosome name and the positions. So just making sure that, CHR1, just making sure that the positions here, that they are going to be able to be aligned. That is, that these are in the same reference build. And I'm making sure that in this case, that this is in HG19, GRCH37. So that this is also in HG19 GRCH37, and that's what I was checking in the beginning when I was looking at the genotype we're using. That's also why I used the UKB imputation from genotype data on the research analysis platform on DNAnexus. That's why I used it, because I knew that that would be in HG19, so they would align. Otherwise, if I was using TopMed, for instance, that would be HG38, and then this is HG19 and HG38, it would not map, and if you lift over and it gets kind of complicated. So whatever format that is, just making sure that they are aligned first. That's the main thing to worry about for PGSC calc. And in the score file, the CHR name and the position are going to tell PGSC calc where to look for in the BIM. And the effect allele is going to tell it which allele's dosage to multiply by the effect weight. So it's going to look here, and then it's going to say, okay, this is the reference, this is the alternate here. This is the reference first and alternate. And so it's going to tell it which is the allele by which we'll multiply that weight by. That's what it's going to do to calculate the PGS score. The RSID is present in both, but PGSE Calc does not rely on RSID mapping and matching. It's used only for reporting. So PGSE Calc will use chromosomes, positions, and alleles for matching, and it will handle the strand flips and the ambiguous variants. So yes, that matching by PGSE Calc will be done by chromosome, position, and alleles, and the reference SNP, based on the way it's programmed, it's not going to be used for the variant mapping. But I was using it to subset the BGen files, and I ensured that all the 21 variants that are needed in the score file, they are found in the BIM file. And that means that they're found in the genotype file. So here we have 100% matching rate, which is great. Sometimes we might not be so lucky. But basically, in our Alzheimer's example, all 21 of these SNPs in the scoring file will match directly to our BIM file because the chromosome position and the alleles, they line up perfectly. And usually in score files, we can also have like the non-effect allele present. So for example, for this first SNP, you see that it maps, right? This RS 6656401. So we find the variant in the BIM file using the chromosome name here, chromosome 1. Then we look at the position, this position here, which is this guy over here. So in the score file and the BIM file, they match. So then we compare the allele. So the score file has the effect allele of A, and the BIM file will have an A and a G here. So the A is present, so it matches here. That's awesome. Then for the strand handling, if alleles are flipped, like the score file says AG and the BIM has TC, for instance, like if there was another column here, then the PGSE Calc detects and flips. So some score files could also have like effect allele and non-effect allele. So just to keep that in mind. Then PGSE Calc will detect and flip. It's going to take care of that. That's what you need to know. It's just going to focus on the chromosome name and the position and the alleles, and it will take care of that. Unless you have this flag, keep ambiguous, which is one of those parameters we could set for PGSE Calc, which is also some things you can play around with. PGSE Calc will exclude them. And so assigned weight of the genotype dosage of A is like 1.2 for an individual, so meaning 1.2 copies of allele A on average, and the contribution will be 1.2 times this effect weight. That's what the contribution to the polygenic score for late onset Alzheimer's disease will be. And then for each individual, we just are calculating the polygenic score, which is just taking the effect weight here, multiplying by the allele dosage for the individual across all the matching variants. And currently it's set to see that like for all these variants here, at least 75% should be found in the BIM file. That's what the min overlaps parameter in PGSE Calc is checking. So here, all 100% are mapped, so that's great. So there's no problem there. All 100% of the variants needed for the score are found here. Otherwise, if like less than 75% are found in this one, like we have some missing ones, it'll say that, oh gosh, the score, we don't have enough genotype data to compute the score, I'm sorry. So basically for each individual, the polygenic risk score of developing late onset Alzheimer's disease is just this effect weight times the allele dosage across all matching variants. So for our example, with 21 variants, each person's score is a weighted sum across these 21 SNPs. So when we run the PGS catalog calculator, we want to, we're going to be using Docker here. That's what we're using. And based on the commands, it's going to be like nextflow run PGSE Calc here, profile Docker. We have the target build here And you can directly put the PGS ID here, but I don't use that here. I'm kind of just using my own score file that I've mentioned here in the YAML file. But basically, when we're running it, this is sort of like the command that we're using. I'm mentioning the score file here. I'm mentioning the sample sheet path here. I have the params file, params.yaml, and I have the target build here. And then I have my output directory that I've mentioned over here. So I have pgsc calc results, and then I have this folder here, which will have the output results. So this is like what you need for running. So the Nextflow run pgs catalog pgsc calc profile docker. This first line, this runs the published pgsc calc Nextflow workflow from the pgs catalog GitHub repo. Nextflow pulls it and flags. The profile flag being Docker tells Nextflow to run tasks inside Docker containers. And a caveat is on DNAnexus, Docker, and Docker. So a native Docker access might not work in the same way as on a local machine. Many cloud execution environments use their own container orchestration. So we need to check whether the profile Docker is supported or whether pgsc calc provides other profiles like AWS, Slim, or generic profile. If Docker is not available, the run will fail. Then we do the input base name, sample sheet path. It passes the sample sheet file name, only the base name as the / input argument to the workflow, because the submitter included the sample sheet as, you know, this II in here. To the DX run, the job working directory will contain a file with the same base name. Then we have this score file here. It's again the file name of the PGS weights file here. And then we have the target build here. And then we have the params file, the params.yaml. So the target build is the genome build argument, which is grch37. And then we have the params file, params.yaml, uses the params.yaml we wrote. I know that there's some redundancies here. I just wanted to show you how we can kind of have a params YAML, or we can have the input sort of separately based on what is needed. And the out directory, pgsc calc results, this one here, it's going to be where the workflow will emit and save its results inside the job workspace. The DNAnexus job will then upload the files to the destination that is set in the dx run command. That's where this directory, out directory, will that folder of its results will be saved in and stored in this output folder here, which is the Saniya tutorials pgsc calc output by pgs id. So I'm just kind of also showing you that we can use the params file, params.yaml, and we could also just, you know, mention some of the parameters in these ways as well. So now we're going to be running the PGS catalog calculator. So we're going to be uploading these files into the environment. So now I'm going through this DX run part over here. And I want you to see that we are going to be uploading these 4 or 5 different files. The first one is a score file, and if you have ancestry and other information, you might need to upload more files as needed. But this first one is the score folder, which is here, and then the score file. That's the first one here. The second one is going to be the sample sheet information. So it's telling it, like, where are the files, and then please bring those into the environment for us to work with. That's the sample sheet one. And then each of these genotype folders and the genotype prefix, you see how it's all the same, just the bed, BIM, and FAM are the ones that differ. So it's telling it where those are, because you'll see in the sample sheet, we only gave the all chromosomes. We just had given this as the information. That's all we had given. And now this is sort of telling it that once we upload these files into the environment, it will know that this is the bed, bim, and fam. These will be the names of the files when they are loaded in the environment. It's just kind of telling it where to go and fetch it. It's like on a grocery list. If you need to go to one grocery store to fetch things, another, like for shopping, you just kind of need to know where to go. And then you can get those ingredients that you need from there. So that's what it's kind of doing. So when this is uploaded in the environment, it's really just going to be called this pgs00053.txt, and when this is uploaded in the environment, it's just going to really be called this, and so on. So yeah, like when we're running this dx command, it runs a DNAnexus Swiss Army knife, the app, it will execute a command. It's good for ad hoc scripts. These IIN, they will attach these input files to the job, and these files will be made available in the job working directory with their base names, as I was mentioning. So these stems will be made available. All the beginning file paths will be crossed out. So basically we passed in the PGS weights here, the score folder, a score file, the sample sheet, the 3 pling files, the bed, bim, fam for the genotype prefix. And then the CMD will be running the command string that will be executed inside the job. This is the multi-line CMD that we built earlier here, that we mentioned. The tag is a job tag to help identify the run. Priority high, the DNAnexus scheduling priority. The instance type that we're using, what machine. It selects the compute, memory, input, output. It impacts the runtime and cost. And the destination, the output folder, where to save the job outputs in the DNAnexus project space. Brief is print a short job ID rather than verbose output. And yes, it's non-interactive confirmation, so don't prompt. Just run it, please. So too long, didn't read what the script does. So it prepares inputs and parameters for a PGS score calculation. It ensures that you don't rerun the same PGS if outputs already exist. It builds a command that installs Nextflow, writes params.yaml, and runs the PGSC calc Nextflow workflow. It submits all required files to DNAnexus's Swiss Army knife app to execute the pipeline on the cloud, and it saves results to your project folder. So I showed you how you can run the commands, using a params.yaml file if you have other parameters that you need to run, or how you can put them in file as well to run the commands as needed. And I know it's not perfect and you may have some questions, so please do reach out to me with any and all questions that you have. But it's just an idea for how you can work with PGSC calculator and get the results that you need to work. And you can adapt the params.yaml. You can put things in and out, as flags or in the params. I've shown you how you can create one from scratch and then upload it and put the parameters there. If you've got extra parameters, no problem at all. Just basically how you can get around with it, with working with this information. And it's kind of cool. So here we're running PGS catalog calculator, and you can see that these are some of the extra flags that I had. They're not used, they're just for my reference information, and they're not necessary, but I just want you to see that the input files, these are the input files that are being read in the PGS C calculator. This is what's being read in here. And I just show a simpler example as well, where we don't have those parameters in. You have the input files, and you can also see this is what the outputs look like. These are all the files here that we are getting for this run. There are quite a lot of them. And you can see that we build out the params.yaml file. And now for the moment you've all been waiting for, we're going to be running PGSC catalog calculator. We're going to see how it works on our data for the late onset Alzheimer's disease for the UK Biobank. So let's work with that little guy. Let's see how it works. We're going to run it and see. Okay, awesome. So now I've shown you the code over here. See, as you see over here, this is the code for step 6. It's on your demo. You can see. Just zoom in a bit. It's kind of walking you through this. And it's going to see if in the output folder that we have, if it already exists. So right now, it's going to see if in the PGS calc output by PGS ID, if that file exists, then it will not run it. It'll say, oh, you've already run it. No need to do it right now. You already have a predicted output. That's what this is saying for that polygenic score. If it finds it for this PGS catalog ID, this one. And so I've designed it again that you can just sort of take it and use your score files, and with minimal adjustments, you can kind of run this for your examples, especially very easily if it's HG37, if we're, for the score files. And for others, you just have to make a little bit of adjustments if it's in like HG38, for instance. But anyways, it's going to see if that file is existing or not. And we just mentioned it's in Plink. Then this is going to be printed out. And then the command will be in 3 formats. As you see here, the first part is going to be installing Nextflow. This part over here will be creating a params.yaml file describing the score and genotype details. And this last part will be running PGSC calc workflow with Docker over here. We mentioned Docker with the sample sheet with the score file. As I was mentioning, those two little outputs, the sample sheet here and the score file. That's what it will be running it with. And then we'll tell it the genome build, which is the GRCH37. It's very important. And it'll be taking this params.yaml file that it's created here, and it's going to be inputting them over here. And then this out directory will be basically storing the results. An out directory will be stored right in the destination, which is the output folder. And the output folder is called the PGSC calc output by PGS ID. So it's going to be putting this output, the output will be called this, and it's going to be saved in the output folder, which will be this one over here. So again, you always have to kind of ground it and tell it like all the information. They do the homework over here. Like where is it in the whole universe? In the whole universe, where is it? Like I'm grounding you here by saying that like, hello, we're in London. I'm telling you that. I'm giving you like the location. Like you're working with UK Biobank data. And so to give you that perspective, I'm sitting here in London. So that same thing you have to do when you're running this. You have to kind of give it the perspective and tell it where those files are located, which is what I'm doing over here. And telling it where to output it and what is the instance to use, like what kind of machine. And so then we're going to run this. And you'll see here, let me just click enter, 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 just for the sake of the new thing. And let's do ls and then bash. Step 6. We're going to run this guy over here and click enter. Running PGSC calculator for this here. Job submitted for this one. And then if you want to, I can see that we have this job here. That's what the job is. And I can do DX watch job. You see, I can do this here. I can, oh gosh, I can do this DX watch job. So it's going to allow me to watch this job. And now I can also go here to the monitor, any launch time past hour. And I'm going to click here and I'm going to see again that I've called it PGS calc. This is the ID. And if you look here in the code, that's what this tag was saying. So just if we end up like making this parallel for many different traits, you can track which one is being done. So it's waiting, it hasn't yet begun. And that's sort of like what this command is also saying. So you can have this score here. You can have a job. And then you can also do DX watch job, and it will also do the same thing. Like right now, it's waiting. It's watching this job. Nothing has happened right now. So you can also track what's happening here. But in the meantime, I'm going to just show you that this is what the command looks like. So it has given these input files here. It has told it, in this order here, it's told that the first one is a score file. This is here. This guy here. Oh, this one is here. Oh, wait, sorry, I meant this one is a score file. This one is the sample sheet. And these three ones here are respectively from these three over here. And now we can see that in the meantime, this has started running. So let's see if this tells us anything. I think it's still watching. Still watching it right now. But what I want you to see is it's uploaded these files here. It has gotten them because we've given those absolute paths for them. And now this will be what it's kind of doing. It's installing the next flow to work with it. It's writing the YAMLs file and so on. And you can view the log so far. It's downloading this. So nothing much is happening. It's downloading QC tool, Play-Doh, bed tool, HTSlib, Java, Plink. So just downloading it. And it's priority because I set priority high. It's still running it. And you can see that over here, when we're watching the job, the same thing is popping up over here. This is the job that we're watching. And all we have to do is do DX watch job for the same job that had launched. This is the job that I had launched when I did the bash PGS, step 6 PGSC calculator. And then I just did DX watch to view it here. So you can see that either I can look at it here or I can look at the log over here. Either or. So right now it says daemon started successfully. That right now it's written out the YAML file and it's running this. And so right now it's downloading Nextflow dependencies. It may require a few seconds. Please wait. So this was the part I was mentioning here when it is actively downloading Nextflow. That's what's happening here in this Nextflow. So this part is the Nextflow download part. So that's what's happening here. So it's downloading this version here when it's created. So Nextflow installation completed. Awesome. So that's what it's done here. And it's kind of moving it to the certain directory, making the directory to move it, moving Nextflow here. That's what these commands are doing here. So it's making this directory. So this one was downloading Nextflow, as you see here. This one had this curl one had downloaded Nextflow. That's what this one had done here. This make directory created this directory. Then this move Nextflow was moving it to that folder here. That's what this next command was doing. And then this export path here was done. So that's what's happening. Now it's running it here. For all of this here. And you can also see that in the terminal, it's finished. It's actually output all these different files, and each of these files has this like little ID here. So I'm sure if I just scroll down really quickly, here it's uploading the file. It has a params YAML file. It's also going to show us what that was. And it's had all of this stuff here. It's uploading all these final files here. In fact, yay! It worked. We were able to run PGS catalog calculator here on the DNAnexus platform for UK Biobank data. Yes, awesome. Yay, awesome. Awesome. Cool, awesome. So we're able to get it to work. And we see that these are the files here. We have the params.yaml. We've got a lot of things here, a lot of files, but it works. We have the aggregated scores.txt.gz, but we have this params YAML. So this is the file that we had created essentially when we were running this part here, the second part. This was the internal file, and then this was fed in here when we were running this part. It created this with the score file, which is what this is called, the score file, because remember, we mentioned this is what the score file is. Then we gave it the genotype format above, which is plink. We told it it's a plink file, and it'll figure out it's a plink one. Bed, bim, bam, bed, bim, fam files. The genome build, it is the also was separate, but the genotype format, the RSID is what the variant column is called here in the score file. Effect weight column is just called effect weight. And this is the genotype path for all the chromosomes, because you can see that in the score file, let me show you here in our score file. What we have is we have the variant is given here by the RSID as a variant column. Then the effect allele has the effect weight. That's where we get the weight from. It's called this effect weight column. That's what we're using. And so that's what the params YAML file is. And then we also have all of these different output files that will be fun to look at. And then what I'm interested in is the aggregated scores.txt.gz file. So this is the aggregated scores. So where all of this has been saved is actually in the Saniya tutorials. When we have the PGS calc output by PGS ID, we have work, we have these results. This is the params YAML file for this case. And then we go in here, and then we have the UK Biobank, we have the score, and we have this file over here. And the other files you can investigate as well. So I'm going to download this one here. And then I'm going to extract it. So ultimately, what I get here is this is the aggregated scores. I mean, I've downloaded quite a few scores, so you just have to be careful. It'll tell this is this sample sheet. This is the PGS. This is the polygenic score. These are the sample informations. And they will correspond to the FAM file. Average is just you take the sum and you divide by the denominator. Actually, let me just copy it and show you what happens. And let me just paste this here. But what you'll see is that these are for the samples that I have. And I think if you remember, I had 487,409 samples that I was showing in my sample file at the time when it said that these are how many samples that we have. Actually, let me show you that. I like to kind of connect things together so that they make sense. So like in this merged file, I think I had this log. And when I was doing this at the time, it says that these are how many people we have. So this is what you will see, 487,409. And this is my number. Yours will probably be different, maybe, I think. So these are the same number of individuals. And so for each of the individuals, we have calculated their polygenic risk score of having Alzheimer's, late onset Alzheimer's disease. And this is the score over here. We had 21 variants here. We had 21 variants in that score file. So this number is 21 times 2 because we could have like two different versions of the alleles. You know, you can have one allele or the other allele there. You can have like an A, you can have a G, for instance, right? And then this is the average, which is just basically comes from taking the sum and then dividing it by the denominator. That's what this is here. But yeah, these are the scores for these individuals. So that's how you run PGS catalog calculator. And I'm going to show you a bit more about like what these different files look like that you also will get. But as of now, what I get here is for this file here, I will have the results. You can have the pipeline info. I'll have all these different things over here that you can look into. And I will also have this, which is where I went. The match folder would look like this. And the score folder, which is where I got the aggregated scores. So yeah, that works. That's how we run a PGSC calculator on the DNAnexus platform for the UK Biobank data. Yay. So yeah, you don't need to build the params.yaml file. We can upload it as well, like the way that we've uploaded the sample sheet to the DNAnexus platform. We can do the same thing for the params.yaml file. We could create it and then upload it. But I'm just wanting to show you that you can also do it programmatically. And in this case, it's just one less file to upload it. You can put all the settings in. And then if you'd ever want to just keep on doing this for different scores or traits, there are many ways to do this. I just wanted to just show you how it can be done, and you can use this as a template for your own needs. And do reach out if you have any questions or suggestions or comments, but this is just one way to do it. And this is how I got it to work, so you can kind of adapt this as needed for your needs, hopefully. So yeah, you'll see here that we've just finished running the PGS catalog calculator here with those flags that are optional. These are the parameters that we have. And if we just are running it on the simpler without those flags, we still get our results, We get the params YAML and so on. It's a little simpler. So little guy. So I'm just going to show you how you may check and see some of the parameters that could be helpful for PGSC calculator for the polygenic score catalog calculator. Just more variance information, score matching, and other things that you may wonder, just where you can find them. So the paper for the tool was published last year, and I know they're coming out with a new tool soon. So you can see here, enhancing the polygenic score catalog with tools for score calculation and ancestry normalization here. So if we go down here to the supplementary information and we click this here, it'll take us over here to the supplementary information. So they talk about how they create harmonized scoring files to facilitate easier PGS calculation, development and implementation of PGS catalog calculator in Nextflow and Python. So the Saniya was a synthetic European cohort. It's just synthetic. data, uses Plink2 internally. So they mentioned the methods that they're using here. So then they talk about all of this stuff here. They talk about variant matching, how they do that. Making sure that we can essentially have the variants that are needed for computing the polygenic score. Do we have a genotype information for a majority of those variants for a given score file? That's what we want to check. That we have to compute the polygenic score. The polygenic score is made up of some number of variants, and do we have enough information for those variants? Do we have enough enough genotype information? Like if we have 100 variants that are needed for computing a polygenic score, diabetes, for instance, like do we have genotype information for at least 75 of those 100 variants? Do we have that information? That's what we want to see. That's what's key here. And if not, I mean, that's what the min overlap is testing here. So they talk about how they do the matching and mapping here. If the overlap for a score is below 75%, drop the score. But you can change this, but they don't really recommend it as much. See, a default minimum threshold is set at least 75% of variants in a scoring file must be successfully matched to genotype sites in the target file before calculation can proceed to reduce the chance of polygenic score misapplication. That means that, maybe, what if we're computing the polygenic score for a trait and with diabetes hypothetically has 100 variants and we just have genotype information for only 20? Then the score might not really be so accurate, right? Because we're missing data, genotype data for 80% of the variants that are needed for the score. So, but they do recognize that sometimes those polygenic scores, like the variants that are involved, they might not be always available in genotype data. Maybe you can use like linkage disequilibrium analysis or other tools to kind of try to impute things or find the correlated SNPs and so on. That could be done. But one thing is that they want to make sure that for a majority of the SNPs in the score file, we do have the matching genotype information for them. That's what's really important. And then they kind of explain, which I explained earlier, how they compute the polygenic scores here. Ancestry adjustment, the workflows, if we need this. So yeah, that just kind of like where you can find additional information on it. And of course, they do have their like website here that you can check where they're going over PGS Calc, the tool. Now, little guy, let's go through some of the final outputs from PGSC calculator. So the pipeline outputs are written to a results directory, whatever we specify in outer. If nothing, it just puts the results with the following structure. So we have results, sample set, the directory with results for your data. Within that, like whatever we call a sample set, it'll create a subfolder for that. Then we have match, scoring files, and variant match metadata, and then a score, which is calculated PGS with summary report, and then pipeline info, which is the next flow pipeline execution data. So in our case, I called sample set UK Biobank. So what we will get is we will get a folder that's called UK Biobank, and then we'll have another one called Pipeline Info. So, you know, in the project Saniya tutorials, PGSC calc output by PGS ID, I have gone into the folder PGS C calc results, you know, PGS 00000053, and then I find these two here. And again, this is what the sample set was called in the sample sheet. It was UK Biobank, so that's why we get this name here. So then we click the UK Biobank folder here. That's what the sample set was. Here we click it, and then we have these two folders, a score and a match folder. In the match, the directory will have information about the matching of the scoring file variants to the genotyping data, you know, in the sample set mentioned there. And it'll be, the first would be a summary file that's also displayed in the report, you know, details about whether each scoring file passes the minimum variant matching threshold, which min overlaps here is 75% by default, and the types of variants that were included in the score. So that's what you see here. So this would be the sample set summary.csv metadata. It has all these different columns here that you can see, and the past matching will be either true or false to indicate whether the scoring file passes the min overlap threshold and is included in the final scoring file. So by default, this value is 75%. If you, unless you specify otherwise, it is 75%. And then ambiguous match type, match status, the data set accession, just all of these things here that we can check out. And this is on the PGSC calc website. And what you see over here is we have like the data set. I've just kind of organized the summary information here. It says score pass. Yes, we passed the minimum overlap requirement. You know, 100% of the variants in the score file were found. We have genotype data for it, but again, this threshold by default is 75%, and you can adjust it in min overlap. Just be careful. But that's what this is just telling you, you know, that it matched, and you can use the scores. So for example, within the match one, we have this log file, and this log file is a CSV that contains all possible matches for each variant in the combined input scoring file. This information is useful to debug a score that is causing problems. So it contains column information about how each variant was matched against a target genome. So here we're focused on, this is the sample set, you know, UK Biobank, and then log.csv.gz, you know, for the score. And then what you can see here is all of these different columns here. So chromosome name, position, you know which one are these are matched. So in the scoring files, we have all the variants, and we're seeing if they're matched or not. And then you can kind of check for the missing ones, if they're not matched, why aren't they matched? So here I'm kind of lucky. I kind of have them all matched. So this is kind of a confirmation that all of the variants in the scoring file for the late onset Alzheimer's disease, all of them were matched. Yay! But in those cases when it's not matched, you can use this file. So I downloaded this file, and then I did this command called gunzip, G-U-N-Z-I-P, and then this thing here. So I downloaded it. But also be careful for the UK Biobank DNAnexus portal. Make sure that you are complying with the standards for downloading the data. So here these are just score informations, and they're not identifiable to any patient. So hopefully that's safe to use. So and then this is the summary CSV file that gives the metrics on the variant matching for the score calculation that was mentioned. And then the other folder was the score folder. So we just finished looking at the match information, and now this is the score. So the match was just saying how well are they aligned between the score information and the genotype. Like, did we find all the variants we needed to compute the score? It's just more like quality control checking for us. So now, before we dive into the scores, we just needed to do our due diligence there. So now the score here, and I'm going to focus on the aggregated scores .txt.gz. It's a zipped file. And here, again, you can look at the explanation, the output file here, but the scores, calculated scores, are stored in gzip text space delimited text file called this. And the data is presented in long form, where for each polygenic score for an individual is presented on a separate row. So if you're doing multiple scores, just be careful. It'll be on multiple rows. But anyways, what we will see for one score is the sample set, the name of the input sample set or reference for the panel. Mine will be UK Biobank. FID, family identifier of each sample, may be the same as IID here, which is the case for us. The accession ID of the PGS being reported, ours is like PGS 000053. The sum, this is what we will be using. The sum will be giving the weighted sum of effect allele dosages multiplied by the effect weight for each matched variant in the scoring file for the PGS. So if we run the pipeline without using ancestry information, the following columns will be present, which is the case here, denom and average. But I will be using the sum information here. We use sum. That's what we need to use. So that's how the score file looks. So you can see here that for Alzheimer's disease risk, this is the score file that we finally get, and we're going to be using the sum information. Oh, what am I doing? This information here, and you'll notice that this number is 42. It's 42 because we had 21 variants for our scoring file, and 21 times 2, so 2 possibilities, you know, for each. So 21 times 2 is 42, and this will differ based on the scoring file and the trait that you're using. So yeah, the polygenic scores, the PGSs, they are developed to measure an individual's genetic predisposition to a disease or trait, like how likely they are to have the given disease or trait. And in our case, we're trying to figure out the susceptibility to late onset Alzheimer's disease, you know, using that score file. And there'll be different studies that have their sets of variants, and hopefully as more information and more individuals are genotyped and we get more of the information, we're able to better we're able to have more enhanced genotype data and score files. So the models, the polygenic risk scores that come out will be even better. That's where all this big data is being very useful, because we can have improved genotype data, more individuals, and we will also have better score files with better predicted effect sizes for individuals. So the output machine learning models that are predicting the effect sizes and so on, they'll be much better. And then ultimately our computation of the polygenic risk scores will only improve with data. So please do contribute. I'm a participant for the All of Us cohort, which is neat, for the genotype data back in the US. So anyways, we can express individual level polygenic scores as a z-score or a percentile here. And we have the standard normal z-scores that are between 0, is centered at 0 with the mean, and go from -3 is like one extreme to another extreme here. You know, this is 3 standard deviations of the mean. But yeah, this is sort of like how the data can look. Yeah, and we can express the individual polygenic scores as a z-score or a percentile, and work with that. So right now we're going to work with the sum information that is output by PGSC calc. But basically, yeah, this is like the final scoring information. Row for each of the samples, we're going to be using the sum column, and that's what we will be using, and that's like the polygenic risk score for late onset Alzheimer's disease for these samples based on the scoring file. And this is how we computed it using the DNAnexus platform. Yay! So each of these is a sample. And I'm going to also show you how you can find some information out these samples using the cohort browser. So here I just organized that PGS catalog calculator scores neatly here, and what you're seeing is that this is the CSV file for the first 40 rows and then the last as well, going up to 487,409 samples in my case, and then the beginning ones for the polygenic risk scores for late onset Alzheimer's disease. And we focus on the sum here, and each of these is a given sample. So these are all for the samples, one row for each sample. The FID / the IID, they're the identifiers here. So one row for each sample, and these are the sums for those individuals. And essentially, these are the polygenic risk scores for late onset Alzheimer's disease load. And across them, the minimum score across all these individuals is, very negative, which means decreased risk of late onset Alzheimer's disease. And it's negative 1.89376. So they have a very low risk of late onset Alzheimer's disease, the smallest number relative to other individuals. The maximum here is 1.324. So the individual has a very high risk of late onset Alzheimer's disease relative to other individuals. And it'll also be helpful to maybe scale these results relative to the other samples, just to see, how it all compares. But this is essentially what we were wanting to calculate for a given individual. What will be their risk score for having late onset Alzheimer's disease, given that we found 21 variants that we think could be predicting late onset Alzheimer's disease? In this example here, this cartoon example, we found these 21 variants to predict late onset Alzheimer's disease, and then we have the genotype information for these individuals. So how likely is it that the given individuals will have late onset Alzheimer's disease, essentially? And these are the scores. So next, I'm just going to show you how you can analyze some of the PGS catalog calculator results. We have the predicted risk scores for late onset Alzheimer's disease in our example for those individuals, and now we want to just understand, who are those individuals, because they're just IDs, right? So I just want to show you that we can use the UK Biobank cohort browser, and I'll be showing other videos for how we can also extract phenotype information on individuals and kind of map them to these polygenic risk scores. But I'll just show you a very simple user interface to kind of see on the UK Biobank cohort browser just different attributes for individuals, and we can find those attributes and phenotypes for the individual samples, and then kind of map those we can map those genetic attributes or metadata to each other. So for the samples, we've predicted the polygenic risk of those individuals having Alzheimer's disease, those samples, and using the UK Biobank cohort browser, we can uncover attributes about those samples. I'm going to be showing you the gender one. So we can see, do females, do they tend to have a higher risk of late onset Alzheimer's disease than males? Those are some analysis you can do. People, family records or whatnot. There are all sorts of things you can look at based on what kind of analysis you want to do. But even smoking, other things, but I would recommend you focus on different, there's so many attributes about the people that you can look into. And you can just map them here and determine, what those polygenic scores mean, and I'm just trying to show you how you can uncover some of the metrics for those samples, because we have that file on the scores for those different samples, and you might want to know, who are those samples, or how can I find information on those samples. So that's what I'm going to show you right now. So that's kind of putting a score to an attribute about a person, potentially, that you could use for intuition and to make better sense of the scores, because they're just a bunch of IDs about people and scores, and you're like, what does that even mean? Which people are females and which people are males? You don't know. So I'm going to show you that. So I want you guys to see that each sample really corresponds to an individual, and there are a lot of phenotype information that you can gather for those individuals, because right now, what we've just seen is that we have this information for, the scores for each individual, what the risk is of developing Alzheimer's disease, late onset, based on the score file that we had. We've given a score file of these SNPs. What is the risk factor of, some SNPs are contributing to Alzheimer's disease progression. They have a positive effect allele, effect weight, and others are protecting against late onset Alzheimer's disease, and they have like a negative effect weight. And so we've kind of calculated and computed these scores for these individuals, with these IDs. And in this case, they're the same here in my situation. But these really correspond to individuals. And I want to kind of motivate that. Like you may be looking at these samples and thinking, okay, well, what does it mean? We've got some names here. Like let me just scroll down a bit and show you, like we've got like different ID, they've got different, like some of them become positive at this point. And I think the negative, it's like ambiguous alleles and so on. I mean, ambiguous gender. So, but what does it correspond to? Like, what does it mean? So what I want to show you here is that there's a cohort browser. So we're going to go here to dataset. So again, you will go here to the dataset record. And this is called the cohort browser. So go here, click on dataset record, and this will take you right to the cohort browser. So this is the cohort browser. So you can browse the cohort of individuals and then kind of map the IDs to the ones that we have, like the 487,409 that I have, and that number may be different for you. So please don't worry if you don't get my numbers. Also individuals, they often, like withdraw their consent for UK Biobank data. So they often say, okay, actually, I don't want my data to be used for research anymore. So right now we have like 501,961 participants in my resource, but I've also talked with other people and they have like different numbers in their resources. So again, please don't worry. I was like, oh my gosh, why do I have like different numbers? But it's okay. So what we have here is that we can kind of browse this filter, right? Like if I want to see participant information, participant ID, I can kind of just like get that cohort filter here. It's not null or something. Well, let's just say, like, for instance, I just want to population baseline characteristics. I can get this one here. Add cohort filter. It's not null, or is any of, like, maybe I just want to get the females. And then we'll be updating this here. So 273,051 participants. And you can see these are the participant IDs. So for instance, I could see if this guy is found in this over here. Yep, this participant is found here. This female is found and has like this negative polygenic score, so they have a decreased risk. Just out of curiosity in my data, I can just kind of look at maybe just the min of the score. So I tend to use the sum. This is the minimum score and the sum value here. And I can also, I took the minimum of this column here. And if I want to just see the maximum, this is like the min sum. And the max sum is equal to max of this, just out of curiosity. So some individuals have a higher propensity for Alzheimer's disease. So they have really positive numbers. So this just shows, for instance, like I can look at the female participants, like this number here, this lady. Let me actually just go up to the top. And let me just do view. freeze top row. And now let me go here so you can see it. So this lady has this negative score, so has a reduced risk of Alzheimer's disease. So good thing for her. And you can see the next one is this 25, which is this lady here, who has, unfortunately, an increased risk of Alzheimer's disease, late onset. So there are many characteristics that we can use for this filtering. We can add filters. They have participant information, their population characteristic, baseline, age of recruitment, month, year, email access, date of last personal contact, assessment center, biological samples, genomics, all sorts of things. And you can use these to kind of filter your samples and kind of make more sense. I mean, kind of gather like information and metadata on those individuals, kind of, in a way, putting a face to a name, but a face to a number, but you won't, it's very de-identified, so it's going to be really hard to find out that information. But you can kind of gather some baseline characteristics, like do females tend to have like an increased risk of late onset Alzheimer's disease versus male, just some basic things, you can include in your study. But this is just kind of showing that you can add all filters that you want and map them to those IDs that you have from the aggregated scores, and then kind of get some insights about your individuals in your cohort. And I just want you to see that you may just see a bunch of numbers and negatives, but each of those is an individual, and each of those individuals has certain characteristics. You can even focus on ancestries, European, South Asian, and so on, like Middle Eastern, all sorts of things. But that's just showing you like that the cohort browser is there and you can use it to filter and identify more things about your samples. So this just also shows how we can use the polygenic risk score distribution, the scores that we get, how we can kind of work with them. Providers, healthcare providers can use them for clinical decision making, if little personalized information is available. So it's good to look at individuals who have a decreased risk of the condition and individuals who have a high risk of the condition as well. So it's always good to look at the extremes. And it's good to encourage patients to do what has already been recommended by their healthcare provider, the physician, and nurses. So there's a concern, though, that if an individual is considered to be at lower risk, they might just dismiss recommendations to look after themselves. Like if you are considered to have a decreased risk of coronary artery disease, maybe you start being more neglectful of your health, or having more junk food, which is not great. So sometimes it could be potentially a false negative, where somebody's thinking that, oh, you won't have the condition, or you're lower risk of having the condition, but maybe you might end up having it. So that's always a concern. And then if you are found to have or predicted to be of high risk, then maybe this could encourage you to strictly follow recommendations, eat healthier, exercise, and so on. And then this can be helpful to find high-risk patients, especially these individuals that are typically missed by traditional risk assessment. So if, for instance, there's certain factors that are not really easily measurable, certain risk factors for certain diseases like Alzheimer's disease or whatnot, and it's useful if you have an increased risk, then there might be something there, and maybe it's worth checking out or doing more investigation as to why you are at high risk. So there are many applications of these polygenic risk scores, and just in general polygenic scores for trait, like why are certain individuals smaller, shorter in height, why certain people are taller, and so on. And just for you guys to know, there are other cohorts with genotype information out there, like All of Us research program in the US, of which I'm a proud participant. Yay! Woohoo! So as they say, diverse DNA data improves disease risk scores for more ancestries, so we're able to better predict, disease risk for individuals. So just something for you guys to keep in mind. So we were focused in this video on UK Biobank, and there are other cohorts that are out there, and more data is being made available. So I do encourage all of you, if possible, to try to contribute to these DNA sequencing studies and participate in these cohorts if possible. It'll really help make a difference, believe it or not. Hi everyone, so I just want to say thank you for watching this very long video on how to basically work with the DNAnexus research analysis platform for UK Biobank, and all the different steps, especially for running PGS catalog calculator. It's been a very long video, and I'm thankful for you guys for watching it and giving any and all feedback that you have. So just thank you again for watching it, and in the spirit of being in the UK and talking about the UK Biobank, I'm going to say, please like, please subscribe, and please share this video, and reach out with any and all questions and comments that you have. I really enjoyed making this video, and I'm hoping it's going to help all of you. It's something that I'm also learning, so please do bear with me. If you do find a lot of better techniques for doing things, please do let me know, and please reach out and share with the community. I know a lot of people are shifting from working on the local computers with UK Biobank data to the research analysis platform, and it can be a bit overwhelming and daunting. So I just hope that this is really helpful for all of you, and it also kind of shows you that if you don't have certain technologies that are already in the DNAnexus platform, like PGSC Calc at this point in time does not have an existing tool package, how you can kind of upload it, download it when running the script, and then run it within the script. So that's what I was showing you there. I just hope it's really helpful for you. So if any questions, again, as always, please do reach out, and please do share this video and let me know what your thoughts are. Okay, so that was my British attempt. Anyways, thank you guys. Little Bear says thank you as well. The spirit of Liverpool. Walk on, with hope in your heart. So with hope in my heart, I hope that you will use the video to help you with your research and hopefully help advance the field of public health in your own ways. Thank you. Thank you.